chrysogorgid with a red face, squat lobster. I now know.
You're uptight, yeah. You're all some big polyps.
Yeah. It's Maxim. These little green things.
Yeah, this is 14 mountain.
What else we got? We got a rock pen.
Oh, this is like the rock that keeps on giving. Everywhere we look, there's yet another little thing.
There's a little hermit crab down there too.
So, good morning, everyone from the ship. We are diving on board the RV Falkor with the RV Falkor with ROV Sebastian. We're currently at 1,491 meters, wow, which is dead on for the sonar. And on an unnamed seamount in close proximity to the Winslow Reef area. And this feature, we just mapped the summit of it last night for the first time, and it's only 200 meters deep, so we've probably got a mesophotic community on the top of this feature, and we're going to work our way up the western side of the seamount, all the way up into the mesophotic depths.
Starting off this morning is myself, Alexis Weinig from USGS.
And I'm Brian Kennedy from Boston University.
We'll get going this morning and then pretty quickly have a shift change, so you'll hear some new voices in just a little under half an hour. But we can carry on. This little rock had more growing on it than we originally thought. But we will look for a rock to pick up as quickly as we can. Yeah, we're already seeing a mix of carbonate and more crusted rock here.
It's coral too.
Oh, I didn't see it. This is our 12th dive on our current expedition. Technically 13th, but we are going to be diving for a couple days here on Winslow. Hopefully a couple different dives, and then we'll actually be leaving the US EEZ and heading back into ABNJ waters as we make our way back up towards Hawaii.
Test 1, 2.
We'll have a couple dives along the way, so it won't be. We will not be saying goodbye after Winslow. We'll be saying, see you later. Brian's laughing at me because I'm being ridiculous.
Test.
Pretty heavy sediment coming down, huh? Yeah, there's lots. It's funny, we saw that one rock had a lot of corals growing on it. I thought we'd start seeing them more quickly.
Oh, it's gonna totally mess up our life now. Oh, that looks better after. I'm always happy on that.
There's a crab. Looks like a big chase down crab up in the top kind of left.
Oh yeah.
You get back on the line or just follow the rock?
I say how far off? I say follow the rocks. Just go in that general direction.
30 meters. Giant squid. South. But, okay, they'll show
I'm trying to keep an eye out for anything that looks.
movable.
Yeah.
Movable.
So today we'll be looking at basically the same kind of goals of the dive we've had previously, of looking out for examples of corallivory and spongivory. And today, because we'll be transiting through the shallower depths, we're more likely to see starker transitions between community composition and where those transitions occur as we get into the shallow, 1,000 meters and less, and especially when we get at 500 meters and less, you start seeing much more rapid species community turnover, and so that's one of the things we're paying particular attention today to.
I'm also personally interested. This is a seamount that has, like Brian said, a really shallow. The most shallow point is up to 200 meters. So we often see the larger colonial scleractinians in the more shallow range between 1,000 and 500 meters, and started to notice that we have larger communities when there's a productive surface community. So I'm interested to see what we find as far as how big those communities are, if we see large aggregations like we did at Howland and Baker, or if we just see kind of individual colonies like we've seen at some of the other seamounts. So I'll be trying to pay attention throughout most of the dive, but really looking back in around, well, maybe when we're actually back on shift the second time, we'll be in that range.
So we're gonna perch here and see if this rock is gonna come with us or not. We're trying to get a rock real quick on this dive because we're expecting to get up on the carbonate cap of the seamount very quickly. Like, I can see a couple rocks that look like they've fallen down already that are probably carbonate. So we want to make sure we get a good basalt with manganese encrusting on here for the geologist before we get into the carbonate.
Yeah, it's 7. 16, 7. Yes,
Nope. Bad rock.
Bad rock.
Bad rock.
Look good?
What do you think? I think that's probably...
I mean, I don't know.
Well, okay, yeah.
Okay, wait, let us. I mean, I'd rather take it than not take
Really
it.
went, it's kind of soft, wasn't it?
Yeah.
Yeah.
It was all crust.
Yeah, it was all
You
crust.
want me to release it? Back to the water?
We might as well bring it up.
I feel like we should take it just in
Yeah.
case we don't. Okay, so
Bridge ROV, recall us?
Can you just rotate the wrist?
Okay, thanks, man.
he's on the.
Here, thanks. I heard something as well, so.
Can you just rotate the wrist for
Alright, we need to go left. Rotate the wrist.
Yep.
Yes.
So we can just see, kind of help us ID it later. All right.
You like secular clubs there?
So at this particular site, we're actually not the first ones to map. This is a pretty well-known area, known as the Winslow Reef complex.
Alright.
There's partial mapping data before we were here, so I'm not sure if we have plans on trying to name this particular seamount.
But naming a feature is actually quite onerous.
It is.
So it's not something
We
you
do lightly.
Ooh, wow, look what you guys think.
I know. I mean, obviously, we didn't watch together too long now.
You should be clear of everything now.
What do you call that shape? Rectangle? But I think it's split into
Oblong cracked.
oblong rectangle.
Alright, and you're clear as far as I can see.
I don't think it's an oblong, like type.
3 bucks. There you go.
3.
No. What number is this?
Oh. Tim, can you let us know the next sample number? Sorry, I should have looked before. I didn't. It's in the dry lab.
You probably hear us stumbling over what to call this feature a little bit because there's 5 or 6 seamounts right here in close proximity that all kind of get near the surface that at different points have been referred to
467.
Okay.
Have been referred to as Winslow Reef
If
or
there's-
not at one point.
I don't know
So
if
we're
there's
still
one
kind
here.
of trying to figure out exactly which features are Winslow and which ones are the greater Winslow
We'll
complex.
keep going,
So
but yes, we
you'll
would
probably
be
hear us use a
interested in
couple
another
different
rock.
terms for this feature today as we still sort through
I
what
mean,
we're going to call it
I
even
liked
operationally.
but it's not about me.
You're in that chair. I'll allow you right now.
What do we say? 467.
Try that rock.
Is that big one?
Is that too big?
Yeah, pretty big. Yes.
Yeah, it looks like it's gonna get half. Yeah, it's good. Alright.
Let's keep going.
I'll make my way back.
Yeah.
Because there's nothing in front of us here.
Right.
Really?
Yup, that happened.
Not gonna sound like man.
Oh, this is interesting. Very much, very pavementy. No more.
Very low amount of steak.
Like the Gorgio?
Yeah.
Flexoid.
I'm gonna just.
There's two right there.
Yeah.
Which one do you want? The first one, the second one?
I'll look at the little yellow one first.
Yep.
I can probably just snap zoom, so we don't have to come all the way down.
No.
Maybe a better lighting.
I'll get it.
Alright.
Oh, yeah, you got lots of time. I'll split that later.
Alright, you happy?
Yep.
I'll put you in manual ISO and iris now.
Okay.
I'll change it.
Thanks.
Do you want to close look at that one-on-one pass? I didn't know if you were happy or not.
It's okay. We'll get a look at another one.
I can go back.
No, it's okay, Chris. It's totally fine.
No.
It really is okay. I'd rather us keep going. It's just hydroids on a stalk. I say that like it's a bad thing. No, I'm just saying, like,
Yeah.
it's not a bad thing. I'm just
My fast is still set. So you're fine. It's just, no, I had to send that away. And you came there.
Where that color is. That's good, we can keep going. Looks like a sea cucumber. Or maybe not.
I'm not sure yet.
That's a
That's
Dumbo.
I'll sit down and look
We
at
got
it.
a Dumbo octopus. Look at this little siphon.
The cephalopod.
What? Oh.
Look at further? Or are we over Dumbos now?
We haven't seen that many Dumbos,
That one
but
looks a little different too.
we can take a look at it for a little bit. I don't want to disturb it too much.
Like it because they put these sort of 10 seconds.
I know, I know.
When they're moving.
Oh, oh. Can we take the lasers off? Thanks.
Devices.
Are you setting there?
Yeah.
Okay.
No, but you're fine. You can.
It's just a little over 10 centimeters, this Dumbo octopus. Might become a little more active as it senses our presence, but seems to be pretty relaxed on the seafloor. They are very cute. Look at that big eyeball.
Has short fins.
Yeah, this looks a little different.
You know, they're kind of narrow too.
It almost looks like kind of a cross between a bobtail and a Dumbo.
Yeah.
Maybe it's part of that posture, maybe, but
A siphon.
it's jet.
Okay, I think we're good.
We do a flyby on the sponges.
Yeah,
that
can
way.
we look over this way as we head off? I'll just come in on this sponge right here, yeah.
It's living on.
It's one of those ones where it has like those holes in the base, you know? Then a couple of anemones.
What kind of coral is that?
I'm gonna look right here, Ben. There's a coral on this rock.
Thanks, RV. Update midway.
New waypoint. Thank you.
looks like Caligorgia to me from NOAA.
Yeah.
Has an ophoroid on it.
It's like a young Caligorgia.
It's okay. Oh, there's just you see that there were a little
Oh wow.
chrysogorgia
no,
back
I
there.
didn't see
And
that
well,
at all. Sneaky corals. there.
yeah, today they're all kind of blending in. Okay, we can keep going.
Anna, you want to take over?
Yeah, I was gonna.
Yep. We have.
No, he's just even registered.
If you do that, it's 1 hours, you go tell them.
Good morning.
Hey, Ted.
How we doing this morning?
Good, good.
All
They're
right.
a little bit empty, so we need to like fill it up.
We're a little empty.
Yeah.
I see that. We have a clean board here. Well, let's get busy. Let's get busy. So a lot of these corals may be a little translucent. We gotta take care of them. Look at them.
Victororgia.
It's the second one we've seen of those. May end up sampling one of those later. Just a quick snap zoom here, Ben. You can just keep flying. I'm just gonna go in like this. Just take a look, make sure there's an oaf on there. Yeah. That's what we've been seeing. That's good. So, welcome everyone. My name is Tim Shank. I'm a deep sea biologist from the Woods Hole Oceanographic Institution. I'm currently at 1,466 meters on an unnamed seamount inside the US EEZ, just north of the Winslow Reef complex in the Phoenix Archipelago. And I am with
I'm Anna Gauthier. I'm a graduate student finishing my PhD at Harvard University, co-mentored by Randy Rogen at BU and Jonathan Kagan at Boston Children's Hospital. Very excited to be here with you this morning and talk you through this dive.
It was really interesting when we first landed. We came across a big rock, and there must have been 5 or 6 corals on it. And we've been sort of the group we've been seeing in the northern seamounts, but none of those did we see yesterday. We were already in a different zone. We dove really deeply yesterday, 3,200 meters, and seeing a completely different type of coral fauna here. So we're on the west side of this seamount. We're going to drive a straight line, more or less. Let's see how far up to the top we can get. You probably already heard that it comes within 200 meters of the surface. Just mapped that last night. So, yeah, hope you'll stay with us while we discover some really neat things today.
So what kind of things were on the rock this morning?
There's a yellow plexard that we hadn't seen. We saw Metallogorgia that was up there. I think there was, what else was there? Is there a Noah there? I mean, there must have been, yeah, 5 or so corals. I don't know if they were noted. Here. What's that? Does that always happen?
It's from yesterday's?
I guess so.
Oh, I don't know if it's always there though.
Yeah. So far we've collected a single rock, which was mostly crust. This manganese crust that we see, but we should be seeing, like there, bits of carbonate. This is a carbonate cap seamount. If you followed along with us, we've been mapping seamounts, many of them as we've gone south here toward the equator and beyond the equator to the south, and we often have these volcanic seamounts that have these ridges on them that extend away from the top of the seamount, these old volcanoes, and those are signs of how they're erupted, those ridges. Basically dikes that formed when the volcano erupted, and now the rest of the basalt seafloor has eroded away and left those relic ridges there for us to dive on. Don't see that on this seamount. This is a different morphology. It's more of a carbonate cap, smooth, kind of like what we're seeing right now. It's going to be a gentle slope. We will see rocky bottom. It's hard bottoms, which these corals and sponges love. So we should be into it. Here's some rocks coming up now.
Stalked crinoid. Who would have thought?
Mm-hmm. That was also there on that first rock.
It's a brilliant color though. Now I gotta, is that a sponge? Oh no, that's a Venus flytrap anemone to our
Yeah.
left.
And maybe proisocrinus in front of us in that.
And a fish.
I think we're good. We can keep going. So usually at the beginning of the dive, we will sit down and look at things we haven't seen before and make sure we document what's here. And as we see them more and more, we stop less frequently because they've been documented. So things we know, we've seen, and we tend to pass by or do what we call snap zooms, quick zooms, just to confirm an identification, and then we keep going.
Like the sandy bottom here this morning.
Yep. So we landed west of the top of this seamount, and we're heading east right now. Sometimes you see the ROV like this crabbing along, but it's still going. It's actually heading 112, which is sort of heading east. So we have a watch circle. We're certainly tied to the ship with this ROV, and it has a sort of a watch circle that it can go out and explore and not be pulled on by the ship. So we often go to the left, to the right, if you will, of a line to explore a little more. So you'll see us crab left and right. But well, on whole, we're moving to the east for the course of this dive today.
I'm just going to do a quick weight balance.
Yeah, sure. Yeah, sounds good. All right. You want me to?
There we go. Realistic.
So it seems like every time that I look at the horizon, the white balance is going to drop out, right? Do you feel like that, or I'll try to hold it here?
Just need to do it periodically. Just
Yeah.
to,
All right.
keep on top of it.
Yeah. Okay.
Seems to just drift
Sounds good.
for whatever reason.
Looks like we get a sea star at the top.
Yeah.
We might need to start going ahead.
Hey Ben, let's take a look at this. Sea star. We tend to stop for sea stars on this expedition because
I think she just came up with another catchphrase for our
what's that?
stop for sea stars.
Yeah.
We'll stop for sea stars. will break for sea storms.
It's like, yeah, well, these are predators on corals and sponges,
Yep.
so we want to document all those here, along with other things that live on the corals and the sponges. Ben's going to set us down here, and we're going to take a quick zoom here. Good? Okay. It's a goniasterid sea star. Yeah, I don't, wow, every time I guess Seriaster, but it's quite not that. So, but it's certainly a goniasterid out here on the sand. If I go any further, that's it. Okay. Just get some pictures here. Other way. All right. Okay, everybody, that's good. Thank you, man. What's that? Okay.
Fish off to our left. It's always eerie to me when they just don't move.
Yeah.
It's pretty normal also.
Always wonder if they're oriented up into the current, if they put their face. They have modified fins for standing on the seafloor.
Yeah.
Often they have tendrils and whatnot. These are just. You can see a barb coming off the top a little bit, I think. Is that a little bit, those little things?
Tripod.
Are you feeling much current today? No. I don't think there's anything here regarding current. Nothing. I don't see anything here. Okay. Let's go over that way and take a look at it. Just a stalk there, I think. Another crinoid there. The rock they picked up wasn't much of a rock. It was really just some crust. I
Just
wonder.
a piece of crust, yeah.
I wonder if we should consider
I think if
finding
we
one.
see a good specimen, it
Yeah.
wouldn't hurt.
Yeah.
Here we go.
Here we go.
This is a nice little
Nice
rock.
job, Ben. Let's see what's up here. There's a lot of metallic gorgia coming in here. See, this is what the first rock looked like too. Can we look at either one of these here, whichever one is easy for you? See a yellow flexaurid type down there. Brictogorgia in the purple with a asteriscomatid ophiuroid that we see. And then,
Bathopathes down this
oh, you
morning.
saw a Bathopathes? That's a black coral. Be our first of the day.
I got bamboo whip.
Yeah, it's gonna fly it here. I'll just go in a little bit here. So these Metallogorgia corals, these are in the family Chrysogorgidae. And they are fantastic because they have these brittle stars in them. And this one is Ophiocreus. There's something else in there. Is there is there something red in there?
Looks like maybe a couple other things.
Not too far in, Ben. Here you can.
What is that?
I don't know. I can't tell if there's something else in there. It looks like there's
The red, yeah.
There's also this thing
The
here.
green, like gall.
Yeah, that looks like it might be an egg or some sort.
Looks like a
Those are
I'm gonna
for
hold it there,
sure
maybe.
eggs on the O flag.
that's interesting enough we can park. You want to park them?
Yeah, if you can, that'd be great. Thanks.
That could be one of those egg sacs you often see broken open.
Hello,
They're
sir.
beautiful. Susian.
Yeah. Yeah. You wanna?
Compose something?
Yeah.
Yeah, why don't you go left just a teeny bit?
Well, he's parking
Oh,
right
yeah,
now. He's
right.
let him set up first, then we'll. Actually, but
I mean,
come
we've
out
got a
for-
lot of those, but they're so pretty.
Well, we've got a couple things. We have eggs in this thing as well as a rounded gall like. Could be an egg case, but it's
Really?
yeah.
The crab, obviously.
Don't know.
Could
We
be
have
something
could
else?
be fish eggs. We've seen that on these corals too.
The Lorax.
He's got a perch on one. Nice. Thank you. How we looking? We too hot?
A little too hot. Yeah.
I thought so too. Yeah, I don't have
And I was
much.
thinking if he went a little bit left, you could sort of frame that up where the anemone is facing you a little better.
That means coming off the rock and repositioning.
Oh, okay, then never mind.
You know what I mean? That's.
No, it's very nice. Yeah, zoom in on it a little bit.
I will.
Yeah, that's great. Right there. Back up just a teeny bit. Get that anemone in the frame.
Yeah, right. Just trying to. Come on.
Right there. Yeah, that's nice. Just hold that for a sec. Cool. Yeah. Maybe zoom in.
Oh, that was too much. Too fast.
Can you see the guy? No, you're okay. I generally don't use the zooms because they're too abrupt.
They are. I can.
That's kind of interesting. So it's a sea star. I thought maybe it was. Is that
So there's a
green
typical-
glob? Oh, I see the eggs. Right.
Right. You see the eggs and the green glob. It's.
What's the green thing?
I don't know.
We don't know.
I don't know.
It kind of looks like those things you see, the egg sac that's like broken open, that almost looks like a
Well,
hornet's nest,
yeah,
like
and
texture.
In the past, that's been a Dumbo octopus
Okay.
egg. I think.
But
we-
I
you
don't
we
know.
think,
Yeah,
right?
it's hard to say.
So I think we may have to do a collection here.
Dumbo octopuses just have one egg, or it's an egg
Well,
sac.
the-
1,000.
no,
Oh no, they have eggs. Just right. Octopuses have the little eggs. That's
yeah,
right.
a deep sea Dumbo octopus will carry 2,000 to 3,000 eggs.
Yeah.
And deposit one
Yeah.
on a coral like
And they
this.
deposit them under,
But I've never-
under rock shelves and stuff.
I've never seen them deposit it on a metallic orange.
I mean, that's a weird. It doesn't look like it's because it's at the juncture of like
Yeah.
4 of those stems, two
Yeah.
or 3 of those stems.
Right, or the branches.
That seems a little weird. That
Yeah,
it's more like a growth around them.
Yeah, like a gall.
but those look like fish eggs to me. Too.
On the right, you mean?
Yeah. The way they're put there too.
It's beautiful, though. I mean, that whole
So great.
scene.
I'm gonna suggest a collection. I'd like to see this. Yeah. So, we want some more of this, Eric? Are we
No,
good?
thank you.
You're good.
Yeah.
Okay.
Hold just for a second.
Do you have the lasers on? These get the size for here. That might be a good idea. Sorry.
Oh, no, you're good. I just wanted a beauty shot for the.
Oh,
Oh, I got it.
trying to see what. There's one.
I'm wondering if we like from the other side.
Oh, yeah. Okay.
So I can see
Yeah,
the
so you can see.
nitrogen in it,
Yeah.
chop
Yeah.
it,
That's fine with me. If you want to come around, that's fine with me.
I just think it'd be.
I'll come all the way out.
less damaging.
Yeah, I agree.
Shake your camera.
Yeah, I'm just gonna straighten it out so you can. Okay. Nice job, man.
Completely 180.
Yeah, I think so. Look it back the other way.
And that's the bath that Becky says.
Oh, I see it. Yeah, right. So these Metallogorgia corals are really fascinating because it's been documented that or at least published, that there are these ophiuroids that live inside them or with them for life. That they, like many of the corals that we see that have ophiuroids in them, that they may be come on when they're sort of both juveniles or both young, if you will. It's a relative term for corals, but and then as the coral grows, so does the ophiuroid. But these corals, in many cases, can be hundreds of years old, if not thousands of years old. So I think that the ophiuroids, I don't know if they would be more than 3 years old, ever 4 years old. So I always wondered about that, right? That we should see in a 1,000-year-old coral, we should see what?
1,000-year-old.
No, right? So we don't. No one's really tested that one, right? No one's really said that before. Now that's so far-fetched.
And how do they get colonized afterwards
so-
once they do pass on?
So why don't So if corals were to be
Maybe.
1,000 years, let's say, then why don't we see them empty of ophiuroids once in a while, if they are dying, or the crabs? Can I go over there?
Just cut out the section. So you use the other stub.
I'm gonna have to turn a little more.
Do we ever
Are there too?
see multiple astroschema on?
Metallic gorgia?
Yeah.
Never.
Which is so weird because you
Never.
think, oh, well, maybe, you know, like it, there's babies, so it's very possible that like
Yeah.
succession, but
Yeah.
you never see it. That's one of the persisting mysteries of deep sea.
We've been so fortunate to have enough observations, even in the Phoenix Islands region here, that we come to expect certain things on things like the Bathypathes that you see across the way there would normally have 2 Europtychus crabs in it or 1 Europtychus crab. It'll have a polychaete in it. So we come to expect certain things because we see these relationships. So when you don't see something, like there's nothing apparently large in that Bathypathes, why is that? Is it a depth issue? Is it that whatever the associate is has died and gone and hasn't been recolonized yet? Mm-hmm. You have to infer a lot of things temporally that you don't have a snapshot of. We're only here for a few minutes, and yet we try to interpret what happens, over years. So we're just setting up on the other side here because I think it warrants a sample. We've seen a couple of these already. So we know there's a lot here.
It'd be interesting to look at the one next to it after this to see if there's any
I know.
evidence of anything else or if it's normal.
Can't wait to zoom. But you can see that egg case in there now.
I know, it's like glowing at us.
I think Ben's flying with the other camera, so I'm going to just do this. Get it a little bit tighter. The egg is smaller than I'm used to seeing.
Yeah.
And the eggs are usually on chrysogorgids. I'm speaking of the Dumbo octopus eggs. It's a nice view from underneath. You see it's more or less a canopy. This is going to be nice.
I'm hoping I can go in and just cut that straight.
Yeah.
Here.
You could cut it here. I'm sorry. Can you see?
Sorry?
You can cut it below the oaf.
Oh, you sure?
Yeah,
oh,
right
okay.
there.
Oh, that makes
That'd
life
be-
easier.
Yeah. Can I just get some images here before we do that? So it's great from this side. See if I can. Great. Are you doing the iris over there?
Yeah, just decreasing.
Yeah, just take it down a little bit.
Is that more?
One more touch down, I think, because the egg is so white olive.
Everyone sees it.
You right? There you go. And then I'll do more of a zoom.
Yeah. It's amazing.
Beautiful.
That's amazing. So we will hopefully we'll collect this and we'll get it and then we'll do DNA analysis on the eggs. And we realized from last time that we were in the region that these were snailfish eggs. I'm gonna come up a little bit. There we go. I'm just gonna hold that for a minute. It helps us to identify these things. That they rotate, we can see the certain polyps. And it was theorized for so long that there was just a species of ophiuroid on Metallogorgia corals, and I think we showed in 2017 there's actually 2 species that live there. All right. Go this way a little bit for a second.
Did you get lasers on this to know how big it is?
I did.
Okay. We going for a quiver or bio box? Probably a bio
I don't
box.
know.
on this one,
I think we're gonna see him grab it, and then we're gonna figure out where it needs to.
Okay.
Yeah, maybe a bio box. You're right.
Just to preserve everything. I mean, this is a really
Yeah, I'm all the way in now.
potentially valuable specimen in terms of what we can get out of this.
Yeah, it's such a smaller egg than I'm used to. Well, we'll see when it gets up. All right. I love it.
Before we put that in, do you want to look around, see what else you want to look at? That's an easy collection.
It is okay. Yeah, there's another one of these just right next to it here. I think I can.
Do we want to
Yeah,
try
right,
and
let's
do
look at
a
that.
quiver, or do we want to put it into the bio box?
I was gonna wait and see how big it is. What you think?
Because that's,
Maybe a bio box would be better to keep
Maybe,
it better intact.
I would say maybe that's true, yeah.
We got something walking down here too. Well, Hamalid crab
Oh, yeah.
with an anemone. Just want to zoom into this one and see if we have anything else like that in here.
Might have to come back to that one.
Might have to come back to that one.
Yeah, I got the L behind.
That's all right. I mean, that's fine.
There's a storm coming.
Hey, we're stable. That's good.
Behind.
It's okay. I just want to. I didn't see anything in this one other than the ophiuroid.
Back to the other side.
Yeah, I guess so. Why don't we go ahead and collect this thing and then we can see if we've cleared.
Before we did that, we opened the bio box.
Yeah, well, yeah, let's get the bio box organized. But I can hold it with the bio box.
Alright.
I love the little canopy these corals make. Their branches up there.
Hmm?
I love that canopy that it makes that it's sort of flattened.
I know, it's
But-
like from Dr. Seuss almost.
We had one down here where they were so flattened, it was almost planar, and it's not typical for Metallogorgia. They're usually like a cloud, sort of a round ball of little polyps. There's a bouquet.
That's a big one.
Yeah, it's a bio box.
Yeah. See me 468. Okay.
Yeah.
Like one of those trees you buy, like the trees.
Bonds on that. They're poaching out.
Like we're racking it, but yeah, you sit there and rack on it back. Each poach anyone?
Yeah, I can poach them out. Yes. Pushing.
Okay, let's.
I didn't know that.
Today's menu: seafood.
Okay, bio box 3.
Seamount, RV Falkor, Bon Appetit.
Yeah. So this is going to be probably an A, right?
I always check too with them.
Such a nice collection. So okay, so
Okay, so I'm gonna press sample In-situ.
yep, perfect. And that was 468.
On our way to 500.
So we can do like one.
Fabulous.
So is that A, Russ?
I'm calling that A.
Yeah, I mean that was technically
Right.
A, B, C, D, 'cause there's four sections in there.
Which side?
Or 3. That was divided. All right.
That was divided, and
Got
that
it.
was divided, so technically you got 1, 2.
Yeah, I call it A.
Yeah,
But
there's basically, no, you're right, there's 3 sections, just proportionally on here, it doesn't.
correct.
Okay.
That's right.
Great.
All right.
Thanks.
You may see this little writing people have done on our bio box that says today's menu is, and yeah, it's because we have something new that the ROV pilots have put together here. It's a coral feeder. We'll be showing that later in our dive. We've got some food in a pair of syringes. It's on there, isn't it?
This is the first I've heard of that. Sorry.
I think it's on board. That's why that.
That's great.
That's all we have.
It's full of food, ready, rock and roll, ready to feed the
Yeah,
coral.
it's got some squid in it, I think, or it's quite a bit of stuff in it. You can tell there's not much current here. Nothing's really moving away, right?
Do you want to go back to the sensors and clear this?
No, I think we can go. Let's go.
Yeah,
We're gonna see this
fly.
stuff some more. I think it'll be great. We got a Bathypathes here. We got that, I call that
We had a
Neomorph
couple
aster.
bamboos. We had a Victigorgia, Oxorid.
Yeah,
So this rock is very high in diversity.
you can see you can see that one of the major tenets of deep sea coral biology is that they want to get off the seafloor and get up into the current, and that's just a demonstration of that. They don't particularly like to grow or don't grow in the sediments and lower to the ground. They want to be up it.
There, are these corals that have taken advantage of that fact, and the sea pens, have a unique ability to anchor themselves where other corals can't.
Yeah.
And rock pens.
So we have about 120 meters to our waypoint, which is due east of us. Ships ahead of us moving east as well. We are into the cloud. Let's see here. There we go. Yeah. Wow, it really just sits here, doesn't it?
As you can see, we're
We've got some hard targets right in front of us.
What's going on?
What's that?
What's going on?
The water temperature here is 3.1 degrees Celsius. I'm not sure what that is in Fahrenheit, but I bet it's close to 40 degrees, so
Trying to clear.
38 degrees Fahrenheit.
since Americans do not know
It's
our
pretty chilly.
Celsius.
Well, I always work in Celsius. Don't ever think about the conversion.
Mm-hmm.
Yeah, it is interesting to note that these bio boxes and these quivers that we have for sampling are sort of insulated. They keep the water cold, which is extremely important for the animals and how what kind of shape that we'll see them in the lab when they come up on deck. We like to keep them as cold as possible to mirror their habitat, and so they'll often come up alive if we're from depths where we are now at 1,400 meters. They might not live very long, but then if we're at say 600 meters or 500 meters, they can last for weeks or months, just refrigerated. So the temperature changes sometimes matter more than pressure until you get to about 1,000 meters. Then pressure becomes really important for them as well. But the vehicle has insulated boxes for this reason. All right, pull up a little bit. So we have a sonar on the vehicle as well, which looks out and can tell us where there might be a hard bottom. So we're picking up little scattered rocks out beyond our lights. And so we can look at that sonar, look for hard bottom targets, and move toward them. And so A lot of the pilots do that, but right now they're sort of scattered, like what you're seeing right now. That way we can see if a wall's coming up or something like that long before we get to it. So.
I'm ready to get to some hard bottom.
Here we go.
It's an interesting looking rock right there.
I think this is a little bit mixture of carbonate here. I mean, this slice here,
Yeah.
this roast beef slice.
Yes.
So, what do you want to call it? Or should we say filet mignon? What should we? You're right. And it looks like so much of this is weathering of the rocks too. You see these deep, craggy pits and whatnot. Just been weathered over time.
There's so many factors that play into whether there's colonization or not. Like, we've thought that these textured rocks are really important, but obviously current, everything else. You know, it's not just the surface of the rock.
No, you're right. There's depth dependence, and there's temperature issues. There's
A sea pen in
flow.
the background there. Can't really tell. It looks like a peduncle.
It does. Good call. They saw that earlier today. Actually, they saw another sea pen earlier this morning.
Is that a rock?
Another octopus. You want to go look at it? Ben, let's take a look at that thing.
Fly by.
I think it might be another octopus.
I was like, well, it looks
It's over
round.
there.
Maybe we should.
Although it could just be an urchin.
Yeah.
Seeing some things that are on the bottom here. I'm gonna zoom in. You keep going, man.
Yeah, definitely. Those are really cool looking urchins, though.
They are. I'm forgetting the name right now. It's like
I know. I
it's
want to
like-
say like pillowcase or something, but I
No,
know
no,
that's not
there's
right.
something else. No, it's like a corosoma. I can't. No, it's not it. Oh my gosh. Something soma.
Still
Write
early.
that
Get a
down.
beer with us, folks.
Yeah.
Oh,
It's a Venus flytrap with
a Metallogorgia.
Metallogorgia
Growing
next to it.
on a Metallogorgia.
That's
That's
what it is.
crazy.
You see the ophiuroid on the other side.
Mm-hmm.
That's great. Yeah. Now we can keep going, Ben. Keep looking at that. That's a good shot. Yeah, these Metallogorgia here are really more planar than. Tilt up a little bit so we can see more. It's a big slab here. Looks so much draped with sediment. Seeing maybe the carbonate edge here. You see how they've been weathered out. I don't know the sea level history here, whether if this was when we get up shallower, if this was exposed or not.
It's so funny
Where?
how our
You see something?
I can't tell what that is, or if it's just my eyes playing tricks on me.
Yeah, Ben, can we go down and look at that? I'm sorry, they are so transparent or translucent that. Oh,
Oftentimes
yeah,
it is my eyes playing tricks on me. That's the thing.
I'm glad you're spotting them though. I can't. You sit
Yeah.
here and just. You sit here and
Yes, I
just.
see the crab.
Let's take a look. Yeah.
You see it, right? The little
Little
pink guy
chryso?
in the middle. He just moved.
Yeah, I see the crab more than I see the coral.
Yeah, it's
But
really
that's
transparent.
that's me. Most people are looking at the corals, and I always am looking at the.
Well, you're starting to convert
Good?
some of us, I think.
Ah, that's great. That's great. All right, there's some lasers there. So they go in. Sounds like a bottle brush. That's a different crab than what we saw ordered today. We saw a different Europtychus crab. It's in the family Chirostalidae. This could be a nice shot.
Does he only have one big arm? I can't tell.
He's got two.
He's got two.
Yeah, he's in good shape. But that looks different to me. I don't want to get a collection of it. Right? You can do it just below the crab. Is that okay? Are you set up?
Sorry, you get a quiver?
Quiver, yeah. It's pretty small.
Pork
You
chop?
can forget you
Okay, I'm gonna just stay right here for a second.
It's pork chop.
I'm gonna take the iris down a little bit. I know. You're good. Go ahead, take it. Yep, you got it. That chryso has really fleshy polyps to it.
Yeah, even though it's
Maybe
clear,
something.
it's like the one yesterday. I don't know if that's something we'll continue to see or just chance.
You wanna take photos first thing?
Yeah, can I go in one more image?
Yeah.
Just a few more seconds. Well, I got it. Here. Sorry.
Look at how each one of his legs is like.
Well, they've got these hooks. There's modified hooks on the ends of each of their legs. You can see one there in the top that just fit right around the size of the skeleton. This is a co-evolved partnership that we're just trying to understand how this species of crab only lives in a certain group, like this chrysogorgian corals, and nowhere else. So how it finds these corals, we don't know. How they grow together, I have no idea. But this is what they do. Whether they feed off the coral, whether they grab scrape things off the coral, the particles that are landing on the coral. We've seen one crab feeding out of the polyps of one of the corals, but not the coral itself. So, yeah, this is spectacular. We don't always get this kind of footage to see the crab like this, but I can see the long rostrum, the real developed eye stalks for the eyes. Yeah, Europtychus.
Well, it's so clear.
Yeah.
That's part of it too.
It's unusual.
Chrysogorgians usually a little more color.
Yeah.
The crab almost blends into. It's like the same color.
So Taylor also have to go.
All right. Okay. You got some snaps and all that.
Yeah.
Thank
And
you.
some beauty shots.
That's great. Thank you.
Can't do
Yep. Thank you. Yeah, those are great. Okay. Now you can see how small this is.
Yeah.
This crab's about the size of a quarter,
Mm-hmm.
a U.S. quarter. Or more like a button on a coat.
Okay,
That's great. That's. Oh,
Down.
wait.
Down.
again. Just keep your hand on this stuff there.
I see.
Trying to find it.
Just come in low there. Yeah. Oh, it's beautiful. Great. No, that's great. That's great.
Right there. It's gonna get just close up there. You
No.
should see the clouds.
Still there.
Maybe we can go off. Get out of that.
Those corals are so wispy; they are easy to float or tumble.
Can you find the camera, please?
Come up a little. You got it. You got it.
It's fully locked in.
You can go. Hold on, that's just how it is. It's over there. See? Sorry. No, no. That's it.
Top right.
You see it?
I think it's there. Right, right.
Straight ahead.
Hold on, yeah. Just remember the no connections,
See it?
Oh, no, he's gone ahead.
I wasn't sure if it was right there.
I thought we see something here, but
Yeah.
maybe it's a burning on the screen, sorry.
Yeah. Okay. Yeah. Then I don't.
Oh, I see it.
You do? Where?
Where's it?
Where?
Here. Is that it?
Yes. Yes.
Whoa. Nice job. I didn't see that. Yeah.
It's like all those years of I Spy are paying off.
There you go.
Okay. Stay. Well, you can.
Yeah, just right.
I think our crab may have abandoned ship, though.
Is that it right there?
That's it right there, yeah. I think we go back and we suction that after.
We'll see what we can do. Yeah, we can see.
Can you zoom in, please?
Zoom in.
That's nice.
Quiver 16.
was the name of that crab? I'm just gonna associate them together. Nice, like where did that?
Eurotychus. Or Carstyle. Either way. That's right, it's going in. That's good. So last I saw the crab was here. Not sure where it is now.
I think I still see him under that rock.
You do?
I think so. Right, so
Yeah, jar one.
You want jar one? Yeah.
since we have this opportunity, let's just go ahead and make it the next sample number if this does work out.
Yeah.
So that was, oh shit.
You still see it?
Yeah.
Oh yeah, there it is. Good job, Russ. Took off. It's right there. Unbelievable.
Focus.
I don't want to interrupt. Yeah, yeah.
Let me move into the left because
Alright, so try this way. I don't see him. Alright, well. Yeah. I'm gonna zoom in just. I think you got him.
I did too, to be honest. Unless it fell
Does it
rather
look-
quickly, but
There it is. Is that him? Oh, maybe not. Alright. we can have to wait and see.
Yeah.
I did
So we won't assign that any number.
no,
We'll just
but
write a note
we'll advance to the next jar though, I guess, right?
that possible in jar 1.
Okay, so but it might have gone into jar 1, correct? Okay.
Seamount is
Okay, so now we're indexed to 2. Ready. Just gonna make some notes here. Okay. All right. Here we go. Okay, let's keep going. It's tricky, those Chrysler gorges are really floaters,
yeah, that's
right? I don't think anything how those things go. Kind of weird for a moment just to see that crab on the seafloor. It was really strange to me. I don't
I know,
ever
it
see
was,
them
like,
there. I was
wrong.
like, it doesn't right. That's wrong.
He's like, where's he gonna go
No,
now? We
that
have to
morphology
get
does
him.
not belong on the seafloor. Oh, let's stop and take a look at this. Thank you. Looks like a primnoid with a sea star. Yeah, a fjord. Stop and take a look at this. We're at
There's
14.
also a metallic orchid there,
Where?
right above it. See?
Oh yeah. Oh yeah. Wait, it's got
Or-
a crab in it.
No, it's
It's a
a
chryso. No, it's a metallic orchid.
or
All right, we're looking at that one next.
yeah, I was like, or is it
I don't think it's
the oh god, why am I Iridogorgia? I can't tell. I think it's
No. All right, you good?
is that predation? Sorry.
Where?
Behind the oaf.
We'll look. I see some bear skeleton here.
Like that oaf looks way too bulky. Or I was just but no. I think it's just multiple oafs.
Looks like a primnoid octocoral.
Yeah, no. So there's crinoids
I
in.
just see a lot of ophiocanthids and astroschemas sharing this. But I will
Thanks.
zoom in. I will
Sorry.
zoom in on the. No,
No,
it's
I
good
don't
sight.
see any predation.
Well, you know, in this cruise in particular, I feel like I'm seeing more abrasion that's being done by these ophiuroids on these coral skeletons. You see how how he's swiping at the very top up there. I think he's taking some of that tissue off of these branches, which I really haven't seen quite this much before. So yeah, there's a clump of ophiacanthid-like brittle stars, an asteroschematid brittle star. I don't see any snails or anything else that would do this.
Nope. Right.
Yeah, we'll see. That's interesting. Primnoid. And then let's look at this.
Is that a Ruddigorgia or is that a Metallogorgia?
Oh, you see how the
Stem is like,
stem is a little wavy there.
yeah.
It doesn't have the branching of a Metallogorgia.
No.
Hold on, wait for it. Focus coming in soon, maybe. Gee whiz.
Well, we know it's a
Well, it's a
Chrysogorgia.
Chrysogorgia. I was going to back up too. It's a Chrysogorgia. Yeah, I don't think it's not Metallogorgia, but it's a weird thing. It's a bit of a poodle up here, but I'm going to hold this for a minute. I always want to sample these things. I don't know if you can see the.
Or is it immature and things are still migrating
Well,
out?
usually you see poodles down lower if it's immature,
Right.
quote, immature coral.
It's a nice shot.
It is a nice shot. It's rare we get that. I
Nice
just don't
hold for
normally-
10
just
seconds.
don't
I don't know if you're
normally
trying to
see
do that.
I am,
That's good.
but well, I don't know. I don't know whether to sample that or not.
No, I was looking to see if like we had anyone on Slack.
Saying
Steve, are
anything.
you there? Steve,
Yeah.
are you there?
No. How close are we right now to that? Sorry, folks. That was way too much. Just wanted a picture of the base here. There we go. I don't know. I should recognize this, and I don't. So. Yeah. Can we get a sample of that, please? It'll go in a quiver, I think. It's a little out of reach, I know.
For you too.
Yeah. Thank you. Can't help myself.
No, that's what we're here
Can't help
for. I
myself.
was
No,
like,
I
oh,
just-
if we see
well,
another
if
one,
we're
we'll
not
get
sure,
it. We're not
if
going to
we're-
see another one.
That,
so.
that happened the other day. I said, oh, we've been seeing that, right? Then we never saw it again.
So.
I can move the camera a little bit too if it helps. Touch it.
Got another one of those bottle brush Chrysogorgias up top. You can see it's shadow casting way up top.
Are you spotting shadows now? You're spotting Chryso shadows. All right.
Yeah. There's that. Keep it on a strip.
Yeah. It's like a heterocarpus type. But
Big one.
Okay. I think we're right here.
It's like, and continue. Otherwise, I'm gonna think you can't get past 8. Oh, jelly coming towards us.
It's a nice shot of the slope we're proceeding up.
Yeah, you can really get a sense of where we are.
Yeah.
Or just like, yeah, how we're going up. You never like really feel that otherwise.
There you go, Ben. It's not the screens.
I'm gonna take a shot quick.
You think that's gonna fit in a quiver?
I do.
Okay.
Do you?
I don't know, I just saw the lasers. It looks like a little bigger than 10 centimeters. So it'll be close.
Thank you.
That's a nice shot.
Okay.
That's great. See the claw and the teeth of the claw?
Mm-hmm. That's pretty neat. I've never noticed that before,
Yeah.
that they have those. You just hold here forever.
I know. You don't think so? That's fine. This is another one of those.
Hey.
Hi. Imagine a zoom on a crab.
Oh, you look like you want a hug or a high five. I'm not sure which.
I was just stretching.
Looking.
What do you think?
Can I say
I'm
something?
thinking you can give it a try.
On
Okay.
mine?
Oh, yeah, I was
Sorry.
to
No,
me.
no, stay right here. No, no, no.
Suctioning off the crab first.
When he said,
Could.
No, just-
Sometimes
I just
they're more
need one
likely to
second.
come along with you.
Yeah.
As you know, it's a, you never know, right? We could try it. It's better.
Give it a shot, like, sure. Let's do it.
Before you collect, can I announce something? Is it okay?
Sure. Before we okay.
Hi everyone. If you are just joining us from the Schmidt family or the Schmidt Family Foundation groups, I just want to warmly welcome you to this dive. We are about, where are we? We are 1,423 meters below the surface of the sea. I'm here with Tim Schenk, and you are looking at a chrysogorgid coral with some, I think, a chirostylid crab in the back. And the ROV Sebastian is about to collect something. I just walked in, so I'm not exactly sure what, but
Yeah.
a piece of it, I think.
Piece of it, and maybe the
and the crab.
Yeah.
Because Tim is really interested in studying the associates with corals, and this crab is one of those associates. We don't fully understand the interactions between the associates and the corals, which is part of what Tim is thinking about and researching, and we're so excited to be here. And we are on the southernmost seamount in the U.S. EEZ in the Howland and Baker unit of near the Pacific Remote Islands Marine National Monument, which is a mouthful to say. We're on an unnamed, previously unmapped seamount at the bottom of the U.S. EEZ and the Tokelau Ridge,
Right.
which is still a mouthful.
It is a mouthful.
So really welcome. It was such a pleasure to speak with you just a few minutes ago, and if this is your first dive with us and on Falkor, you're in for a treat. You get to see all the things: beautiful coral, beautiful crab, and a cool collection.
Yeah. Always. Do that. Take another shot here. Up close and personal. It's a great shot. You can see the coral polyps. It has 8 tentacles around the mouth. Those are feeding polyps, and then you see little pinnules coming off each one of those tentacles. It helps capture particles of the water column that the coral will then put into its mouth. So these corals love to be in areas of high current to bring those particles to them. And I am zooming in now on this crustacean. This crab lives only in corals, in deep water corals. Only certain corals will live in, like this chrysogorgid coral here. And when I see crabs that may be new, something unusual, we make a collection, and that's what I'm seeing in this case. I don't think I've seen this crab here on the southern Tokelau Seamount chain. Could be a new species, but what we want to do is see if we can't collect it and get it up into the lab, and when we do, we'll do all kinds of analysis on it, DNA, isotope, all kinds of things to understand the ecology and even the evolution. We look at the genes and how it's evolved compared to other crabs around the world. So, super important work. I love it, and we'll see how this goes. We may try to collect this now. Okay.
All right, are we
That's
gonna do this
fantastic.
thing?
Yeah, let's do
All
it.
right.
What's so interesting with these things is that the branches are not extensive, and so for some reason here on the southern Tokelau it doesn't seem as dense to me, and so we're able to actually image a lot of the associates really well.
Yeah, I
In-situ,
mean,
so
that
it's
was
not-
pretty damn
right?
good.
Yeah,
I mean, in
it's-
terms of imaging,
yeah.
that was a great
Yeah.
beauty shot.
So
You can learn a lot just from the images, right? So.
yeah. And so the coral will also undergo DNA analysis. We'll do DNA barcoding on everything that we collect. It becomes a really awesome database for comparisons to other places around the world. So
Yeah, this one's a mystery, man.
You happy to see him again?
that looks good.
Here we go, that's an angle.
That's the tip.
Hey, don't take copious notes on Slack or anything.
What?
It's just making a joke. No, it's just, I was like, oh good, there'll be all this chatter on Slack. There's nothing.
No.
It's because Steve's not up.
Well. See how that crab stays right with it? It's not startled by anything. It really, I think in some way it seeks protection inside the coral.
So collections like these are really important. I know sometimes there's just, this is a really, really common type of coral, but what's interesting about this is that this is an opportunity, you're putting this in an RNA later one?
No.
No. This is an interesting opportunity to get an associate that we probably haven't seen before, which can tell us a lot about the biology of the region. And so with one collection and sample, we'll be able to, have a whole lot of analyses in both the coral and the crab, which will tell us a lot about their, we don't know the relationship between the coral and the crab. Is it parasitic? Are they mutually beneficial? Are they eating each other in some weird kind of way? Are they trophically linked? And so each collection helps get us a little further towards understanding how not just what's in the deep sea, but how the deep sea works and the little relationships between all of these different animals and organisms.
470.
Mm-hmm. Yeah, I mean, we collected this one. It was a little bit of an enigma, which is why we collected it. It's not a metallogorgia. What kind of chrysogorgid it is, again, don't quite know. Do we call that a chirostylid?
Yes, absolutely.
Okay.
Yeah. What you take for granted too is the skill that the ROV pilots have. There's two here working in tandem, and they are in many ways doing better than a human hand, if you ask me, the way they've been able to take these samples and put them in the quivers. You just saw a great example there. we're working on 2-dimensional screens in a 3-dimensional world, and so we every day we're so grateful for these pilots and what they're able to do. It's just astounding.
And in between them, I don't want to count the years that Russ and Ben have doing this. It's
Oh yeah,
really
think about
impressive what
that.
they're able to do as a team together.
Yep, ready to go, Ben. These things are so amazing. These corals and whatnot are so amazing that it's so hard for us to make tracks. We want to get to a certain depth. We want to see how the composition of the species changes over depth, how diversity changes, and here we are
Get ready for the
looking
shadow.
at so many
There's
things.
that shadow I was telling you about up
We
in
want
the
to see.
top.
Oh, I do see a shadow. Well, let's just take a quick.
It's fine.
What do you think it is? You want to look at it?
I think it's another. I don't know if it'll have an associate. I don't see one.
No, no, we're okay. We're okay. We saw that one before, right? This
Yeah.
time. Right? It's the bottle brush.
Yeah, definitely.
It's a big Vercingetorix sea star. I'm going to iris up a little bit so we can get a little bit of depth of field. Ben, can you take a look at that white coral or whatever that is? It's just a quick.
I didn't see
Just
that
say
one.
as soon as I really want to take off, I can't help it.
Looks like it's got a couple associates potentially.
I can't tell. Can you tell what it is? Is it me?
I can't tell if it's a sponge or a black coral, or it might be a sponge.
Yeah, maybe you're right. I can't tell. Just wait and let Ben settles out before I take the camera. There we
It's
go.
definitely got astroschema or like
Yeah, I thought so, too.
associates. So
It has opioid associates on it, but oh, there we go.
Yeah, let's see if I can get this,
Let's go this way and see. Okay, it's a sponge and it's covered with oats.
does that have enough spicules for us to put in like the?
I was gonna ask you the same question.
It's like Brandy, get in here. You just laughed, but
This is exactly what you're looking for.
yeah.
It is. Do you want to slack Randy quickly or not?
Yeah.
Just to make sure. I don't know. Let's see. Get the lasers on. So we'll see how big this is. We're doing respirometry experiments on board. So awesome. You got some lasers?
These are on. Just zoom in quick fine. Back here a little bit.
There we go. Okay. Yeah. So it's you know.
It's a decent size. I'm honestly counting the spicules. I'm like, do we have
All right. Let me
18
zoom in. Let me zoom in again.
spicules?
Sorry. Excuse me. Let me zoom in again here. Oh, I got lost. Okay. It's a blurry image, but let me just see if I can. There we go. Thank you, everybody, for your patience.
I
Loaded
don't know.
ophiuroids. I don't know if you want to hold off because we're already in the dive. So
yeah, let's-
I would.
Let's hold off.
Yeah. But remember, we've seen
Oh,
that.
she said yes.
She did.
Yeah. Okay.
I was gonna hold on. Is that okay? All right. Collection.
Yep. Let's do it.
We'll
You're
do
putting
s-
these guys to work today.
We'll do some more. We're gonna. Yeah, I don't know. We're gonna want to collect this. I
I mean, you'll be able to use it. I'll just
I
snip the
think you'll
top,
hold it together.
right?
Do you want to slurp it or cut it? based on what you want to do with it. We can suction sample this, or we can snip it.
What do you think is going to keep the spicules most intact? So the things on the outside.
A cut.
I think a snip, to be honest.
Okay.
Would work better.
Yeah.
I think we need to cut a section.
Otherwise it was
put in
good.
a jar, put it in one of the
Okay.
Okay.
buckets. I think I would say this, to be honest.
Okay.
Yeah. Okay.
Let's do that.
Let me back out first, 'cause I'm on a very thin ledge.
Okay.
And if I push out, I'm gonna drop.
Well, you know what you could do?
Well, I can push out.
If you rack back in and just look down, I can take the lid off flow to you, because it's quite close.
Right back in.
Oh yeah.
Oh, hold on.
Hello.
I'm finished.
Yeah, I'm finished. Yep.
I'm good with that. I just need to see, you just rack in the camera.
I'm right in.
Alright, sit down
Let
then.
me sit down.
You should be able to take the lid off.
Let me try pushing out slow.
Yeah.
I'm just very. Yeah. There you go. Might be alright.
So how do the sponges look when you bring them up? When you see the spicules of the surface, are they in good shape when they come up
Yeah.
these depths? And you're able to put them in your little respirometry chambers and measure the oxygen levels, right? For how they're respiring.
Yeah, exactly. So yeah, no, they stay relatively intact. I mean, this is silica, so as long as we're relatively, I think, gentle with the sample upon collection,
Yeah, they're
we
like
can
glass. You know?
literally just like snip them off like hairs
Uh-huh.
when we get them in the water
Right.
and transfer them to these
Give me
little
one image
2 mil
before I
vials.
collect
No, we're ready to go.
And they breathe
Thank you.
just like corals breathe, which we had no idea what to expect, to be honest, with a
Right.
we know they're filter feeders. We know they pass a lot of water, but yeah, they respire like within the same time frame that a coral does, in terms of getting to zero in this
Yeah.
closed container.
So how long will you watch them? How many hours do you?
Experiment takes anywhere from 4 to 6 hours, really, to get down to zero. Sometimes we'll let it run overnight for convenience, but it's usually
Okay.
bottomed out at that point.
Right.
Which is, I mean, in comparison to shallow water corals, like sarcophyton at home, I mean, we let them run just as long.
Yeah.
It's not like there's a huge difference. We brought out other assays with us because we were worried that because everything moves so slowly in the deep sea that we might not actually see respiration. We didn't know if things were alive or not. But
Right.
that
Yeah,
hasn't
they
been
are.
a problem.
No, that's really good.
Okay, you happy with that?
He's not gonna cut
Yes.
it.
Have you?
He's just gonna grab it.
Okay, it's probably gonna come. Yeah, that sounds great.
Right, and
Looks
all
good.
of those are off. Those have to both be off.
You want me to cut it?
As much as possible, Russ.
Right.
Just
Yeah.
because we're
Keep
going
coming
for
down.
spicule volume.
Right. All my question marks is because what it does is it will go to whatever decomposes in stream. It'll stream.
Perfect.
There you go.
I'm just worried about overwriting then if you, then it would.
Yeah, those bottom spicules will be great that you got to pull off. Nice and long. Of course, I'm like,
We're going into. Sorry.
no, you're good.
That's what I was trying to understand earlier when I jumped in, because I was. I was trying to manage to tell us about
This is going into. This is 14, yeah.
It's freaking perfect.
That's fantastic.
Awesome. That was perfect. Couldn't have been better.
Yeah.
It was worried we weren't going to get the bottom of it, and that's where a majority of the spicules were. So that's why I was like questioning it, but that was great.
Yeah.
I've only done one experiment with a sponge so far, so compared to our, I don't know what we're on with corals, but it's quite a few. In the 10 to 20 range. Alright, now we swear we're gonna make tracks after this, right?
Yeah.
I'm kidding.
No, we always say that. Until the next thing.
It's almost a joke.
Until the next thing.
It's a joke.
Great.
Is there anything else you want
Nice
to
job.
add?
Nope, that's perfect. We can head back on our path. Really a lot of draped sediment. We're at 1,420 meters deep. Give us a little more. Iris.
There's some pictures on my right. You want to take a look at that?
Sure. Over here, you mean? Yeah. Again, we're watching the sonar and seeing where hard bottom substrate is.
Oh, there's another
Front of us.
metallic gorgia for you too.
I'm almost exhausted of them. No, that's got an astroschema in it. It's,
Yeah, we can see it from here,
I
right?
don't see anything else. Nope. Yeah, we're good.
You gotta ask
No
for
eggs.
something else now.
Uh,
I think it's deep sea grass.
just earlier we saw two groups of red eggs on one of the Talagorgia, and also
RV, that's waypoint shifted.
another golf ball sized egg case, it looked like, that we sampled. I really look forward to seeing that when it gets back up to the surface.
Looks like we got a Plexaurid possibly on the left
Yeah.
of that rocky outcropping.
Look how
Yeah.
scalloped all this margin is here. I'm imagining brachiopods with some sort of bivalve, but an ancient reef here. Just a snap zoom on this for a second here, Ben, right there. Just
It don't land, just
I'm just
fly
gonna go
by.
in here.
East plus Plexaurid with an astroschema.
I think so.
I can't.
Another
Yeah.
way to. One more quick here. Trying to see if this might be Acanthogorgia.
Oh. Could be. It's definitely bushy
Yeah,
enough.
I don't know. You're good, Ben. Yeah, I think it is actually. I think it's Acanthogorgia, but we'll see.
I
We'll
put
see
that
some
down
more
in the
of these.
notes. I put
You did?
one or the other. Yep.
Okay, great. Thank you. Let's keep. I can't remember what depth we started at on this map. I'm not sure how far we've gotten really. We've got 40 more meters or so to what next waypoint.
So Jingle Meow just asked if these would be sent to the Smithsonian when done, and yes, they will. So our scientist Steve will do a lot of the characterization, and when he's done with all of the dried voucher samples, they will go to the Smithsonian directly. Exactly.
Another Metallogorgia there.
Yep. This is Metallogorgia land.
Well, they tend to
Little
really
bit.
come into zones. They really are sort of abundant or prolific.
More
So-
routes to my right.
Roger that. Full out.
Yeah, just take a swim.
First thing I look for when we see a slope like this, a sediment that runs into some blocks like this, a rock, this is look for rubble, coral rubble, to see if there's something above that's been dropping off over time.
Is that a hard coral?
We're going to want to look at that white coral, Ben. Thank you.
Looks like
Yes, we're stopping in every coral.
a sponge
But it-
and a
But I think
paragorgia.
that's an alpsamia, which is pretty deep
Yeah.
for that. We're at 1408 meters. I would expect it more around 1,000, but I'm gonna take a look and see what it is. Looks like all the polyps are on one side.
What is on the underside too? Is that a sponge? I mean, I know it's a plexaurid, but or is that a
you mean
coral?
that?
Yeah,
Here?
to the left.
Yeah, I was guessing chryso. You're good? Let's come in here.
Oh yeah. Got a sclerotinian.
Yeah, so that's an Alexandria. And that's that planar
It is a
chrysogorgia, so we're
chryso,
starting
oh.
to see the,
That looks so weird to me from that angle.
alright. We're gonna see these again, I know, and we're gonna set up on them and we'll image them later, but that's it, an Alexandria and a planar chrysogorgia. No associates on that. Now Samia. Okay, Ben, thank you. Keep rolling. Oh, come on. Just checking to make sure.
Another chryso, probably.
No one's.
From this view, I can't really tell, but I don't know.
It's a really neat rock formation. See the layers there.
Yeah, it is.
It's
interesting.
almost like I'm gonna quick zoom. Just to there. Come on. I am not getting good focus on these things.
Perfect. One of those
Well,
planar
another
guys.
chryso. That's a planar chryso again. Same. Okay. That's right. We know what that is, so good.
Is that a Victor Gorgia down there? I mean, I know it's not exciting, but it could also
There's
just
one,
be a
yeah,
rock.
that's the chrysogorgia bottle brush up top. I think you think it is? Sorry.
It could be. It's not a big deal though.
Yeah.
Not worth it. Yep, there's
All
another
right.
bottle brush, like you said. Sorry, I
Yep.
just said that. You're in the land of chrysogorgids.
Yeah, we are. 2034.
And just as I say that, a primnoid pops out.
Okay. There's a big sponge coming up. Nice sponge. I'm not sure what that is. No, we're okay. So you see a little bit of rubble here at the bottom of the.
Is that a bamboo worm or a stickopathes? I think it's a bamboo. It looks too bulky to be a stickopathes.
Just a close drive-by of that would be great, Ben.
Yeah, bamboo. See the nose.
Oh, no, you may. Please, I like you in shots.
Who was it?
No,
You want to talk?
no, I want to play. I haven't been in the control room in a while.
Yeah.
You sure you don't want to sit?
I want to play.
Thank you, Ben.
You know, Tim,
Yes, ma'am.
out of all the clips from this entire expedition that could have been picked to show this morning at our outreach
Yeah.
event, you want to guess which one they picked?
We were on it. Well, no, I don't know.
It was you and me. You want to pick? If you were to pick our funniest moment so far.
Does that have a
Dan, can you
lobster?
sit down? Sit down and look at that. Thank you. Sorry. Yeah, I'm all the way out.
Which one?
Deep sea
Sorry.
blueberry collections. Remember that teeny
Yeah,
tiny
oh,
little?
do I remember? God, it's burned into my brain.
Oh, stop, Tim Shank.
Always look like they're scalloped out, like they're shallow water reef. I'm thinking like
Yeah.
brachiopods
They
everywhere.
absolutely look.
but I don't-
Steve and I were just talking about that in the
Sure.
mess.
Where you?
That they look, that looks, I mean, it's carbonate. Yeah, I think
Yeah.
brachiopod reef or
I know it's
something like that
I
for
know
sure.
it's well weathered and whatnot, but it just seems like it's so scalloped.
There's the mucus nets.
The what?
Mucus nets.
Yeah.
They sold the
Okay.
I swear I saw a pair of eyes on this, by the way.
You think you do?
Did you,
That
have you
upper
seen any
right corner,
brachiopod
you know,
shells?
Middle.
No.
It's probably not brachiopods then, because they often
know.
they leave a trace.
I'm,
I know you're trying.
no.
You're doing a great job. I like fear having to do
It's
anything.
such a shaggy coral.
Polyps
And-
facing down.
I know.
Is it mice and shrimp? Or crab?
Nice
There you
job,
go.
Ben. I'm just gonna hold this. Look how long those polyps are. All pointed shaggy, pointed down, bamboo, mice and shrimp flying around. What may be a Heteroptychus, new genus of crab that seems to live exclusively on bamboo whips in this area.
I'm gonna slack Steve and see how he feels about.
Yeah, if he's, I didn't think he was up and around. I was,
Oh, he is. I just saw him.
Almost looks like a polyp. Almost.
see the polyps in the foreground.
Yep. So let's find out. But
What's he?
I'm waiting.
Oh.
I slacked him.
Right. Yeah.
Can you sit for a second while I go do that?
What?
Sorry,
Yeah, of
folks.
course.
We're just waiting to see if a collection
Sorry.
may be in order here.
Of course.
He may already have this one, which is fine.
All
Come
right.
back out.
You know, yeah, I think we're good. So it's up
We're
to
good.
you.
Okay.
It's up to you.
All right, let's keep going. No, no,
Okay.
we're good. All right.
God,
Thank
I
you
wish
very
we
much,
could-
Ben. Nice little bamboo there. It's the first one we've seen of those, I think, so it was good to document that, where it is at this depth.
So this pretty craggy, reefy stuff is definitely indicative of something that used to be in shallower waters. But the fact that there's no brachiopod shells, right, it might suggest that it could be other things like urchins or other boring organisms that, you know, kind of bored holes into the calcium carbonate limestone pavement. But I don't know. We'll just have to watch.
It's not much, right?
Pardon me?
It's not much to check out.
Xenophyophores, submit. See all that pavement?
Yep.
I bet it's the same kind of thing we'll see near the top, but it's, you know, here it is much deeper, sloughed down over the years. I mean, these are these seamounts in this chain are roughly 80 to 90 million years old, and so we're talking about a long. It's really hard to guess when this was last shallow, and because you have two forces here, we have subsidence, and of course
Yeah.
the changes in sea level.
Yeah. I was just reading that when Captain Winslow came here,
Uh-huh.
down here in 1851, more to Winslow Reef, that they said they saw two big rock formations awash above the surf, if you will, and they came back in I think it was 1941. There were no rocks. It was 11 meters deep. I don't know. I mean, that's just
yeah.
people have subsidence of 10 meters, or could have been not in the
Well,
exact
could've
same
just-
place either, but just it makes you think about subsidence and the processes which form
Well,
the end of what we see in here. What's that?
what is
black
that? I
thing?
have no idea. Maybe just a rock?
Need some
I
light
don't know.
on
I'll have to look. One of the things, this is the same story for Corallivory, which is further south along the Tokelau
Quicksand?
chain, where there were, it looks like basalt amidst the carbonate. We've seen a couple of it.
What
There
in
was
the
just
world?
another one. No, there was just another one, a big one, just a little further
Alright.
back. So I think it's kind of mixed in, but I don't know how it got there.
I don't either. It's like a pair of brown shoes at a formal wedding. And a black tie. Right? Oops, you stick out a little bit. I don't
Tim.
know what that is. Yeah.
Fit
Keep,
the basket.
No. No.
Looks like a
Well, that-
How many
when
rocks
did
have we
that-
taken already?
One, and it's questionable. It's kind of a.
All right,
mushy-
all right. Can you think you can grab the smaller of the two?
Yeah.
Is it too big?
I mean, you could even maybe break it off.
Yeah, why don't we see that?
Try to
Brown shoes at a formal wedding.
I don't
What?
know. I just make analogies that help me remember. What's that?
What do you want me to do?
Yeah.
All right. How about their?
Well,
Yeah.
I don't need it right now. I'll need it for taking it. See, I'll be back now. You hold it for a while.
Have you guys tried the coral pop
No.
push popinator
Wouldn't do that
3000?
without you here,
Oh,
no,
good. I'm
so
so
we-
glad.
We introduced the concept to that.
Okay, good. I can't wait.
Test the ice soon, actually.
That's good.
The minute we are done collecting this rock, that's my next. I'm gonna look for a
That
place
sounds
to
good.
do that. Is that
Yeah,
all right?
I'm ready.
Can we
Let's
do
do
that
it.
together?
What do you think? What kind of coral would you like to do that
I
with?
don't know, but I'm gonna call
Big
the chefs
polyps,
in
little,
too.
little polyps.
It's not coming up.
Chrysogorgids? No. Some
No,
you
I
can
think I
actually
want
image.
some big meaty polyps. Maybe a bamboo,
Yeah.
but
It's
not
gotta
ones
be-
with
it's
not ones
gotta
with
be-
polyps facing down.
You
though.
need a good setup where you're not with the ROV prop wash is not
Exactly.
impacting.
And you want it to
right?
be, you want them to go, you don't want the pressure of the syringe to close the polyps. I want them to do it above the coral
Well,
and let
that's
it
gonna
fall
be
down.
tricky. That's gonna be.
And I want you to do it above the coral and let it fall down in a place with very little current. Or you have to anticipate
Well,
the
you,
current.
you got the current right. There's not much here today.
Right.
At all. So.
So if you're listening,
little
Tim and I are
stuff.
strategizing on how to use a new tool created by our amazing ROV team called the Coral Push Popinator 3000 or something like that. It's an incredible collaboration between Science, ROV, and Galley on this ship, where we are going to attempt to try to
There's Anthomastus.
look at some coral behavior. Yeah, I see it. Hang on.
Yeah,
And a
sure.
crab. It's a homalid on the left.
The little anemone. No? Yes. I don't know. I don't know. The crab is moving, the rock's not. Can I get the crab for a second? We can always get the rock. And I'm going to. I think I'm in manual. Is this? There we go. This is a homalid crab. Yes, family Homalidae.
Mm-hmm.
This is going to be a great shot if it doesn't move. And it has modified legs in the back that actually pick off an anemone or a piece of coral or a sponge and put it over its head like it's doing, or its body like it's doing now.
It looks like a cheerleader, but it's actually, because looks like it's got a big pom-pom on its back.
Pom-pom.
But what it's really doing, right, is protecting itself with the anemone. Either physically, because the anemone has nematocysts that would sting other things that might try to go for the
Yeah.
crab, or chemically by shielding the crab from being scent detected.
Yeah. So it thinks it's hiding right now, doesn't it? That's fabulous. But we'll often see them holding one type of coral being on another kind of coral, so it's not quite camouflage, if you will, like we think about it. That's fabulous. All right. Much more interesting than a rock. All right.
Well,
I'll-
hey, much more interesting than a rock. You know,
Well,
I
isn't
agree
it?
with you, but if a geologist was listening, you would have just, caused a war, Tim Shink.
No.
But I agree.
They must admit, though, the crab moves and the rock doesn't.
It's true. Well, maybe the rock does. Look, how did this rock
Yeah,
get here?
that's true. That's very true.
I
That's-
don't know what you're saying.
I mean, I don't know if how does that rock get here like that and not be encrusted? I mean, glaciers would drop rocks, drop stones.
Right.
That's how you get a weird rock in a weird place.
It rolls downhill, except that the only thing uphill from us is carbonate.
Ooh,
Yeah.
that's pretty light. Sort of. What do you think?
Or maybe it's like a mix of.
It is.
Maybe it's like mostly pumice and it's volcanic foam and it kind of floated up here when the volcano was.
That's
I
prob-
don't know. It's not possible. I'm just kidding.
No.
It's not possible.
My mind's wide open, Randy.
It's not possible.
I don't.
Is it? I don't
I
think
don't
so.
think it's
I don't think
pumice,
it's
though.
possible. But look at it. I mean, it's not. It can't be fully pumice, or else it would float.
No.
But maybe it's
No.
very. Maybe there's lots of pockets in it.
Sometimes they can break a piece off.
It's not gonna break off very easily.
No.
That one.
Fit in the basket.
Oh no.
Good fit basket, but maybe a little low under that.
Yeah, maybe the little
I
one.
think so. Yeah. That's plenty big too.
I bet you they came off of this. They came from the same source. We're just gonna assume that.
I think so.
I think so. I mean, like you said, you
Wherever
got two
that
brown
is,
shoes.
Wherever that is. Two brown shoes. Yeah, right.
Tim. Now I have to know: are you the kind of person to wear brown shoes at a formal wedding?
No.
Mm,
No.
I mean, neither's my husband. Although I think I would be.
Yeah. Might be that person.
Sorry.
Red Sox. Oh.
Oh, perfect. A little nugget.
All right. Let's.
More manageable piece.
Yeah.
He's gonna image
We're gonna want to image this for you.
Yeah.
Put it in, Russ, as you know. Line up here. We'll just.
the rock itself in situ too, not just the rock sample.
Ah.
Beautiful.
Steve in the room.
Anna's got
Steve's
it.
in the room.
Who else is Steve out?
I didn't know Steve was here.
Steve and Anna.
Okay.
So if you're just joining in, we have 4 of the science
So,
team with
little
us. So I'm
rotation.
Randy Rogen. I'm a faculty member at Boston University. We have
Can you
Tim
rotate
Shank
us
from
a
the
little
Woods
bit?
Hole Oceanographic Institution. We have Steve Askovich, who is also at Boston University. And we have Anna Gauthier, who is joint between Boston University and Harvard University. And we are half of our science team. My goodness.
So this was the side of the rock that was facing up off the seafloor.
Yes.
Steve? Yeah.
Yeah.
Mm-hmm. Yep. Also in the room are Ben and Russ
Okay,
flying,
great, Russ. Thank you.
flying us the flying Sebastian today, and we've got Debs, our fabulous marine tech, who's running our live stream and taking pictures and helping to manage our data and doing everything we can do.
Should ask Steve where that black rock came from. See what he says.
Steve, what do you think?
What about it?
Where did that black rock come from?
I mean, it's possible. So it's crusty. There's a lot of alteration inside the rock. It could be some sort of dark manganese coated source around here. Not all of these carbonate blocks around us are in place. They could have fallen from above. So we're kind of could be volcanic, surrounded by carbonate blocks that have just
That transition?
fallen
What's our
off over
sample
the years.
number?
That was 472.
Okay, thank you.
Above us, I noticed the dive track cuts through a little plucking
Yeah.
in the side of the like a V-shaped notch.
Yeah, exactly.
So this, could all just be like
Yeah. That's
Yeah,
it.
maybe.
talus, blocky.
So
talus.
Steve, ever the closet geologist, has pointed out that a very reasonable observation that where we are is kind of transitional between where the volcanic material is and where the crust might have sort of been starting, and actually our dive track on purpose kind of takes us through some crigs and crags in the seamount to sort of give us some more interesting substrate, but that actually could have implications for what we're seeing geologically. What is that? Shiny? Oh no,
I don't
it's
know.
just another piece of rock. Could have implications for seeing this mixed rock structure, because you could have all of these different layers combining with some talus, which is the word for rocks sort of falling down the side of a mountain and landing in a plateaued spot. So you can see lots of them now, kind of coming together. So I'm sure that's what we're seeing. Thanks, Steve.
Yeah, that may
I'm
be
gonna start calling you Geology, Steve.
No.
Please. Gonna get a bad rap on the internet.
Once, if you're a geologist and you're listening to this, all we can say is we're sorry.
So
We're learning. We're trying. We're biologists in a geology world.
So we were at 1,386 meters, and we're just coming right up to waypoint number 2 on this dive. See a lot of those black rocks. There must be material up top too that's just falling off the top. I think we're seeing much of a current today. As I look at all these hard surfaces that could be colonized by. Look up.
There's a sponge. You
Yeah.
know, but the lack of current is going to be perfect for our experiment. I'm so excited.
Well, stand by because it's probably going to happen pretty soon.
Well, we got to find a coral first. But I like these sponges. You know, these ledges are just so craggy. There's so many spaces for attachment. I would have thought this would have been full.
Just keep rolling. Yeah.
There's a plexaurid. Have you looked at those yet, here?
Yeah, can we take a look at that one, please? Yeah. Yeah.
I don't think I want to use this one. Their polyps are too small.
I don't think so. Well, there's the anemones on this one too.
Can you take a close zoom on the rocks too, and just get
I
a
am.
sense of the
We'll get-
fossils? Oh, look, solitary hydroid. Small one.
Oh yeah.
Look at all those bases. This
Right.
looks like this used to be a hot spot.
It's a great shot in the pilot cam looking down
Oh yeah.
the boulder system. Yeah, more from the side is better. Whatever you can do, Ben. I know it's close. It's like the stratified rock, the sedimented rock, sedimentary rock.
Mhm.
Looks like these bands of erosion.
And of growth, right?
Yeah, no, right.
Can I borrow the stick, please?
Absolutely.
Thank you. I am interested in this.
Don't know yet. Steve, what do you think? I'm fine with the snap zoom.
It's a flexor and
Yeah.
most. I mean, it looks something like what we collected, but I can't put it as well more species from here.
Mm.
Get a little closer.
A little sponge back here too.
Hang on. Just missing focus.
It looks very much like something we collected on our further north sites, either ABNJ or the first Seamount within
Yeah.
EEZ.
Nice, Ben.
I mean, it looks almost like what I've been calling Plexauridae species 2, which was sampled on both EX and during FK17.
Yeah.
Okay.
So
Did-
I'm okay with this.
Do you see the damage on the bottom? After you're done looking at your
I see that. No,
overlaid, can we
I'm
take a look?
getting focus where I can. Nice, Ben. I don't know. I don't see any predators or that kind of thing
No,
on there.
the list looks like it might be a regrowing polyp, and it looks like there's might be some damage in the bottom. So can we look to see if that's really regrowth?
Yeah, I'm all the way in.
Mm-hmm. It's maybe. It looks like a little nub, like a little bud that's starting to get ready to regrow.
There you go.
Mm-hmm. Yeah, I think it is. See that little one on the side?
Yeah, I see it. Yeah.
Nice example of regrowth. So it looks like something snipped that branch, right there, that big, thick branch, and it's just starting to regrow over it. So for those of you who are watching us collect snips of corals, you can. This always gives me. It always makes me happy to see regrowth over areas that have had damage before. You know, obviously nobody's ever been on the seamount before. We're the first people to see it, but so this got damaged organically, and this coral is able to regrow, which is really exciting. And if you scroll down on the colony, it looks like there were some sparse areas where we see damage. I don't know what caused that. Do you see the culprit anywhere?
No.
Me either.
No.
So this may have been, my best guess is some kind of corallivore, especially in the middle here and to the right, that has eaten and since moved on.
Mm-hmm.
But I don't know. Could you hit Ivory? Right, no corallivore. Thank you.
Okay.
No
All right.
corallivore. Nope, none present. Just so that I go back and look at it later.
That's great. Nice job, Ben. Thank you so much. That's great.
That was great.
So, Pixar number 2.
Just looks like a dead
Yeah, it is.
face.
We did see an Samia.
There you go. There's a Narella.
Here we go. Was that a sea star on there? No, it's an oaf.
Some kind of primnoid, but it might be a good candidate for feeding.
Do you want to look at it? probably
Yeah.
sit
I mean, they
there.
can sit down there,
All right,
right?
now let's see if we can look at that one. The
Hey.
primnoid coral right there.
Hey, Anna, can I ask you a big favor? I'll walk you, but can you run down and get the chefs and tell them we're about to try their feeding device? Here, give me the,
Oh, I'm sorry. I had the pointer. This one here.
invite them up to the control room when we have time.
We're gonna want to sit down. Thank you.
It's not something like the boxes in the back heat now.
No, so check.
Is it okay with you? Is now a good time to try it?
Oh yeah. Why not? If we can get set up on it right, there's not much in the way of currents
We
here.
should probably prep the pilots for what we're about to do.
Ben's gonna sit down, then we can.
Yeah.
We just switched out to Chris.
Ah. Right. I didn't notice the
So
switch.
you got, well, I think Adam. He'll sit down. Maybe Adam will come in here.
Right.
So
We'll let them go through their watch change.
Yep.
Or their shift change.
yeah. It's 10 o'clock in the morning here. Local Tokelau time.
Tokelau time?
Tokelau time.
Tokelau Ridge time. I dropped the stick.
I thought I was sure.
You can't take me anywhere, Tim.
I thought I was a coffee.
I wear brown shoes to a wedding. I drop the sticks. Just
All right,
kidding.
I think we're good here. Let's see if this is the angle we want. Polyps outstretched.
They're down though.
Is this a one and done? No, you can shoot a little bit and hold on.
Yeah, exactly.
Go ahead. Yeah, it's a little bit of.
They're facing down,
Yeah, but they're open.
but they're open. Did you?
What?
Oh, good.
What?
Okay,
Okay. So,
we're gonna try it. They're small polyps, so this might not work, but we'll give it a shot.
well, it's also the ones we can focus into really well. So don't know if you want to
Okay.
do.
Hang on, let me tell them what's going on.
is we got to pick the part of the coral that
Yeah.
will show it the best, right?
So what we're going to do is we're going to try something brand new in the deep sea. We have the chefs on board with us in the control room right now because this lucky coral is going to be the first patron at the Deep Sea Falkor restaurant. I don't even know what to say. How do you want to
Well,
do this?
first, I guess you should back up and show the menu.
We should show the menu. Actually,
We're gonna do
you
this.
gotta tell the pilots then.
Okay. So,
Yeah, before they move too
yeah,
far.
so, Adam,
Yes.
before we start, I guess we have designed the menu for the corals for today.
Right, yes, okay.
So if you can, I don't know if you can.
See what's on the menu.
We got to see what's on the menu. We're
let's see
going to
what's
try
on
to
the menu.
feed corals because we're really interested to see what their behavior looks like. I've been watching marine snow fall on corals this whole trip, and I don't really see their polyps reaching out and grabbing it. So we're going to try feeding them. And so we have this great collaboration between the science team, the ROV pilots, and the chefs who have made a menu. So we have Bert and Pete in the control room right now. Today's menu, seafood cocktail a la Falkor, bon appetit from Bert and Pete. I love it. So they are.
So we should probably get.
Yeah.
Is Bert in-
Bert and Pete are here. You guys should explain the
They
menu.
should explain the menu.
You should explain the menu.
Somebody. come
Please,
on.
both of you. You guys, this is your moment. Come on.
So what did you put in the syringe? Just what's the list of things in there?
Oh no, this is like a little cocktail made out of actually only 2 ingredients. There's water and fish in it. So it's we do not really want to overfeed them and to
Right.
give them too much of things they would
Complicated.
be too strange to them. So we kept it simple. Sweet water, I must add,
Yeah.
not salt water, and fish. And
Okay.
it's
What kind of
blended to
fish?
a very fine.
Bert, what fish is it? What kind of fish is it, Bert?
The barramundi? Barramundi.
Okay, so we're going to
Wow.
try just a tiny little bit of fish here. We have the RV pilots have designed the Coral Push Poppinator 3000, which is this new tool that you're looking at right here. And we are going to try to feed, just put a little bit. And no, you cannot eat soft corals for many reasons. They are full of all kinds of defensive chemicals and defensive chemistry, some of which, there are some cnidarians that have really potent neurotoxins that could kill you. So I really recommend never, ever, ever eating a coral. But we are going to try feeding one, just a little bit.
They do not smell appetizing either
No,
when they're in the lab.
not
right?
even
There's
a little
one
bit.
I think smells like mint.
So corallivores can eat them, but they've evolved to be able to deal with the defensive chemistry that these cnidarians have put out.
So we have pilots Chris and Adam who are going to execute this never before executed thing.
Rubber bands are
So
so great.
and so that slurry that you made was frozen, right? You put it in the freezer.
Yes, so when we blended it, obviously it was not frozen
Yeah.
with the water, but then afterwards, once we filled the syringes, then we put it in the freezer in order to make sure that it stays fresh and also stays within the syringes on the way down.
Yeah, I think I need
Right.
a
So
flash
it's been
here.
thawing, I guess, during the course of
Yes, on the
since
way
we've been
down
diving,
it has
yeah.
been thawing.
Yeah.
Hopefully,
So
let's
yeah.
see what happens.
Yeah.
I think if you get on top of it, just you gotta lock it.
Yeah, it's locked.
So now here,
Good, I got it.
I'd like to
We're good, we're good.
I'd like to give
What are you
this
doing?
chair to
Oh, there we go. Come.
Oh,
have a seat.
thank you so much.
So, why don't
tourists
you
here.
introduce yourself a little?
Hi guys, I'm Pete. I'm the chef here on one of the 2 chefs here on Falkor, and that was Bert, the head chef here of Falkor.
All right. So
So
wanna
this
get
is
in-
now the moment where the waitress actually brings the food to the table.
So we'll have to get a close up on the polyps to
Yeah,
see if they have any
that's what I'm gonna
reaction
do.
whatsoever. So why don't you take this stick over there? Yeah.
Okay. I'm gonna bring this a little closer.
Yeah.
No, we're good. No.
Nope.
Enjoy your moment. This is good.
So just to look, see if, want to see if we can. Oh, I'm just.
Oh, sorry. We're fighting. No, oh, you.
Yeah, we're just gonna get the arm in position
Yep.
and then we'll.
And then I'm gonna full zoom on the polyps so I can see if they actually eat anything.
Right.
I apologize. We're like fighting for zoom control here.
I hope it's thawed because it was frozen when it went in, and it's only 3 degrees down there.
I know, but it went through the
Should be alright.
went through the tropic. If it's not thawed, we'll just wait a little while.
Yeah.
But I sort of thought sitting on the deck for a bit would have thawed it pretty quick.
Yes, fine.
It's pretty tight. Just so you know, it's gonna take a serious push.
Okay, so if you hold there, Chris, I'm just gonna zoom. You want to verify that the polyps are open, right?
I do.
Okay.
I'm gonna oops.
I'm gonna actually, what I'm gonna do before we do this, I'm gonna extend the starboard arms to touch.
Yeah, it looks like
more,
it's not quite over yet.
more over. So,
Okay, perfect.
You got that, huh? The two holes in the diesel or something.
Basically right on top of it there.
yeah.
Come down and touch.
I think you can do it without touching the coral. It's even better because it'll otherwise the pressure will jostle the coral.
Just right above it, but
Yeah, perfect.
let's keep an eye on it, make sure they want to push down with the fork. It doesn't push down the entire arm. You ready?
Ready.
Yep. Okay.
Ooh, perfect. I think that's great. Ooh. Okay, I think we're gonna save the rest for another day, and I want to sort of see if any of it made it onto the polyps.
Suggestion is to go maybe behind the coral more, and so
Yeah.
it flows
So
into
we
the
want
coral.
to extend a little bit more, try it in.
Yeah, I'm going the other side with that, bro.
Um,
You can see it.
but we do have confirmation that the Coral Push Popinator 3000 works. This is a very good mechanical test.
And the thawing worked on the
And
way
the
down.
thawing worked. Yes, it was perfect. It didn't explode.
This is like a happy coral.
Well, we'll see.
You want for me?
At least it's a lucky one.
Still gonna have to sit. That should be.
Come back in a little bit more because I think that's the current just flowing through it.
Yeah.
Yeah,
If you come,
They are. Not what we did.
I think that's okay. I think they have to catch food somehow.
I think they're.
I don't want to touch the coral.
We'll go the other side, right?
Yeah, I think you. I think so. Hang on. Yeah, Adam, Chris, can you go like behind the coral, like all the way behind it, not on top of it, like, and then let the current drift it in
Good to sit. We are behind it, but
Yeah,
how far
yeah,
back
you
do
can
you
see
want it?
here.
a little further.
A little more? Okay.
Oh, that looks good.
Alright. That's our actual. See
spot.
Wanna make sure I don't hit the wrong one.
Yeah.
Okay, perfect. Okay, that's more than enough.
Yep, we're off.
There's lots of stuff caught on there.
Okay, camera's yours.
Thank you. So what I'm looking for right now is just to sort of see what happens. I'm looking to see if the polyps grab, if they start seeing active movement, as they start trying to grab things and particles. I mean, these are polyp down corals, so I think it'll look really different on corals with polyps facing up, but they still are obviously capturing food particles, and so they've certainly caught on to some here. And my question has always been, can we actually observe the behavior of feeding? I would think that in the deep sea where food is really scarce, that if and maybe they just, I mean, I don't know how anybody could not love your cooking. You guys are wonderful chefs, but maybe they maybe they like this, maybe they don't, I don't know. But I would think that the minute a highly nutritious particle or morsel kind of passes through them, they would try to grab it and eat very quickly before it goes away.
Maybe you just found the one picky coral.
No, I'm just trying to see what they do. They're certainly hanging on to it. And now that they've caught it, right, now that it's stuck, they can take their time. So we might not wait too long, but maybe we can zoom out and see what's happening on the seafloor. See if your cocktail has brought other diners to the restaurant. And then come back and take a look in a second to see what the coral's doing. I see no action. I can see little bits, but no action. Okay, corals, you've had your moment of privacy. Let's watch. Come on. I'm going the wrong way at the focus. Hold on.
It's tough.
Do I have control? Manual control?
Try now.
Ah, much better. Nope, but some of it's still attached.
Yeah.
The polyps are still out. They haven't fully retracted. So when they retract, they look like they're little sort of crystal squares on the side. I'm not seeing any movement. Are you? I'm not seeing your oaf react either, which I was sort of wondering what your associate would do.
No, I know.
This is not what I expected.
Yeah.
That's always very interesting.
So
Well,
So if you poked it, I wonder if the polyps would go in. We've seen before on other colonies. You know?
I don't know.
I don't know either.
I think we should try it again in a different kind of coral with
Yeah.
with big outstretched polyps before we abandon this. But I'm very surprised. I mean, again, just playing with the hypothesis that if food is scarce, I know that nothing happens quickly in the deep sea, but if you have to grab a particle quickly before somebody comes and takes it from you, but nobody's coming to take it from them. And they do have, the oaf hasn't moved. The coral hasn't moved. But it is sticky, right? So you can see immediately how there are nematocysts. There's sticky, stinging tentacles and the mucus they have are able to trap particles. And now I wonder if it's just, once it's there, you get this slow game of,
Yep.
I don't know. It's not what I expected. All right, well, can I make a suggestion? I think we should get ready to fly, but we'll come back and once everything's put away, let's look at this coral one more time. We'll give it its privacy for a second and see what it does, and then come back, and then maybe we'll try this again in a little bit. But
Yeah.
it's really cool. I mean, this is how you learn. I mean, there's an element of curiosity and of sort of experimentation that happens when we're here on the seafloor, and
That's right.
trying new things is always really exciting. And just we try it once, you try it on one kind of coral, then you try it later on another kind of coral, and we see what we see. But I would have thought there would have been, in the shallow waters, this would have been eaten immediately by everything. And so at 1,379 meters, it's a different story.
Well, I'll tell you I forgot what you called it, Randy, but the device worked pretty well, right?
The device worked perfectly, and I think the chefs did a beautiful job. They created a food stuff that was completely compatible with the device, and the ROV pilots, everything was perfect.
Yeah.
Maybe we just have to revisit our recipe.
No. I think we need to try a different colony, a different kind of coral. I was interested in trying this one. I was particularly interested in this one because this is the family that we've been using for lots of other experiments.
Yes.
And so I wanted to try this one, even though I knew it would be a little bit tough. So,
We'll get the lobster out next time. We'll see.
There you go.
Maybe that's better.
Yeah. Well, thank you so much.
Thank you.
What's that?
Can you call us when you guys?
Yeah, absolutely. Wonderful.
Absolutely.
Okay, thank you very much.
Thanks,
Chef
guys.
Pete, Chef Bert, amazing. And pilot's incredible. Really perfect job. That was such good maneuvering. For a world premiere, that wasn't so bad.
Well done. Yeah.
I mean, aren't you
That was
surprised?
neat. Yeah, I think we should go zoom in again.
I
Look at it.
think
Never
we're
know
going
what
to.
we're gonna see.
I just wanted to give the coral a little minute.
Yeah, I mean, these are ones you've been doing most of your experiments on, which is, makes total sense. I'm trying to think what other, maybe like a bubble gum, something with
Big meaty polyps
Yeah,
or a
you could
bamboo,
get-
but
you could-
not the one that was facing
so
down.
you could really image in and see if, anyway.
Or maybe even like an anthomastus.
Right.
Yeah,
Camera yours.
it's
Thank
a big,
you.
big polyp like that.
Exactly.
Would be good.
But I really wanted to see this one as a first one because, like I said, we've been thinking a lot about this particular kind of coral. This is a primnoid. I'm so surprised your oaf didn't move.
Oh, I'm not. The metabolic rate's got to be really, really slow. These corals, for example, just microns a year, so in some cases, but it's not like they're, barnacles. You see this, you see the Syrian barnacles come out, and
Yeah.
they're actively moving and pulling back in again all the time. And we just don't see these polyps on corals doing that a lot, right? I think they just stay stationary for so long. They don't need much to grow.
No, clearly not. I mean,
Right.
they're letting it go.
Right. Yeah.
Really interesting. Okay, well, I think we
Yeah.
learned something here, and we'll just keep going
Yeah.
and trying it again at some point.
Yeah.
Is that okay with you?
Yeah.
All right.
Yeah.
Thank you so much. That was an
Thanks,
incredible
guys.
job. World premiere.
Good job, Chris.
Going northwest.
Never been done before. It's a matter of good.
All right. Yeah.
Wow.
Yeah. Well.
All right. Onward we go through this beautiful landscape.
Yep. All right. 1379 meters. Oh, goodness gracious sakes.
All right, that was really helpful.
Things you get me into. Alrighty then.
I
Here
get
we
you
go.
into beautiful
All right.
coral reefs.
Yes, you do.
Ancient
It's
coral
fabulous.
reefs in the middle of the Pacific
Just
Ocean.
fabulous.
Look at this landscape.
I don't think we have focus
I don't think
over
I
here.
do too.
I don't know what.
No. Look at that wall.
We can lighten up though. Super grainy though.
That's because we're
See the
we're
bottom?
pretty
See
far
much
up.
Better?
I think. Do I
our-
have manual focus? I do.
Let's
see
take the
it.
ISO down.
There we go.
And now Samia.
Oh yeah.
There you go. It's better.
You wanna let's.
I got it. What? You're looking better there. Another primnoid. 2124. 1374. Now up Samia.
Well, let's go take a look at this coral.
Up.
Can we? Sorry. Thank you.
And primnoids.
Yeah.
Move to the other side.
Alright.
You'll see a better picture. Which is what we're seeing in the Geological.
Don't think I've seen any cup corals today. Yeah.
No.
We haven't. We've only gone.
Yeah. We haven't gone very far.
Where do we start?
No, we've gone 2 waypoints. That's pretty good.
There should be a ledge off that north. This right here.
People don't write depths in here. So we were 1465 more or less when we started. So we haven't even gone 100 meters yet.
There's no associate on this. That's why.
I don't see.
And look at how tight. What's going on this? It looks like there's no.
Those are dense polyps.
No, I don't think they're. Are they dense or are they stripped?
And there is that a gastropod on there too? I don't know. Stripped?
No, they're dense.
Oh, I see where you're. I
Look
think
at
they're
how
just
smooth
dense.
that is. It's almost.
Yeah.
Have you ever seen that? Or is this just me?
Well, they're all pulled
They
in.
look like icy.
There's a couple things on there. Yeah, it is different.
No, they're just pulled in.
I seem to have these bifurcating branches.
So these are the same kinds of corals. This is a primnoid that we were just looking at. This is what we've been using our experiments on. But the reason I'm drawn to this is, like, we've been working in between the polyps. There isn't even a juncture. Like, this is tight, tight,
Yeah.
tight.
You're right.
Like,
Yeah.
really tight. And look how.
They just seem really dense to me. But that's they are all pulled in. So maybe, but you're right. There's
What is this?
not much space between them. I don't know.
Here we go again.
You're way in there. I don't know.
But 200
All right.
ground level, there's not working.
Well, that was cool. All right, thanks.
Any interest in collecting, Steve?
Steve, you interested in this one?
I don't. He's gonna say no.
He's
We
probably.
know what it is.
He
It's
knows what it
Norellis.
is.
It's Norellis.
Okay.
It's just dense polyps.
Okay, great.
So, okay.
Thank you.
Doesn't make sense.
Yeah,
It just looked very different to me, so I wanted to take a
does.
close look
I
at it.
agree. I agree.
When we turn the light on.
While you're suffering with. Here's what I'm going to do. I'm going to take the ISO down and take. It's too bright.
These two right here. What you're talking about?
I'm trying to bring down the ISO, which is this makes grain. And this one you want to turn up more. Sorry, I always go the wrong way. I think they're opposite actually.
And we got a bunch of bases.
Wow. This is not working out for me.
No.
Sorry,
I would-
dudes. Those are holdfasts over there.
Yeah.
Some of them. It's just not tweaked the way I'd like to have it tweaked. having to put more ISO up, which makes it super grainy.
Mm-hmm.
So what I want to do.
And it's also because we're pretty high above, right?
No, we are up. Still.
This is what's called too much good imagery, too much control.
Well, I'm just they
Yeah.
they gave us ISO now it's.
You want us to take it there? We'll just play it.
Well, I'm just trying to get it set so it's.
No, so, yeah, just
Yeah,
I'm
you know what I mean?
quite high up here. I'm just trying to get
Yeah.
back to our.
Yep, that's fine. We could also white balance pretty soon too. Get a shot here.
What about that coral?
Can do. Let me turn something down.
Now you're using fun.
That's what I want to do.
That's pretty good.
Sorry, Randy.
That's all right. Do your thing, chicken wing. Here, let me bring this closer to you so you can have all the control, Tim.
That's what I want. That's really what I don't want.
I
any
want you to have
control.
it. I do. I don't know what it is about
This
us,
is white. I
especially
don't
in this
know.
particular spot.
It's a funny bone issue.
It is.
It's a funny bone issue.
Digging your ball in there.
All
I
right.
just think it's a fresh air. Of course. Just 'cause it's dark.
Yeah.
Ready?
How's it going?
Oh, it's captain.
Oh, yours.
I can't do it right handed. Nothing. Jeez, Tim. All right.
You can like kind of shift.
See, I don't know why. Here we go.
These are not as
Okay.
tight, see? But they're all still polyped down. Marilla. Okay.
These are different to me. Do you want to try to white balance on this or not?
Yeah,
normally
sure.
I like something
Let's
white.
white balance and then
You can
let's
white balance too if you want to.
and then let's go, I think.
Good times.
Rock and roll.
Thanks. Having a hard time today.
So different. Let's go.
All right. Yeah.
We can zoom in on the sanders.
That's better to white balance on, right? Actually, something that's white.
Not the bright
Like-
red or
like-
pink oaf in the middle.
Oh, I know, but like,
Do you want me to move back to the line? Are you happy kind of just to this direction?
how tight is that on the?
Oh, no, I think
It's
that's
not-
fine. Anywhere
doesn't
along
matter,
that. As long as
really.
we're moving towards our next waypoint. That's
Yeah.
good.
Done.
Des was looking at the map and she said that if we go to the other side of the line, it might have a better view of this.
Sure.
The
Sounds
ridge?
great.
It looked all the same.
It looked all the same to me too.
Me three. But Deb has magical abilities to see things in maps that the rest of us can't see.
I will, stand by one.
I don't know why I am having such a hard time with this thing today. Nothing's working for me.
Oh, Tim.
I know. It's like I've gotten to a bad
Okay,
place
so
and can't
we
get
are
out.
right here. If you look up at your
Yep,
map.
got it.
So this side of this line has more of a ridge here.
Yep.
So
We
if
could.
you wanna be on that ridge.
Oh, I
not
see, on that-
across the sand, you wanna sort of slide to the more northern, this
Yeah.
is.
Yeah, that sounds good.
To this side of that line, and then it kind of curves around, but
Sure.
just, as you're going up towards waypoint 3, start going more north.
Okay, I'll head a little bit north
Yeah.
here, and hopefully we'll.
Yeah.
Perfect. Thanks,
Great,
Debs.
ROV, that's a new waypoint for you.
I am so perplexed. I mean, the particles were big, so there may have been all kinds of little particles that were consumed anyway. But there was no movement.
You don't know. You know, they have fed Lophelia in tanks before.
That's what Alexis was
Actively
saying.
seen them close or whatnot.
Yeah, and Alexis said it looks just like a tropical coral eating.
Yep.
So we're learning
I
things.
understand. I know.
think it's interesting. Look at this landscape.
You can see our track up slope.
You can see this pavement, right, which is evocative of what we've seen in some of these other shallow reef spots,
Yeah.
which is just
This
wind
is just
pounded.
drape of sediment right now.
Exactly.
Not seeing much in the way of sea pens.
Corals here.
Yep. And a sponge. You guys
Oh, I
collected
see.
a sponge for us, right? Yeah.
Yeah. There's a euplectellid, and then there's
There's another Norella right next to it. Kind of looks like it.
Gorgid.
No. Oh yeah, on the top, the bottle brush.
Oh, look at that.
Okay, keep going. Yeah. Thank you.
Yeah, I don't think he knows that.
Camera is better now.
Half an hour.
Just gonna say that.
Who would have the rail control right here?
I think this one. Yeah, easier.
We're right here.
One just should be there.
Oh, there we go. There was one that was screaming loudly and then it was.
It's a rock.
Look at
Right.
that. Yeah, I agree.
That's what I'm thinking. You want to see that? What is that?
No, it's a rock.
It's a
Oh,
coral.
that's a Metallogorgia.
It is.
I agree. Those aren't things I would have predicted. I was tired. Sorry.
Quick snap zoom here, as long as you're stopping them. Earlier today we saw egg mass and what may be an egg in there.
Oh, cool.
Got it. So it looks to be an Ophiocreus ophiuroid and a Metallogorgia. All right, great. Thank you.
Some under that rock there too.
That's
In
the
the corner,
Howard
over
crab with an
there.
anemone.
Yeah, that's good. We've seen that already. We're good on
Yeah.
that too. Another crab coming down slope. It's like a chaseon.
Just tell the sun, hydration fluids.
What? Oh, good. Perfect. Look at that. Let's go look at that feature for a minute. Just, it's very different.
So there's more above it.
Want to follow the trail?
Sure. Yeah. It's also scalped under there too, right? It's under that ledge. It's like this.
Yeah.
Come down.
Yeah, you're right, it is.
why the color temperature of these monitors is so different.
These are the, yeah, you see the difference between
They're all, yeah.
they're just,
Fish.
Right.
you can see like some up here, that stuff's burned into the image.
Yeah.
So I think this one's got, you can see some lines through there.
Hang in, here we go.
I'm good.
Coming up right here.
I'm just trying to stay awake. I
I
had
know.
trouble sleeping again last night.
I hear you.
Oh, there we go. Lixurids.
Yeah. Anything you want to look at?
Behind it too.
Yeah. It's bigger like a
Just
bamboo
a couple
behind
of
it.
snap zooms and a
Here.
yeah, just to make sure we know what it is. Actually, that might be a Acanthogorgia instead of a. It's hard to tell.
Looks like a plexar down below, and
It's
Adam's
probably
stable.
a plex. They look for probably the same thing, but it looks a little different to me. It's the only reason I am.
Come on, Shank.
asking.
Let's go, Shank. Come on. I got it. Yeah.
Little nice trims on it. Oh, Steve's
Huh.
typing. We'll know in a minute.
Yeah, I don't know. I see some.
Okay.
I'm saying it's a plexard.
Yeah. Steve says it could be different. It's tough to tell. The last one was pretty haggard. I agree.
Yeah. Does he want some of this?
I don't know. Steve, do you want some of this?
There's some polyps. Some mysid shrimp here.
See, look at all. It's,
I know.
to my point, you see some of the polyps are open, some of the polyps are closed.
This side moves.
The oaf is waving around. There's more activity here in general. There's food particles
Funny.
bouncing all around, but I don't see a coral grabbing one.
The polyps that with the that the oaf is hitting are not closing.
What?
The
Yeah,
oaf,
exactly.
they're still open in
Yeah.
many cases.
Well, no, it's closing right there as
Which
it's
one?
waving around. It just closed right in front of us.
Yeah, but
See, this
Yeah.
one's good. This one, watch, it'll. If it. I just watched this whole section. Steve says, sure, a little clip.
a little clip. Okay, can we get a really good?
We have not gotten any water yet, so this would be a good place maybe to pop a disc in.
Do you want that little top part with the three branches?
I'm trying to figure that out right now. I'm trying to see if we can't get part of an ophiuroid, really the middle star with it or not.
Okay.
But I don't think so. Unless we took the bottom branch down here.
You take a whole bunch or have to cut one in half?
Well,
Shucks.
Maybe.
what about
Are
the one to the left
are
of it?
Lasers. Are the lasers on?
What about this one down here? Is that
How
an
big is
easier
it?
one to
Yeah,
get a
no.
piece of? Why don't you see where the oof is on it? Could probably take that top piece and get part of an arm.
Let me see here. Yeah. Okay, 10 centimeters. It's actually not much right there.
It's
You
not,
know
it's
that.
not. It's very small.
Okay.
Tim, if you don't want to get a big piece of the coral, you might be able to come.
Where? I know.
You want a stick?
Yeah. Might be able to go. I'll get closer.
It's alright. You get the wings.
I'm super tired.
Sorry, it was a big morning.
No, it's alright. Big morning.
The
Yeah?
feeder 3000.
Yeah, yeah.
Yeah, we're pretty good.
I thought so.
I thought it was great. Did
Yeah.
you see it, Al?
I didn't.
We're gonna try it again. So, but it was very cool. The chefs came in, and we have a menu written on the ROV,
Okay.
and
So,
the device worked perfectly, but the corals didn't eat it.
so, Chris, what were you saying?
I can't really. I can't
It was
really
amazing.
do it anymore. I was gonna try to come right over here. There was a bit of a gap.
Yeah.
It would have taken about a considerable bit of the
Big chunk of the coral, right?
coral, but I might have been able to get a full other animal on there, but
Right.
kind of hard for this end of the.
All right. Well, it's not very big. What do you think, Randy?
I think it's your choice.
Yeah.
This is for you and
yeah.
for Steve. I do.
No, I know. But Steve needs some branches too.
Right. I think. What do I think?
We can-
If it were me, and you weren't not in the room, I would cut down here, get
That's
part
right.
of an arm, and just
Yeah.
take the thing. But if,
That's
I don't know
what
if an
that's
arm is
what I
enough.
thought. Okay. You know.
Part of an arm?
Okay. Yep. That's fine.
But if you want more than that, by all
No.
means.
It
Okay.
Oh, okay. Sorry.
So another
Yeah.
mystery.
Sorry.
Sorry. I didn't think we were gonna do
So
that.
what we are
Yeah.
discussing right now, we didn't mean to have the live stream off. We got distracted. We're discussing is this hole.
With a little crinoid next to it.
Yeah.
Looked like the eye of an octopus
I don't
when
think
I first
so.
looked at it.
Now I just think it's just a
As
hole.
long as,
I
it's not,
don't
as
know
long
what
as
to
that's
say.
not the edge of an octopus right there.
No, you can't have a coral growing on an octopus.
What?
Tim.
You can't?
Watch an octopus like.
But do you see that? Look, there's a little shrimp behind it. I
crab,
wonder.
that
Well,
crab.
I'm wondering if this is somebody's home.
I don't know.
I don't
All
know.
right. We're ready, you guys. We were just conversing.
Listen, I think so. I
Yes,
think
sir.
I think Tim makes an excellent point, which is if we don't get the sender of this ophiroid, we're never going to know what it is. So do what you need to do.
Now that I look more at them,
But that's
can't
a massive biological
Yeah.
collection. We
I
should
wouldn't be
probably.
surprised if that was an octopus now I look more at it.
What? You think that's an octopus?
No, but
No.
Coral base is
Yeah.
growing on it.
It's exactly. It's a black hole into the center of the deep sea.
When it appeared.
Al's right, it could be an old base of a coral.
No, that's
Yeah.
what we think it is too.
Yeah.
We're just being
Okay.
silly.
Think of that.
All right.
I'm good. Let's just sample.
Is this the arm?
Let's just.
Okay.
Yeah. It's fine. Just, you know, it'd be massive by a box sample.
Yeah, yeah, we don't wanna do that.
That's fine.
They're just saying, hey, poor Tim.
It's
I
fine.
don't know about what, but
Doesn't matter, does it? It's just whatever it is.
poor Tim.
The portion of the day.
You win the sympathy vote.
Poor Tim. What happened to you?
What doesn't happen to me?
Oh, Tim.
Golly.
Poor Tim.
Poor Tim.
Poke something in it. It wants to poke the eye. Thanks, Nick.
Give me that stick. You can poke it.
Thanks, Nick. He knows it better
Aw.
than. Okay. All right. Well, let's get focused here.
I might note that poor Tim is
It's-
doing just fine.
No, it's fine.
I
Yes. Wonderful.
What-
Mhm.
What's more with feeling?
Yes. All right.
All right.
If we could come down into here, if that's okay,
Yeah,
and take that.
just whatever I get.
Yep. Whatever you
Whatever I
get. Whatever you get. These arms do grow back.
We now have multiple lines of evidence to show that, which is pretty exciting
They
on
grow
all these
back.
different
Oh, yeah.
corals. And of course, the ophiroid arms do that too. We've seen lots of ophiroids recovering.
Oh yes,
Strip.
absolutely.
Oh, yeah.
I've always wondered what's been nipping the arms of the ophiroids.
I was
but,
thinking about that too. Fish?
that's what I've. Yeah, that's what I've come to
It's
think.
pretty
I don't
pretty
think-
neatly clipped.
Yeah. Ooh,
How about that?
that's fine. That's fine. Let's.
Oh, I didn't really get much anything. Well, I got all of it, actually.
Oh, after.
Well, biobox it is.
Bio-
Well,
you
we'll
know,
try it in the
I know.
respirometer. Now
That's
we've got
how it
enough
goes.
tissue.
Yeah.
We have a respirometer
Goodness
with us on
gracious.
board where we've been looking at the basal metabolism of corals, and as we lift up the bio box, you'll see we have a menu written on it. And part of the reason we're interested in coral respiration and metabolism is because I keep thinking about energy budgets in the deep sea. So that's pretty cool. And then one of the other things we're doing are immune challenges, trying to understand the origins of innate immunity and asking whether or not corals' immune systems work the same way ours do and respond to some of the same stimuli.
This will be 473.
On this B,
Yeah.
That works.
so is this a putative acanthic order, or are we calling it a pursuer?
Buxard. Question mark.
That was astroschema that was on there, right?
Yeah.
Okay, great. Thanks.
Anything else you want to look at here?
No,
No, we're good, thanks.
thank you. Sorry, I should have let you answer.
No. I just,
I was just,
yeah. Okay, I
I
didn't
went-
write this in. Hold on, sorry.
no,
I
it's
just forgot to do that. FK 21.
File box B.
Yes, 473.
Well,
Well, Randy, we have quite a ways to go.
I know. That's okay. No?
No. Let's go.
All right, here we go. Up the ancient reef walls. Another one of these tunicates as we pass by.
So we're nominally going point. We're really 0.1, 0.2 meters per second.
Yep.
What is that little?
Well, I mean, it's averaged.
Yeah,
Yeah.
Where-
right. I mean, that's,
Yeah. That's what we're working the shift.
I would have
to.
guessed 0.5, 0.4.
Well, in vertical, but this is over ground.
Right.
So,
It's both, right? So
yeah,
it's
but
like big sponge. You want to see this?
Yeah.
Nope. Do you? Not unless you see
No, I
spongivory.
don't. No.
I mean, it's a very nice sponge. I'm happy we know that it exists.
No, it's already in.
Yep.
I don't see any
Little
shells.
sponges. No, I don't either.
Just fits.
It's a very weird landscape. It's beautiful, but
Little to the left there, back to the left.
that's a primnoid.
Where?
See?
Oh, okay. Are we good on that?
Yeah. I think we can get a just
Drive by?
get a drive by and keep going.
Going really fast.
Yeah, keep going. There's a little cup coral at the base. Let's just note that.
Zuma. There's a cup coral at the base. Why is this so difficult today?
3-point connected mucus.
More importantly, I want to see the cup coral.
That is cup coral.
So you see some other things.
All right. Note the cup coral.
Note the out of focus cup coral.
Well, Tim.
Oh my God. All right.
It's trying to hide. So we're looking at a little cup coral here. It's a scleractinian, a deep water scleractinian, 1,320 meters below the surface of the seafloor. This is the first one we've seen on this dive, to my knowledge, at least. We are, where are we? We are on the Winslow Reef complex on an unexplored seamount until today. We finished mapping it last night. We're diving it today for the first time, and we are climbing up the shallows of this, or from the deeps to the shallows in this ancient reef, which tops out at about 300 meters-ish. All right, well, let's keep moving. We've seen this.
Just keep going.
Same one. All right. We are just at the very edge of the U.S. EEZ, Exclusive Economic Zone, in the equatorial Central Pacific. And what we are doing on this expedition is trying to characterize habitats, looking for sponges and corals, looking for coral and sponge associates, of those associates looking for corallivores and spongivores, looking at the predators, looking at the wounds that they leave, the potential infection of those wounds. We definitely want to look at that, and maybe this one too.
Want to see this one?
I don't know. I think it's the same plexurid we just collected, but we should just look at it.
Yeah, I'm okay
And
with that. Look at that.
It's a bamboo whip on the bottom. And we're also looking at, in these wounds, we're looking at wound healing and coral immunity and microbial communities that are in the area. I don't know yet. I don't know yet.
Oh, either.
So we have a very ambitious scientific portfolio, but the workflow is really smooth, and we've been at this. This is our 15th dive.
It is?
14th full dive,
Somebody
but
said
15th
okay.
dive technically, and we are getting it done.
Not convinced. Oops. Coming out, coming out. Leave it there.
Just came out to say, like, what the heck? I didn't read data in 14 million places. Why is he saying
Oh yeah, yeah, that's right.
Okay. That's right. That's good.
So Steve says this is a white swiftia. It's a very strange colony shape. It's very large. I agree. It's really caught me off guard.
Yeah. I have not seen this before, I don't think. I'm going to zoom in for some more of the polyps. It has a swiftia branching. You can see down below there. It was.
So delicate.
I'm also looking at these associates.
So if we can get a small piece of it, that would be great.
Yeah.
I don't see the zoanthids. Oh yes, you're right, Anna. Oh, look on the bottom. She absolutely called it. Good job, Anna.
Sorry, I'm trying to still get focus on this thing. Hang on, folks.
Okay, so we're going to be
Let's
diving
go
all
here.
day. Let's see, it's 11 o'clock now. We'll be diving for at least
Do you have focus over
another
there?
8 hours, maybe 9.
Did we get our focus? Are you got the button on there? You got the button on?
Yeah.
Okay. There you go.
What?
Keep going, keep going.
All right, there we go.
That is a gorgeous colony. I want to try.
So we want to collect a little bit of this. There are a variety of associates on it. Seeing these amphipods, these red little crustaceans, and there's also looks to be a hermit crab on there. Quite a bit.
Actually, no. What's that? And you can count which.
Or know that we're gonna feed again if they're interested.
Yeah. Just like try to
Hi,
just see
Vern.
over the.
We're feeding again if you're interested.
Yeah, I can come in a little more.
Okay. Yeah, bye.
And I'm gonna call the bridge and let Al know too.
No, the bird's making lunch, so Peter might be up.
It's interesting. I want to see the base of this too. This coral.
Yeah. It's a really cool one.
Yeah.
Hey Adam, maybe try the other side this time because it's smaller and these are really small polyps.
What do you mean?
Okay.
The back side of the coral?
No, the two tubes. One has a bigger hole than the other one.
Oh, sorry. Yeah.
So let's try the smaller hole.
I would try this one. Sir. Oh, I got nothing.
No, please. Guest of honor. So we are joined again by one of the Falkor chefs.
Hello, Internet.
Want to say something about? Introduce yourself. Say something about you.
Okay. Yeah. For everybody who wasn't here the last time, my name's Pete. I'm one of the chefs of Falkor and was collaborating with head chef Bert in making the seafood cocktail we're trying to feed to this new species of
Swiftia. Yeah, coral.
coral, not algae.
No,
Coral.
yeah,
of course.
definitely coral.
So let's see if this one is less picky than the last one.
I don't know. I'm not sure.
Just true.
So you want to try the other push, right?
Yeah.
So I'm behind it.
You can see that. It's perfect positioning.
Can we get a little bit closer to it, or are you happy with that?
I am going to want. No, I think your position is great. Once you have deployed, I'm going to want to zoom way in on the polyps as usual. As usual, as if we've done this 20 times. This is only the second time this has ever been done. I think you should try the other. Yeah, you got that. Okay. It's going to be harder to push because it's got a smaller hole in the bottom.
Only one of the syringes have been widened. Okay.
This is the Coral Push Poppinator 3000, invented by our fabulous ROV team, and our chefs on board have created the inside dish du jour.
Dish du jour, yeah, the fish of the day.
Catch of the day, yeah.
It's barramundi for everybody who wasn't here.
And we are going to try pushing just a very little bit and seeing if these corals react. I can see the argument to be made that they have very slow metabolisms and therefore don't react really quickly, but I also think that food is so rare that if something falls on you right away, I would think there would be an active movement to go capture it. And so I'm very curious to see what happens when we try different coral species.
What are they feeding on normally?
The marine snow that's drifting down, these little
So
particles.
will they actually recognize it as food, even if it's get caught in there, or is it so strange to them that they, even though it would be good for them, they wouldn't recognize it?
I don't know that it's that strange to them. I mean, having fish flesh or, you know, seafood pieces might, I think if you fed this to the tropical corals in my tank, they would eat it right away, the shallow water corals, no question.
But they are close to fish.
Yes.
These aren't, so...
No,
or
there's
there are a
plenty
lot
of fish down there.
there?
Yeah.
Okay.
So, you know, it's
I
not the
thought it's
same as
tricky
marine
for-
snow, but I think, I don't know. This is why we're trying it. I can't get into the head of this coral. I'm not a coral psychologist.
I wouldn't be the first time my guests refuse to eat my dishes.
Oh
so-
no, your food is delicious.
So, I think that we're in a good spot, but you can go ahead.
Yep. Tell me when I can zoom.
Yeah, I think we're in a good spot.
Okay. I'm gonna go in the coral while you're playing with the top.
Alright.
All right.
Just get to the coral and zoom in just to make sure we're focused before we ask.
This is what I said.
All right.
Should be in manual focus.
Yep, I'm working on it. Nope. What?
That's me.
Okay.
Stand by. I'm just gonna take it for a second.
So all the polyps have closed.
How's that?
Wow, well, all the polyps have closed up.
Well, that's not right.
It's not gonna work. Hang on. Let me see if somewhere on this colony the polyps have not closed up. Well, they can move quickly, right? They all closed pretty fast. Yeah. All the polyps are closed. We could try pushing just a little bit anyway to see if they try to open up to capture it, but try not to push too much out because I don't think this is going to be a really
Okay. Well,
solid
do you want to
test.
just hold it in the hand and we'll find a different one?
Yes, let's do that.
Okay. Alright, so I'll go ahead and secure the,
But we have to collect, no, we can't because we have to collect a little piece of this too.
Okay.
We want to also collect a little piece of this coral, so we have too many agendas for one thing. Do you want to?
Do you want to find
What I
another
can
coral like this
do-
and collect that one instead, just so we don't have to...
All right, so the coral has closed on us, so we are going to not feed this particular coral at this particular time because it doesn't make sense. What we're going to do instead is transfer the push propinator to the other arm so that it's ready to go next time we want it. And then we're going to collect a little piece of this coral and then move on our way. Sorry.
All good?
Would you like us to call you again when our moment
Yes,
comes?
please.
Okay.
Yep.
As long as it's not annoying.
But just the thing you called.
But they can react really quickly, obviously.
No,
right?
they're absolutely. So those are mysids on there.
Yeah.
Yeah. 2208, 13, 14. Okay. So what's happening now? We're gonna transfer.
They're gonna just transfer it from one end to the other, so they don't have to go to the basket every time. It'll
Yeah.
just be hanging out in the arm, ready to go.
Roger.
And then they're gonna clip and
Okay.
collect.
So then we can go back and collect here.
Exactly.
Just image
Because I don't know if we're
quickly
gonna
and then
find
collect.
another one.
That's
They
fine.
sort of suggested we could go find another coral, but we haven't seen this yet, so I'm worried we won't find another one. And now that I've said that, it'll be the most common thing on the
Yeah.
seafloor, and it'll be everywhere.
We'll just do a quiver collection here.
Yep.
Okay. Excuse me.
Ah, nice hand.
It's locked.
Such a good handoff.
Like you have done a lip there. Rotate your wrist to the left a little. There you go. There you go. Got it.
If you hold it, I can just bring the other one down another one. Yeah. Wanna do that? Yeah. Frozen? Frozen. Jan, make sure it's not colored or anything, right? Okay, frozen.
It's really delicate
All right.
handiwork.
Just wanna put that away for me real fast, sir. Be so kind. Alright, it's too hard for me. Speed rotate. Speed this up all the way. There you go. Side seat. And maybe with each other. Alright.
Can you slow it down?
No, we want a piece of this.
Oh,
coral
right.
here.
So it's gonna take like a minute of imagery and then
Yeah, so
we'll
you-
collect. What you saying? Right. I just want to double check this thing. So we've got. I want to go into the
We have a quiver over and stuff.
there was a substantial base here. I thought, right? This is it here.
Yeah. It's small, but it's a small profile, but it's a pretty serious coral.
Okay.
A lot of branches. So this is a Swiftia coral.
All right. So it's got
with
zoanthids on it.
Zoanthids on it, exactly. And a bunch of little shrimps. Nice shrimps.
Do you have focus? Or is it me?
I don't know.
Tell me. Is that in focus? Several mysid shrimps here. I was trying to see those are
See the ophroi?
see the ophiuroid, and then there's the mysid on the end here. Many mysids. There we go. Okay. And then the figurid.
It's a really terrible shell. You see how there's 2 holes in it?
Mm-hmm. More O's. Okay. So we want to get a clip of this, and I'm trying to think if
You want to take the end with
That's a
the
bit
override on
long.
it?
yeah, let's try that.
So you think there's one right there?
See if we can do his.
All right.
Is that okay? And a quiver. Be great. You got the camera, sorry.
Yep. Yeah, I'm gonna get there.
Let's do what you said too.
We'll stand by. Look at that. Well, that's the thing.
It's a swimming sea cucumber.
Yeah.
Holothurian. Checking out the ROV. Getting a tour of Sebastian.
Mm-hmm.
So I've studied hermit crabs in terrestrial environments for a long time. They would do anything to get out of a shell like that. It really shows how scarce shells are. You know, I don't ever see the gastropod that made that. Do you?
See what? See the guest?
Huh?
That little brancher?
Yes.
Yes.
I think that's great.
Right where you are. It's great.
I want to say I don't think I've ever seen the gastropod that made the shell that the hermit crab is in.
Ah,
Have you?
maybe. I may have it in the lab.
Huh.
I don't know.
I mean, often they're showing us shells for creatures that I don't see.
Yeah. True.
It really goes to show you how much diversity there probably is here that we're not able to see because it's tucked away in nooks and crannies.
Oops.
They'll climb right back up.
Bonsai. Okay.
Should be all right.
$474.
Right. I don't know how you're amazing condition. See. Super bright.
474.
Yep.
Thanks. And it's going into quiver 13.
Yes. 13.
Thank you.
Did that have any associates with it too? Or is it just the septum?
I think it had one ophiuroid. Yeah. Okay. So are you one of those investigators who remember hearing this ingenious way of collecting hermit crabs to put shells out that are attached like with thread to a bar?
Yeah.
And you have a bar and you have all these threads coming off and attached to empty shells and you put those out.
I did not
And the
do
hermit
that.
crabs go in there and you take the bar away, right? So you don't have to go looking for them.
I did not.
You
I've
know,
never done that.
I thought it was ingenious. I don't know why I did.
Tethered.
No,
I have
so-
done other tethered shell experiments though, which has been
Yeah,
pretty
tethered
fun.
shell, right.
So
Great.
what have we done? I was trying to figure out how terrestrial hermit crabs, whether or not they recruit other crabs to the site
Yeah.
of an empty shell. Because when
Yeah.
we've discovered these vacancy chains, when you have a large empty shell and you have all these little ones, we call them waiters, waiting for the catalyst crab, the
Yeah.
crab of the right size, to start this huge cascaded chain.
Oh, right. That's right.
And so in order to figure out whether or not the cue for recruitment was visual or chemical or physical, we had crabs. I had a tethered, what the fuck did I do? I had a shell with a tethered crab to it, the first waiter. So it was the recruiting crab. And I would put them in, I would leave them exposed. I would have them in a Tupperware container that was clear with holes. One with a Tupperware container that was clear, no holes, black with holes, and black with no holes, so that you could isolate visual from chemical from physical cues. And we looked to see who was more successful, which one successfully recruited more crabs to the site. And
Great.
I'm trying to remember the results. This was years ago. I think it was it's totally visual, but visual plus chemical was slightly more effective. So I think they're releasing pheromones that sort of say, because a little crab was going to wait by an empty shell for a long time, even if it's way too big for it, just because they're so rare. Empty shells are just so rare. And so the faster they can recruit more crabs to the site, the faster it'll catalyze a chain, and they'll be able to get a shell to come in. So just sitting there and waiting is no good. Sitting there waiting and recruiting more crabs to the scene, better.
How was the coral?
Right.
So we used a tethered crab experiment for that. But no,
Wow.
that's a really
That's
smart
great.
idea too.
No, I love it.
Has not eaten yet, let's put it that way.
Huh? All right.
Okay. So I grab this real quick.
You got locked in. It's locked in.
Polyps in or out?
Just think for people to like.
But it was one of those things where we started
They're
playing
all
with
in.
hermit crabs because of a storm. You know that story, right?
Are those polyps open again?
No, they're
No,
closed.
they're closed. Okay. All right. You ready?
Let's fly.
No, I haven't heard that story though. So square up, and then
Yeah, we were working on a shallow coral reef, and there was one right next to it. The conditions got rough. It was like stormy for a week. We couldn't get into the water. We couldn't do any shallow water work. We're just stranded. This teeny tiny little island with 6 humans, 63 palm trees, and we counted them. We did the mark recapture study, 1,084 hermit crabs.
yeah, wow.
And we found vacancy chains, and we were really stranded on land. We'd worked through all of our samples. We'd worked through all of our data. We were just waiting to get back in the water. We started watching hermit crabs and watched this behavior and characterized it and started doing a bunch of new things. It led to a whole new line of science.
Fantastic.
It's something.
Wow.
It does seem grainy, doesn't it?
It's very
It
grainy,
does.
and I've just been.
Decrease the shutter speed to something.
Didn't know something changed. I'd love to have
Over there
that.
for a second.
Yeah, sorry. Yeah.
That sponge. But Tim, you've got lots of projects like that too.
Decrease the.
Yeah. There you go. Something better. How's that?
Yeah.
Well, actually, it's a little low.
I mean, did you take the ISO down?
It's at 50 now.
Is that another crassagorgid in the
It's
back?
better, right? Sorry, Randy.
Is that another crassagorgid in the back? It's long.
It is. You can see the crab in it.
Mm-hmm.
Which one, lower or upper, sorry?
No, we're good.
Okay.
We're good. We're at 1309 meters. And the last waypoint we have is at 279.
Mm-hmm.
What's the
But it's vertical, like
It is, yeah. Once we get up,
what's the top depth?
279.
Oh wow. Is this one an island?
No, but it's on a very shallow guyot.
Cool.
It's like the mid-deck was 199 last night, so.
Oh, really? Into the Pasiphotic.
something over there.
Fish. Oh, you see the coral.
Yeah.
Oh, there's a big one here to the left.
Can we take a look at that?
Oh, worse.
Look at that.
Corallid?
Let's go see.
It's huge.
Yep. Let's see what we got here.
There are bits of broken pieces in the bottom.
Yeah.
It's really clear that it's brittle. So I would call it something in the
We're gonna need to
gravity.
just sit down for a second here. Look at this. Okay, thanks.
I don't see any associates right off the bat, do you?
No.
There's some zoanthids. There's one. There's an ophiroid, you see?
Yep. Polyps out and polyps in.
Polyps in.
I think that's a corallium.
I agree.
But I
Look,
don't.
more evidence of regrowth. Can we
Zoanthids too. Yeah.
zoom
where?
in on that one? See that one with the broken branch
Sure.
right in the middle? And there's another ophiroid right behind it.
Yeah.
See that? There's 3 little polyps on
I
the
do.
broken skeleton.
I do.
It's great. Hang on, I just want to take a picture. Anna, could you mark this as regrowth, please, in the
I'm all the way in, and I think it's focused.
regrowth? I don't know. Take a screen grab and write and make a little comment.
Should we need to sample this or not?
No,
This is-
I don't think so.
Do you want to swing around to the other side and look?
No, I don't think so. Actually, Adam, this is. I think we have to get the polyps on this side.
Right.
Let me just see.
Look, whatever's making that mucus net is making one right here on the coral.
Mm-hmm.
Which is amazing.
You see the contact between the dead and live tissue there
I
too.
do.
I zoomed in for you.
Thank you. But what's funny is it's just that one piece, right? It's dead on both sides.
Yeah. Those polyps aren't so nice either, right there.
No, but they're not non-existent either, which is also surprising.
Different shell for that crab.
You can see there's.
That's alright. It's just my broken
shell.
phone. Don't worry about it.
Tim.
Just my broken phone.
Again,
Let
it's
it go.
really in a very precarious spot.
Is it?
Yeah, yes.
Okay. I haven't seen anybody ask for
No,
anything either, so.
but can I?
What do you
can
want
I
to do?
ask you to? Can I borrow the
Go.
camera for a minute?
Yeah, go.
Nope, wrong way.
Quite a bit of overgrowth along the branches.
Look at that. Everywhere.
There's a single polyp there.
2 of them. You see lots and lots of
Right,
spaces where
they
it's
are.
regrowing.
They're facing the other way too, but
Yeah, but
it's a,
there's lots of little ophiroids that are just not visible at the first look.
yeah, I've seen them. It's the white morph.
Yeah.
So that's again.
It's so funny how you have a single polyp kind of sticking out in the wrong direction.
Yeah, you see an older section to this colony, like a living section that's older.
Yeah.
So that's at the top.
Yeah. But look at
Hydroids up there too.
even at these nubs. I like this colony. There's lots of evidence of healing from broken off pieces.
Looks
It's
really
great.
old.
It is really old. It's beautiful. Okay.
Do you want a piece or not?
No. Do you? Yeah.
No. That's fine.
All right.
All right.
Okay. Great.
22, 29, 1304. What else was on there? I haven't seen the barnacle.
There was the
Anyway.
hermit crab.
Yep.
You get that?
Yep.
Some
Okay.
mysids.
Okay.
I'm so tired.
You are so tired.
I am.
I know.
So are you.
And it's gonna get better for you when you get shallower too.
I know. I
You're
should
gonna want to
probably
be awake.
Go take
Maybe.
a nap now.
Yeah.
Except that we have the coral push poppinator. Maybe we should put that away.
Yeah.
Now,
Well,
of course, there's a gazillion Swifties.
Yeah.
Now that I said we wouldn't see any more, we're seeing lots of them. This is
That's
their
good.
space. What is that?
The base.
Base. Yeah. Alright, let's keep going.
Base. A little fish there. Shrimps, maybe.
Alright. I'm gonna leave you with Anna. I'm gonna talk to Anna Merne.
Okay.
If you decide to, if you find a perfect coral push poppinator, just WhatsApp me. I'll
Roger that.
find you.
Roger that.
Okay. Banana, you're up.
That's an ocean.
It's cool rocks.
I got some sunset.
I've only seen a couple of antimasses. Haven't seen
We
the
saw
whalefish.
one.
Another whalefish.
It's like a nice
Oh, a little seat vent. Wait. Hold on a second. Sorry. Oh, that's a chryso. Okay. Just a quick image or
Yeah.
some imagery and then. Yeah. Sorry.
Oh, wow.
That's the one we got before, right?
Yeah.
That's the one we got before.
It has two
I know.
crabs now.
I understand. Thank you. Okay, let's go. That's fine. Got him.
Is that a Hamala down in the corner of that white rock?
It's a rock, yeah. No, I think it's more of a Chaseon crab.
Oh,
I don't see the back legs to that. It's just a piece of carbonate. This is a crazy little complex here, is it? There's something up there. What is that? Is that another Swifty up there? Coming up.
yep.
We're just seeing what we're dealing with real fast.
Yeah, no, I got it. It's fine. It's good.
What happened with this?
No, we're good. We were just on that one
See
thing.
the rubble there?
Where? Down below?
Oh, maybe it's just a rock structure. Never mind.
Yeah.
Well, it's not a cool feature. That's flat there.
It's rather jumbled, isn't it? It feels like it's.
Yeah.
It was.
Fracture.
It was once. It was once the Parthenon, and now it just collapsed.
You know, okay, I was really uniform right now, but
It's pretty cool, isn't it?
Super cool.
it's a good thing I had the idea
It's a great idea.
No reaction from that.
Amazing.
That's a good thing I had. That's a good thing I had.
We're okay on that coral too. Just keep going.
Oh, yeah, definitely.
Yeah, look how it's, the rock is fractured and slid over.
Yeah.
Yeah, I like too.
How it's broken up must have been amazing. Another Chrysogorgia bottle brushes with 2 crabs in it. There's another plexard coming up with an ophiuroid in it. There you go. We've hit this Chrysogorgid zone here. 2235.
Is that still,
Is that a star?
hey Deb, on your overlay map there, your
Yeah.
overview, is that updating?
Yeah. There we go.
Bridge ROV, we do waypoint flight.
Fly the edge of this,
Yep,
rock here,
that's
see if
fine.
we get any
Yep.
corals on it.
Okay.
Here we go.
Up or down there?
And that's when we sampled before, right? That's that acanthogorgid-like thing.
There's another, just below it there too.
Another, it's a bramble. Here we go.
Is that a basket star too?
Yes, it is. It's great.
Yeah, it is down here too.
Yep. I'm just trying to figure out which one to look at. No.
That's weird. The track is there, but the box is.
Yeah, we're higher up, aren't we?
Yeah. I see your track here.
Yeah,
That's fine.
that's what it's
I
on.
think we can keep going, actually.
Just keep going, Tim?
Yeah, just keep going. So,
Okay, there we go. Here you are. Just off wavelength 3.
Right, that's where the track takes a little angle to the left there.
Yeah. There's.
Those are all basket stars.
yeah, those are. All right.
Features here. Pop over to this one.
Wow, it's big blocks, huh?
Yeah. Pop over on top. Go back.
Nope.
All right.
Alright, a couple Victorgorgia and a bamboo whip.
Yeah.
There's another Swiftia possibly. That's what that looks like.
That's good. That's fine. I didn't see any polyps on that.
Oil brush.
To the danger zone? Dangerous. Already played with both for
Yeah, I know.
Who's in that?
Very leading
Is that a coral coral?
Yeah, I think it's a bamboo. I think I see the
Oh,
note. Oh
yeah.
no, it's hard to say. Might have been a Versigorgia.
If it was a chrysa. Don't see much of this on to make some tracks here while we can.
Stuff in.
Last 20-minute push.
Whoa. It's a bit red.
Yeah, I didn't see it the other time.
Damn,
There you go.
it's better. Thank you.
Rock? Still sorry. Still going.
Do you ever collect basket stars, or are they too unwieldy?
I do, yeah.
Yeah.
And we did before. I think I recognize those, so I don't really.
Yeah.
But
So then,
yeah,
I was just
Actually,
curious.
you can do more to the west, because it's gonna curve to the west anyways. Or the east, excuse me, east.
is there an underlay there now?
Pardon me?
Is there an underlay?
Yeah, if you just zoom out, you it just
No, I
goes white.
didn't think it was up there earlier.
Yeah, so just, it's pretty white.
Okay. You can see these. Oh, here we go. Seems red again. Just quick. It's okay. It did turn pink on us, didn't it?
Let's try white balance.
Yeah. Okay.
Just gonna zoom into the sand there.
Let's go here.
Yeah.
you ready?
Yeah. All right. Let's keep going. Good.
Get a good picture? Sorry.
No, it's fine. Yeah, right. There's a frame grab. Huh? All right. Can you come out on the NAS screen a little bit? This is. Can you zoom
yeah,
out
oh,
on the
sorry,
NAS
yeah,
screen?
sure, sure.
Sorry.
So waypoint 4 is next,
Yeah.
and then straight up into the shallow, basically. I think that big wall is more like waypoint 8.
Yeah. It's gonna get shallow fast, isn't it?
Well, it's 1240 to what? 290? What did you say? So
Yeah, at waypoint 8 is what you said, 1, 2. 575,
yeah, so
then 387.
700 from here to waypoint 8.
Yeah,
So here and then a big. So this will still be quite an elevation gain.
should
300 meters from
should be.
there.
We shouldn't be in sediment too long, but I don't know. We'll see.
There you go. That's far
Is that for you?
Oh.
In the apartment?
Nice.
We're just heading east northeast up the side of this Seamount, the southernmost U.S. EEZ, just above the Winslow complex of reef systems here.
ski slopes.
Yeah, well,
It's okay.
I'm looking for.
Got a fish, so it's all good.
No. Thought that might have been a sea star or something. I'm not sure if that fish was in Sanafa Branket or not. I just saw it swimming. It's a little crab, another fish just sitting on the bottom. There's a sea star. Could.
Bring the RV.
It's not for.
Yeah, we're gonna try to like just scoot across this empty barren sand area, so I'm gonna go a little faster, that'd be amazing. 0.6. You go. Alright, got faith. Definitely not Wilson.
So I think a Nantucket whaling captain found, well, documented it, so we know about it now in the Phoenix Islands area. It was a ship called the Phoenix. And it found Winslow Reef. It was Captain Winslow. It's a heterocarpus shrimp with anemone. About, there's a couple of them there. It's good.
Never get to see that. That's cool.
Cool thing about it is,
Favorite.
the cool thing is like we have videos, and watching a video is the same as watching this video.
I know.
So yeah.
Is that?
Shadow.
Oh, the shrimp.
Another heterocarpus. Every place you see these little holes here, these little very aggressive shrimp.
Just feels
We just, I think 2 days ago, we dove Taitov Seamount, and Taitov was where one of these shrimp actually attacked a fish and ripped the stomach out of a fish and ate the fish that was in the fish's stomach. So that was a really unusual sighting. You see the large rostrum there, very aggressive, coming out. All right. Oh. Probably senses the pressure wave from the vehicle and says, hey, what's going on? All right, we can go. What's that?
Does not want to be touched.
No.
That's far
Oh.
forward from the circle. East. Straight up there.
What else? Hey, Jared. There's a holothurian.
Alright.
Sea cucumber. That looks more like a Sanafa Brancadil on the right over there.
In your report arm, you have
I was watching out.
We're heading into a pilot change here in a minute.
Push poppinator. Might go with alright.
Yeah, it definitely
Yeah, it worked.
Functional.
Function.
Probably.
Whether or not the coral is wanted to eat remains to be determined. I don't know if you're going to see it on a macroscopic level anyways.
Well, I feel like these polyps pull in when they're disturbed, right? We touch them, they go
Yeah.
in. But I feel like that they will just be they're fishing, so sort of just hold the net out for a while. And you're not going to react to every piece that comes in. It will be more like once a day I'll just go in and see what I caught. I don't know. We don't really sit there long enough to watch, in-situ feeding.
No. Definitely
If
not.
that's happening.
Like I was up at 6, 7 yesterday, so it's been probably after 4 right now, so it's good.
Not seeing much of the sonar either. This is a big stretch of sand.
Someone on the chat asked how you become an ROV pilot.
They're picking the tripod. It's,
Like everyone makes their way here in a different way.
oh, that's no.
But I
Yeah,
don't-
each
I mean,
you
I
get a
can't
different,
answer
you
that
get a
though.
different story for each person out here. But they want to know how to become an RV pilot. that
No,
what you said?
so how does one become an ROV pilot? This looks like a fantastic job.
It is a fantastic job.
That's fine.
Some are engineers, some are military.
That's okay. I'm just gonna move your
Yeah.
waypoint up there too, so whenever
You
you
can become an electrical engineer.
slide and
And
come to an end, you can
Oh, we've got a
just
Dumbo.
head due east there.
is it? Oh, it is.
Perfect. Okay, thanks, fella.
Came out just to say hi to Jared.
Huh? Aw, yeah.
It's a little guy here, right?
Never gets old though, it really doesn't. There's really
This is a serrate octopus. It's gonna hunker down now because it felt us coming, but it's
So far, Tim.
there's a bit of a tripod fish there too together. Yeah, serrate Dumbo octopus. Very early on in the dive, we found a coral with a what looked to be an egg case on it. We sampled it. It'll be coming up when the RV comes up. It may belong to this guy. I don't know. This is you see the arms there wrapped. The tips are wrapped around itself. Typically, we identify these by looking at the rows of suckers, how they're oriented on the arm. Whether they're single row or double rows or alternating pattern back and forth. Yeah, feel free to. This has really small fins, those modified fins for swimming. So we call them Dumbos. This is a pretty small one. This is how we don't really see them swimming that much, right? Usually they're off the bottom 2 to 3 meters off the seafloor, and then they come down to the seafloor to feed. And typically Dumbos will feed, and other octopus feed on mollusks. They'll drill through a mussel shell or a clam shell and eat them out that way. Large eye. You can't see the siphon. If you saw the siphon, we could look at how fast the respiration rate is. We can actually count the breaths that they take, if you will.
yeah, the last one had its siphon out.
Right. So they really do protect their arms, especially the tips. And you see how they have them all curled up. There you can see the siphon closing down, opening. There's no doubt that we're throwing a little bit of a current its way, but probably not too much. Nothing else is moving. Field of view. What do you think, Jimbo?
I think it's great.
Yeah. Really large eyes.
I just don't know what he's thinking of us right
I know.
It's fascinating to know what's going through his head.
Sometimes we can see the eyes that are lights in the. Oh, see, there it is. A little bit of the lights in the top of the eye there from our vehicle, but we really didn't know about the life cycle of Dumbo octopuses in terms of we see them usually as adults, almost always as adults. So in 2002, I think it was, or 4, I discovered that the octopus lay eggs on corals, and they will put a single egg singly on a coral. They will cement it on the branches, and it'll be there for months. And then it will hatch a little baby Dumbo octopus with a massive amount of yolk. So it doesn't have to feed for a while, but it is fully sort of formed, if you will. It's ready to eat on its own. So they come out as little mini octopuses.
Has someone actually witnessed that?
I witnessed.
You
I think
did?
I'm the only one who's ever witnessed the birth of a Dumbo octopus in the deep sea. We had it in a bucket on the ship, and about 20 minutes after we got it on board, it was in a bucket, and it came out of its egg. The egg was sort of already open. I put it in a dish, and it swam around for 10 minutes or so before it expired. But we did an MRI on the octopus, and we looked at the structure of the interior parts of the juvenile, the recently born. and learned a lot about its physiology and, like I said, the yolk sac, and has a fully formed stomach, and it's ready to go.
So
So
do you reckon that's the same for the Dumbo octopuses in the Atlantic as it is for these ones?
no, I would not begin to assume that. That would be where I would start, though. But this wasn't in the Atlantic. It wasn't this octopus. This one had much larger fins. Anyway, all right. We got a good 10 seconds on that?
Yeah, I think so.
Just want to make sure we got it all. Okay. All right.
I think we're good to fly.
Thanks, everybody. It's like sharks and some fish and octopus. We always have to wait, so we seem to stop for them regardless. And in 2017, when we were down here in the Phoenix Islands protected area, we actually followed sharks, and they led us into coral beds. Remember that? They would lead us into places where corals
Yeah.
were really abundant. So we started letting the sharks show us the way. So I look forward to seeing that golf ball sized egg-like thing that we found on that metallic gorget. What's going on with that? That's not me.
So do you think
Weird.
out of an egg sac comes a lot, or one?
One. I'm sorry,
One.
a single one.
Okay.
It was weird because when we started following, I was looking for associates on this cruise, of course, and so we were getting branches that had these golf ball sized things, olive covered. They were sort of highly textured because like sediment was agglutinating onto the egg, and I'd open them up, and some of them were just sort of liquidy. Some of them there was a blob in them or two. And I can't. The more we looked at them, the more sampled them, the more they looked like squid. I could tell there was something in there that had some mass into it. It was kind of rubbery. It was white, and then lo and behold, one. Yeah, I never
so
saw.
they put a lot into their reproduction then if they only do one animal per egg.
One animal per egg, and it looks like it's months that it must sit on the coral. The coral that we've seen these eggs has grown through the egg. So,
Oh, interesting.
So there's enough growth there, right? So, yeah.
It's like, where is...
Yeah, I'm sorry. Is that scared? Maybe he still has time.
Still seeing nothing in the sonar in terms of hard grounds. We're getting a nice view of the habitat, the soft sediment habitat here. A few brittle stars. Not a lot of sea pens as we're looking here.
No, surprisingly, right? Usually at least you see a couple in these kind of places. Nothing else can grow.
Yeah.
And there's one down there. What's a xenophore?
Xenophyophores?
Yes, thank you.
Maybe we're seeing one or two of those.
Alrighty, so our last sample number was 474.
Yeah. Maybe I'm not sure what that fish was. I thought it was a Synapha branchidial for a second, and then it looked like a gulper for a second. A much longer stretch over sediment than I would have anticipated, but then looking at the map, it's a gentle slope. Ooh, there's a big crab coming. Let's have an image on this. We got a charismatic. Is it partially buried or not? No. It looks like it might have been. They do that sometimes. This might be like a Chaseon crab. I'm just going to zoom in a little bit and let Adam drive it in. Looks like a golden crab. It's going to recognize that we're here and it's going to get in a defensive posture. You got it, Adam. You see the tips of the legs are all buried in the sediment, hunkered down. sometimes they get underneath the sediment. You just see the half the top of their heads. Yeah.
However you want.
Nice job.
If you what? Oh, there's a battery, yeah. There's a plug here if you want. A charger, you want to get your charger for that.
Good place to stop. Fun to watch this guy walk away.
Old carapace.
It looks really old, doesn't it?
Yeah, sometimes they have barnacles. This one has?
Yeah.
Maybe. Yep.
Maybe some. Usually they're covered, right?
Yeah.
There can be. Sometimes we see them with their partner.
Yeah.
The male holds the female. Yeah. Good morning. Look at that. There's some barnacles there.
It was so bad.
Looks mad, you think?
What's that twitching? What's he doing? You know?
They have little antennal scales that,
Sensing.
yeah. It's really actually a broad carapace. It's actually vertically broad. It's a couple of, yeah, barnacles on top too. Anyway, yeah. So,
You want to go grab some lunch?
yeah, it's
Bear with a second, we're just getting into place the 12 to 4 watch. We're doing a quick watch change. just real quick.
All right, good afternoon, everyone from the RV Falkor. We are the 12 to 4 watch.
Which ones do you have on?
2 and 3.
My name is Steve Oskovich. I'm going to be the watch lead for this portion of the dive next few hours. And I'm joined by my colleague.
I'm Ana Meredith. I'm an undergraduate student from Brigham Young University of Hawaii, and this is my first time joining the expedition research.
We are diving on a different site than our previous days. We are on a shallow bank feature. We're working our way up the sides of this seamount from we started at about 1,400 meters today. And our goal for the end of the dive is to be somewhere up in the 300 meter range or shallower. So we'll see what we can do. Right now we're... Oh, that is I can't tell, it looks like some sort of stalk. Unidentified gelatinous stalk. I suspect it might be a solitary hydroid stalk. But we're trying to move rather quickly through some of the soft sediment, trying to get a handle on what the hard bottom communities look like here, specifically on these large carbonate blocks. This seamount had was originally most likely volcanic in origin. However, sometime in its geological history, it had developed a shallow water coral reef, which provided a carbonate cap and a relatively flat top, which could indicate it's a guyot or some sort of subsided reef. We are diving in an area that's closer to an area called Winslow Reef, which is in the Phoenix Islands protected area. We're almost due north of it, but still within U.S. waters right now. Pretty good current here coming from eastward. Sounds about right.
Yes, it's coming from east. Yeah.
From east, yeah.
There are some
Can I take a
corals
zoom on the
there
crab?
on the rock.
When you're ready. Sure.
Is the big one is a caliculture?
So we've got, let's see, let's zoom in and take a look. I'm not sure. But there's a few things we might want to look at here.
Oh, that's a crab.
Yeah, there is one crab down there. And we're going to take a look at that.
Here, man.
Thank you.
So there's a question over there. Can we get zooms on some of the fish, please?
Yeah, we'll try to get some fishes. We have some science goals here we're going to take a look at first, namely these associations of crabs and corals, and then as we see fish, we'll kind of get opportunistic zooms on them. I'm not sure. This crab looks like it's more so on the rock than on the coral, but there's probably maybe three different species of corals right in this frame on this overhang. All right. Can you shoot the lasers on that?
Yeah, no problem.
Thank you. Get some size perspective. Alright, and then there's another couple of associates up here we're going to take a look at. We don't need to get any closer. I think we could probably get enough here. Ophiuroids, yeah, with plexaurids. Looks like all the same species of plexaurid. Mostly in this area, Narella, plexaurids. However, over on this side, focus really far on this one. We have a species of Calyptrophora, some sort of lyrate branching. Looks pretty ragged. There's
Lighten up a little bit.
yeah, thanks. It is pretty distant, but it looks good. Just for identification and scanning for associates. Actually, can we move over to that one?
Yeah.
Take a closer look. And then when we sit down there, after we're done zooming, I'll take a scan here as well.
Okay.
.475 is our next number.
Mm-hmm.
Okay.
There you go.
Is there a little entropod?
I'm not sure. It could be something. Am I in auto right now?
You're in.
Okay, got it.
Kind of just teeter-tottering there.
Yeah. I'm trying to focus on something really small, so just bear with me.
Auto real quick.
Yeah.
Grab it. And back in manual.
Is it a jellyfish or anatomy?
Yeah, it looks like a jelly. That's a pretty high priority collection target there. That's all what I can see right now. Is there any way, do you think, of getting something like this on that?
Yeah. Let me set up a little better for it.
Sure. I don't know how sticky that jelly is. We tried to collect one once. Not really successful. It kind of dislodged just by slight movement. So I don't know what the best way to sample it is. I've never done one of these before. I think trying to slurp it up might be actually equally bad. Yeah, so if we can kind of grab it carefully, I think that might be the best thing.
Always saying just try to grab it.
Yeah,
Grab the coral.
grab it, snip
grab
it.
the coral.
Yeah.
And then just ease it over.
Yeah, I think so.
Were you going to set up to collect that corallivorous jelly? Thank you. Sorry, I left it. I was like, no. Hi, by the way.
It's a really tough position, so they're gonna have to reorient a little bit.
This is it. This is the corallivorous jelly we've been waiting for.
We tried to collect one a couple nights ago.
You saw one a couple nights ago?
It wasn't like this. It was not red. It was white, translucent, and as soon as we touched the colony, poof,
Yeah.
it went away.
I have one of them, and I sent it on to Mike. We're trying to.
Mike Ford?
Yeah, we're trying to describe it, and we need a couple more. So I'm gonna try to get it. Suction definitely works. On these things, they have to kind of put over the branch and.
Yeah. Can you hit the In-situ button? And when you do that, after that, the corallivory button. That's right.
Mm-hmm. This is corallivory. It's got this specially adapted feeding to suck right on a coral polyp, and it's been mostly on chrysogorgids, but it's really cool to see it on a primnoid.
Yep.
Oh, hey. How's it going?
Oh, hi.
Hi, Bill.
You
All
ready
right.
for
Yeah. Can we do
inspection?
a wide shot? Just hit one more time for the coral, also. All right. Yeah, I think we're ready to go. I think maybe here is enough, right? This branch, point, like this fork here.
Looks like
Yeah.
It's great. But
I just want to make sure everyone gets enough material.
But are you gonna try to suction the jelly first, or are you gonna try to just cut it and see if it'll come in?
I think the safest thing is to cut it. The suction could be equally bad. Do you have a preference? It's your call.
I think you're gonna more likely to get the jelly with suction, and then you can cut the coral afterwards, but it just takes more time. So if you wanna try it by cutting it, we can try it.
Yeah, I mean, I don't think it's not really one right way to do it. I think
I agree.
if it's that high of a priority, we can spend the time.
It's a priority.
Yeah, but I'm saying we can spend the time to slurp it and then get the coral. It shouldn't take that much longer.
Okay, I think that would be my preference, just so we don't lose the
You
jelly.
wanna try to slurp it?
Slurp the jelly.
Right. That should be good. Perfect. Sir?
Thank you.
Okay, so we'll slurp the jelly.
Slurp the jelly.
We'll give it separate numbers though, just in case.
Yeah.
So the next number is.
475.
That's right.
Is there gonna be a jellyfish?
Yeah.
Yeah.
Jellyfish. Jelly.
Can you just, Matt, I forgot you can't take screen grabs with that. I'm gonna take some screen grabs while the action's happening.
This is really exciting.
That flow? Before you go in, let me just clear this out a little bit. Okay. Nothing's in 2, right?
Nothing's in 2.
That would be.
Nothing's in 2.
Okay, cool.
Got it.
Awesome. Yes.
They got it?
Yeah, so it should be appearing somewhere in jar 2.
I haven't seen it yet. But I'm sure it's probably in there. It's just very little.
Yeah, we're gonna have a little small.
Great. I've been looking for this the whole
Let's
trip.
take a
I'm
look
so excited.
real quick before we do that coral.
I didn't see any.
It's really little. But I don't know.
Maybe stuck on the grate reading on top.
Yeah. Give it a little shake. Shake.
It's either in or it's not, but if you see another one, please grab it again. You can keep putting them in the same ones. Again and again and again if you see more.
You don't want to have different depth information for each
I
sample?
don't care that much. I care more about the animal. But yeah, it would be great, but if we run out of slurps, don't not take it, right? Just put it in the same one.
Yeah, we've only taken one slurp so far, so we have
Okay.
6 left. We should be in good shape.
Okay, can you click for a minute? Can I tell you something?
2 collections of the corallivorous jelly and the primnode it was feeding on.
Okay, Quiver 11 ready to be submitted. 2 collections of primnoid branches.
Very nicely done.
And a Niskin?
Yeah, let's do a Niskin also. So we're gonna do water sample for that one. That one you can check. We're gonna do a Niskin water sample here. You can just hit. Well, yeah, that's fine.
IV?
Just hit in-situ here, and then samples. So then we're gonna do FK 21-477. 2327.
Niskin 1?
Niskin. Let's double check to make sure that there was some concern that they had popped one. Can you just double check that one is open? One is open. Good. Great. Let's go for 1.
What your additional text?
Water sample near coral outcrop.
Near coral?
Coral outcrop. OUT.
Okay.
CROP. Got it. Thank you very much. Alright, so on that site there was corallium, primnode species.
Whoa, that's a big fish. There are some diversity.
Yeah, this is excellent because this is exactly what we can use the eDNA to tell us. We're seeing a lot of small stuff on underhangs and overhangs. eDNA can tell us, so with that water sample we're able to tell if there are other coral species in the area that we're not seeing, which will be informative. Yeah, it's a pretty good diversity here. Let me zoom on that purple one in the upper left corner. So all of these small ones are almost certainly Narella or Intiaca. This is where we saw them last, and they form these small bushy
There's a sea star
colonies.
under the pink.
The lasers on or off?
On the pink one?
Yeah, right there.
Lasers on, please. Yeah. I don't see a sea star.
Is it a sea star or just
I think it's just a blob, maybe a base of a former coral.
Hey, Steve.
Yeah.
Any interest in that Victor Gordon?
Not for the coral, but I'm happy to collect it for you. Yeah.
Is that just a lining or is that a white roof?
It looks pink to me.
Okay.
Yeah.
Okay. We're good then.
What do you find in? I can't see the.
We had a large white oath that was
Ah.
different, so it was all 50 gorgeous. So I'm always looking for it.
Okay. Well, we're gonna try and do a quick zoom, and then we can make a decision about collection.
This is
Forehead-
gonna be a
forehead
hard-
suction cup.
hard
Yeah.
collection.
I
It'll
know.
be all free flying.
I know.
So we can do it.
Well, let's get a closer look first, and then we'll confer back here. Let me know when I can go in.
That's a big shrimp.
Yeah, big shrimp on the underside of the rock there, right? Ready? Coming in? Got it. Hey Tim, are you getting what you needed with this?
Yep, I got it.
All right.
Thank you. That's it. That's
The if we want to collect, it's going to be a free flying collection. There's no place to sit down
There's
here.
a grab over there.
I'm okay.
Okay. All right. We can move on. Nice. Another crab.
Can you go one more touch soon? Just one.
One in.
Yeah, one in.
Yeah,
Perfect.
that's great.
A little bit too much.
That's it.
What are these purple corals up here?
I think those are small Victorgorgia. Yeah.
It's a nice spot.
Yeah. This so this overhang. These are Narella arentiaca. This is exactly how we found them in Pippa. Although there might be some other Narella mixed in as well. But they grow in these small bushy tufts. It's pretty common. Very nice. I think we should be seeing and entering the Scleractinian zone soon, where we're starting to see more Analepsemia and
We saw a couple.
yeah.
Did you see the crowd that we saw?
I did see. Yeah, there's been a number of them so far.
Yeah.
Yeah. Yeah. It's a. But this is a note about the terrain is that what we're looking at now looks to be carbonate blocks that have fallen off this feature at some point in the history, and it really does make excellent habitat for deep water corals, because a lot of these blocks are associated with these flows. You know, when water hits one side of it or hits the top surface flows over, it accelerates the currents, which result in better feeding for suspension feeding organisms like sponges and corals.
Go take a look at all that stuff.
Yeah, I mean, it's a lot of the same stuff, but I will zoom in on one of these ophiurids and this Chrysogorgia over here.
Oh, Chrysogorgia.
Let's do next time we see a little jelly on a coral.
Yeah.
Scrap it.
Yeah.
It's
It's like it's one of the hardest collections, because we did one the other night, right? It was much deeper, as soon as we touched the thing, the jelly swam away. It was very sensitive to touch and slurp. Yeah.
It's
And,
probably really, really soft.
It's probably really soft. Yeah.
See the second one up top?
The shrimp.
Is there one up top or not?
There's a shrimp up top.
Yeah, okay.
That you can see the claw. See the Bathypalmanoa?
Yeah.
Yep.
3 shrimp.
But this these Chrysogorgia have these saddlebags to them. Could indicate that they're in some sort of reproductive mode. Polyps have these bulges. Another Steroschema pale on a colony of Narella. Frame it up.
So what's Arantiaca about it? The base?
It's the branching pattern. It's the depth range. It's all the variables.
I meant sort of where's the orange?
That's the thing. Some of the colonies, when they were originally described, were very orange, and it's true. Some of them do have an orange color, but in a lot of the cases, I don't see much orange, particularly after it's been in ethanol. But when it's imaged on deck, sometimes you can tell better that way. But I've sent images to these for expert ID, and that's what they've come back as. And collections as well. All right, thanks. Ready to go. Yeah, it seems like that one species has a lot of different size classes. It doesn't have one particular size. It can grow small and bushy or large and branchy. Tilt up a tick. Solitary hydroids. Lots of these Narella, Victorgorgia, Hemicorallium. This is usually what I would call like the mixed zone, mixed assemblage zone. A few years ago, off of Jarvis Island on the EV Nautilus, we did a really excellent exploration of one of the longest ridges over a series of 5 kilometers. Yeah, massive crinoid. Actually,
A sponge.
Crinoid. Sponge. Yep. It's impressive. Yeah, these mixed assemblages, they have quite a bit of diversity, but even moving up just another 100 meters or so, it might transition again. There tends to be a pretty stark, very long arms on those crinoids. I haven't seen that very often. But we've sampled this plexaurid at least once on this expedition, just recently, in fact, on this dive. And I'm pretty sure we have another piece of it too from shallower dive in Howlander Baker. Could be. It looked like a bunch of tentacles. Could be anemone, but it's tough to tell. It's in a crevice like that. Ophiocanthids on these stalks. This one pretty much has no coral polyps, but it seems like the Ophiocanthids prefer this to the live stuff.
Big time ones here.
Yeah, they're massive. They're very extended too. It's kind of a crinoid body plan I haven't seen very often. Very long tentacles, or very long arms rather.
Sorry, these yellow planar ones.
sorry, I thought you were. What do you call it right now as your tentative idea? What do you think?
You know,
Seen it before, but-
I have to check. So this is the depth range for Flexoridae species 2, which is one of the most widely distributed corals with depth. I think, can we take just a look at that Swiftia colony over there?
So pretty.
I just want to confirm because, you know, the other day we were.
Tell the story.
No, the other day we were looking at some white and red Swiftia colonies a bit deeper, and this one kind of reminds me of the same kind of color, same colony shape and branching pattern, but in a white color morph that we sampled earlier.
Yep.
Steve, do you want this kid here?
I did. I just took one a few minutes ago. Yeah.
Awesome. It's so beautiful.
What is this called again?
Swiftia.
Swiftia, okay.
It's really delicate. It's really pretty.
Yeah.
When you get a chance, can I ask you to click off for a second just so I can tell you the plan?
So this one's a basket star here.
Yeah.
But this green plexaurid, we've sampled it a couple of times on this expedition so far. It has polyps that are very reminiscent of Anthomericia, but you know, it's one of those that you can't identify it without some closer examination.
Interesting.
Can you guys back off a little bit so we can get a more of a perspective of the kind of the feature we're moving up? Yeah, I know. Mesmerizing, right?
Crinoid mess with this basket star.
Yeah, that is it's an interesting association.
It is.
What's that?
Crinoid on the basket star.
And what's weird
You
is
see that?
every time that basket star moves, like crinoid freaks out because it's thing.
It makes you wonder why that. Well, you know, a lot of what these deep sea animals respond to is pressure and specifically pressure waves. So if the ROV is pushing a lot of water, for example, mind taking a closer look at that one?
Yeah.
Or just enough, like hovering close enough that I can quick zoom in and out?
And go ahead.
All right. So, Tim, I wanted to show you something here, which is the difference between this possible acanthogorgia in the background and then this plexaurid in front. So when you're doing your associate analysis, it seems like the acanthogorgia has a lot fewer associates from what I've seen so far in this expedition. But yeah, definitely interesting comparison here. Polyp density is similar, but the polyp shape is very different. All right, great, that's it.
Many basket star
Lots of basket stars, yeah.
and phosphor.
Tim, are you getting what you need from these? If we zoom in on all of them, or do we need to zoom in on all of them?
No, you don't have to.
Okay.
You know, a good representative zoom.
Okay.
One solid zoom does me a lot.
All right.
You know.
Great.
No, please don't do every one of this.
Yeah. All right. Let's move on. Let's make a little bit of way. Try and because we also want to get, you know, it's really hard to analyze the video when we're always zoomed in, so we want to get a perspective of kind of a wide shot like this moving upslope. But basically just some flying through the terrain, you know, like you guys do.
Okay. So just not so much stopping, just kind of overviews, and if you see something.
Yeah, yeah, we will. Yeah, I'll be able to point it out with enough notice here.
Okay.
Very nice cliff. This site reminds me a lot of Corallivory Reef, actually, with these small, stubby, and actually Nicomaru as well. These small, stubby Narella overhanging with the plexaurids mixed in. Molid crab there, walking around. Although I watched a lot of it, but I don't remember these Swiftia being a bit further south. Do you?
No.
Not these big,
No.
big branching colonies. It's very striking.
Have you seen Paragorgia?
Have not seen any. So it's mostly been Hemicorallium so far.
Yeah.
There's a chance we could see it. Although by my reckoning, a lot of the Paragorgia is a bit deeper, you know, plus or minus a few hundred meters in the 2,000 range. These bases are interesting, though. I'm wondering if these are possibly old scleractinian bases or maybe hemicorallian bases. They could also be primnoids, but they're quite weathered, which indicates that there's been a lot of something growing here over a very long period of time. But these the lip of these overhangs are typically fantastic because you get strong water flow on pretty much 3 sides of the colony, underneath, on top, and along the branch tips. Oh, there you go, Anopsemia. Yeah, this is the most interesting transition for me to look at, and one of the most striking ones we saw in our transect of Jarvis Island. We were seeing a lot of transition around the 1,000-meter isobath because of both topographic changes. You know, the rock and geomorphology was changing from, in some cases, more volcanic or carbonate blocky stuff into a little bit more cohesive terrain. And in the next couple hundred meters, I would expect to see more and more coral rubble if these stony corals are forming reefs at this in these deeper depths. Can I take a closer look at this one down here? Down on 6 o'clock. I know it's in a weird spot, so I'm just going to do a flyby zoom.
Go ahead and start zooming up. Stop there.
Yep.
Zoom. Oh.
Yep.
I'll just do some slow.
You want me to follow the coral or just?
No.
Okay.
I'm rotating the screen slow around it.
Yep.
Oh, you got some coral livery down the bottom right. You look at my sit cam? Let's see.
Oh yeah. Yeah, we'll take a look at that after.
Okay. Go wide real quick.
Sure.
Just want to see what this is doing. Just opens up. There it goes right there.
The one reminds me of Jason Isis. But it's
Yeah.
not. but it's,
The branching pattern, yeah.
you know,
It's a primnoid. It's probably Narella. I just wanted to confirm since it has a more variable branching pattern than we've seen so far.
yeah.
Most of what we've seen has been long, thin branches, which is pretty characteristic of Narella, but just it could just be where it's growing and how it's growing that's resulting in this.
Go for zoom.
Okay. So, yeah, Narella for sure. Yeah. All right. No, that's all right. I just want to do a quick in and out. We'll go take a look at that sea star now. How much corallivory do we have? Do we need how much corallivory have you gotten so far?
I don't think we've seen
Any collections of it?
any collections.
No.
I see stuff like this.
Go for zoom.
I think it's the same coral, but this is the first corallivory I've seen of the day, other than that jellyfish.
Yeah.
What's the likelihood of a sample on this? Are you still in a position that's reasonable on this wall? Or
Let's have a look.
So right now I'm like up against the wall on
Yeah.
the stick cam.
All right.
I can't go in anymore, but
Yeah, I know.
let's do this. How much on the back side? This one we do.
We are on a schedule, yeah, and I mean
we're gonna get it.
we can try, but we don't want it to take a crazy amount of time.
Roger. Can you science cam down on your camera?
Cam down.
Yeah. See what that forward, way. Keep going down all the way until you see the porch.
The top of the porch or the?
We're just trying to get the sea salt, right?
Um,
Or are you trying to get the flow as well?
we're gonna need both actually, yeah. As a part of the collection, but if we get, I mean, I guess one is better than none. So
Oh, let's see what we do.
if it comes down to one, that's what we might have to do. All right. So that one was a little bit precarious, kind of tucked in an overhang. So we're gonna try to bypass that collection, but we took some pretty good imagery of it, so we're hoping that it is good enough. But there's a massive amount of marine snow here, which could be fueling that productivity we're observing.
Some big old primates. So abundant too.
So, we are still moving up the side of this unnamed shallow bank in the US EEZ in the vicinity of Winslow Reef, just to the north of Winslow Reef proper. We started diving on this feature earlier in the morning at a depth of around 1,500 meters. We've come up about maybe 500 meters or so just in the past about 6 hours of being on bottom. We have been doing a lot of imaging and sampling along this site. It is one site that we think is the third shallowest feature within the US EEZ around Howland and Baker Islands, after Howland and Baker Island. So that would make it a pretty remarkable site to do some further characterization at. Take a look at this fish. Just a flyby. Can you hit the fish button, please?
Sure, are you ready? Do you have any?
No comments. Just hit the fish button. All right. Thanks. So it seems to have gone a bit more sparse in this area. I think a lot of what we were seeing in the high density communities were largely in these grooves and overhangs. On the vertical surfaces, there's quite a bit. This is a pretty nice carbonate mount here. Yeah. Be careful as you get up in here. It can get pretty craggy. So this does look like more of the carbonate cap. This is definitely not volcanic material, not basaltic. It's most likely carbonate that's been formed over possibly millions of years. At this depth, most certainly millions of years. And there's quite a bit of topography here. We're sitting on a wall now that's about 20 meters tall. 20, 30. Pretty good sized. Starting to see some stony corals as well. But similar cast of characters right now. Plexaurids, Victororgia, a few species of Narella, Chrysogorgids, a bunch of these Oreo fishes, Oreodories. Definitely a lot more of these.
Oh, there's a crab on that.
Yeah, chirostylid crabs. They're actually more common on. Well, maybe not more common, but we're seeing them a lot more on the rocky surface of this feature rather than on the corals themselves, which suggests that they're getting what they need, feeding wise, not having to climb up on the corals. Maybe it could be some sort of precarious perch. But it seems that they're getting what they need on the rock. But we've seen them both on the rock and the coral, which is a noteworthy observation. Curious why they're doing that in this area. Crinoids have been quite abundant, though. Yeah, good view. Science camera. Anelopsami, stony corals, a bunch of these Narella. Bamboo coral right there.
Ooh.
quite long.
That's a very long. Whoops.
Yeah, these are very
Oh my
nice.
gosh.
There's Narilla and Kiltrophora down there as well.
Jesus.
Oh my goodness.
It's a good size. Couple meters.
Yeah.
Can I look along the wall to the left side? Tilt out. Yeah, this way. And try and get the background shot too. Now, how would you feel about popping a Niskin in this area as well?
I'll get back with this one forward.
Can you hit in situ sample
Sure.
or in situ button? Yep. And hit the sample button. And then this is going to be sample number. Yeah, we're going to look at that later. 478. At 1078 meters. For the description, so it's going to be Niskin 2. And for the description is coral wall. Just write coral wall is good enough. Yeah, that's some pretty spectacular stuff here. It's some of these carbonate features are some of my favorite topography to fly through. Got it? Good. Okay, 21-478. Coral. Octocoral. Random species. Okay. Yeah, we'll take a look at that big fan over there. A lot of these squat lobsters, they're very common both on the rock surface and on the coral. I'm a little confused about this one coming up here. It's one of two different genera. And we'll be able to get a better idea if we get a tight zoom on it. It's a great white balance target though.
Yeah, wonderful. You want to do that?
You know, it looks okay to me, actually. Maybe on the maybe yeah. Let's do a white balance just to periodically check that box. All right, a little bit better. Good. So I'd like to do a tight zoom on the polyps as much as possible. You know, I know that you're flying. Yeah, at least 1, 2, 3, 4 crabs in that one colony.
Start zooming in slow. I'll get you a little closer.
Sure.
Zoom in.
Say when. Just gonna try and focus closer. Got it. Yep. Can you come out towards the branch tips a little bit more?
Yeah.
There's another colony down below that has a little bit of a lip where you might be able to set down. Is that something that's an option? I can't quite tell from here. It's a little bouncy, and there are some fine characteristics. You know, it's sufficiently different that we might want a collection of this, but you see how there are many of these colonies?
Yeah.
Kind of here. Is any one of these look reasonable?
Well, let's see.
Because I see I know it kind of puts you in an overhang here, which is pretty awful. So if there's something that is easier in the next little ways, you can get a toe hold on, that might be the best.
Same with myself between this and here.
Yeah, yeah. Yeah, I think it's the same.
Fish. Is that a sponge on it? Like on the
That's the base of the colony,
Oh.
where it's attached.
Oh.
Yeah, that's really strong. This thing has been here for a very long time. It's over 1 meter tall.
What's that?
Yeah.
Single company.
You know, I do have a good idea on this. Do you want any of the associates here?
I would love one of those crabs.
Go ahead and zoom in on me.
Sure.
That's-
The one on top. Yeah.
Yeah.
The problem is it's tough to sit down here
I
because
know.
yeah.
I can see that. It'd be
If I can get a good zoom on this, I don't need a collection because I've got a really good photo reference here for Calyptrophora clarkii.
Take it out of manual for a second.
Sure. There you go. Thank you. Yeah. Okay. I'm satisfied with this shot. Yeah. This is almost certainly clarkii. Basal spine and everything. Very nice.
Do a zoom on that crab, maybe I can
Up to the top, in the
do
middle.
something. It's just the, I don't know if it was Iaspis or something different than I'm used to.
Yeah. Did you see how many were on the rock compared to on the corals? It was pretty
These?
impressive. Yeah.
Yeah. We've seen several now, right?
Yeah.
Just on the
Yeah.
rock. If this was something different, the same as they have the,
That's full zoom there.
right. Spines, all the lateral spines are there, so. I think I know who this is too. Don't know if they're all the same or not, if this, you know,
Mhm.
it'd be, yeah.
That Ophiocanthid though is pretty mean looking. Yeah.
Yeah.
Dark.
What a place to hang out, right?
It's a fantastic place to hang out.
Yeah.
Yeah. All right.
Yeah.
Very nice. Yeah. I mean, it's almost a 1-for-1 here.
Do you want to collect it?
No, I don't want to collect it. Thank you. This is one of the most abundant primnoids that was collected during our 2017 expeditions, and
Yeah.
they all look slightly different depending on how old they are, how well the colony's grown, the shape of the colony. So for the most part, there's a lot of variability in how the colony looks, but morphologically the polyps, if we can get an image of that, we can almost get to species without needing a collection. In this case, we have a lot of collections to compare to.
Moving on.
We are moving up. Just see more interesting animals. It's weird that mostly of the corals here, like the crinoids, love to stay on them.
Yeah, the crinoids. So the crinoids, like, they have these projections on their arms, right?
Mm-hmm.
They're somewhat sticky, such that they can grab on to all these particles in the water.
Mm-hmm.
And what they're trying to do is get up into the flow where the flow is faster. So the closer you go to the rock, the slower the water is going to be moving because there's friction between the water and the rock. So if you can get up off that boundary layer, it's called, up off into the current as much as possible, you're going to have a better time feeding. And you're going to be able to get more nutritious food particles. Now this is one of those zones where, you know, the purple clavularia. Hold on. Can you stop and look at the purple? It's the first one I've seen.
That looks doable too.
Yeah, that doesn't look like Victor Gorgia settlement. That looks like Trichothelia or whatever we're calling it now, clavularia.
Wow.
Yeah, no, that's still an Stoloniferan.
Yeah.
Yep.
All along.
That's fantastic.
It is fantastic.
This is one of my top wanted list species too. Can I go in?
Maybe. Yep. Think so?
I just want to know if you're
I don't know
stable.
what I'm on.
You don't know what you're on?
No. No idea, but it ain't staying still.
Hopefully the Seamount.
She's still.
Yeah.
Wow.
Yeah. So, yeah.
It's gonna be a suction, right? Or you break down that ball.
That's what I'm thinking. Do you think you can grab a chunk of that and just break it off?
Definitely give it a try.
Yeah.
Are you chucking the rock?
Yeah, let me, can you hit the In-situ button? We're gonna image it first. And then, we're gonna do a lasers on that portion. Alright, hit it. Hit the lasers one more time. Yeah, so I'm thinking, you know, obviously we don't want to mangle all the coral, but if you can just grab a chunk of that and break it off with a bunch of polyps on there, all we need is maybe a dozen or so.
Okay.
That would be great. Yeah. Something quiver sized too, if possible. I know a lot of conditions, but you guys are pros.
That's what I was thinking.
It's definitely on the farm. Zoom up and see what happens. Curious. Yeah,
Do you have any idea what's coral is that?
Yeah, it's so it used to be called clavularia, but I'm not sure if this applies to every clavularia, but the purple ones have been, it seems like they've been placed in a new genus called Trichothelia more recently, within the past 5 years or so. But I'm not sure, those were Atlantic stoloniferans, and I'm not sure about the specific specific species if they are even follow that same rule. It's harder than it looks, though. This rock. I'll do the sample storage on this one. I want to write some extra notes. 479. So what we're going to try and do is we're going to try and break off a piece of the rock with a bunch of this coral on it, since it's pretty much attached, encrusting on the rock and connected by this fleshy stolon tissue. One other way to get it would be to do something like a suction, but that generally mangles them pretty well, and you know, it's actually worked out very well. So if the rock breaking technique doesn't work, then this is the next best thing.
Perfect.
Yeah, I think that's great. Yeah,
About
excellent.
a dozen or so?
Yeah, that's great. Yep, perfect.
Nice and slow.
So, for the folks who are wondering about the coral feeder, yeah, we tried it a couple of times, and the results were pretty mixed. There wasn't a lot of response. So we're going to try and maybe work on another trial a little bit later on in the dive when we get much shallower. So this is going into quiver 1, 2, 12.
Is it 7-9?
Yeah. I got it right here already.
Oh, okay.
I said I was going to do this one. Chart 12. Great sample. Thank you guys. But you should be able to rewind if you want to rewatch that coral feeding segment again. If you're on YouTube, you should be able to rewind and watch some of the experiment. All right, so one of the coral feeders was a little damaged, so we're gonna try and stow it so we can use it for later. But stay tuned, we'll probably be breaking that out again sometime later in the dive.
Alright, you ready?
Ready to go.
Go.
Yeah, thanks for that. It was a great collection, the coral. It's always useful when you can see the entire stolon intact.
Yeah.
Yeah, it makes a difference for imaging sometimes. Wow, that's a big anemone. It's like 20, 30 centimeters. It's like a dinner plate.
Never played an enemy.
I hit a lot of inputting a lot of corals. Do I need to keep on doing it?
Not for every single individual, but
Just
yeah,
I just put a
once-
bunch of them.
Yeah,
Okay.
yeah, just once in a while. Yeah. Yeah, so most of these are plexaurid, plexaurids, and Narella, Calotropha, various species. So there's a bit of a diversity here. Crinoid. This one, crinoid best.
Is that a fish?
There's a crab there. Yep. That's some pretty fresh carbonate, actually. Can you, hold on, can you?
Yep.
Down and to the left.
Down to
Yeah. And we'll kind of work our way back up. But
Okay,
this
no
here
problem.
is very interesting to me. This is a really fresh break right here.
It is.
Yeah, so this is really, really recent. And you can tell, so there's a crusty layer, right, on top. But this is a really fresh carbonate break. And you can see the limestone. You follow this down as much as you can a little bit.
Definitely can.
Yeah. We'll kind of work our way back up in the same direction. I just want to get a perspective of what kind of things are attaching here. Yeah, I think that's good enough right there.
I think I see what
Yeah.
it was.
Yeah, I think that's this is a pretty big block to have cracked or something else cracked off of it.
Something big cracked off.
Something big cracked off of this one.
This doesn't come to the right as well.
Yeah, wow. Huh. Can you come off a little bit and we'll do like a
Yeah.
wider shot? Just to get a perspective of how big this break actually was.
That's a break too.
Yeah. Yeah, this looks like it was relatively recent. I mean, the crust and discoloration often takes a very long time to start to appear, but when you start to get really fresh breaks like this could have been maybe even years. Relatively recent.
Nothing's formed on it.
Yeah. Yeah, there's no incrustation, there's nothing. This actually reminds me a lot of some things we were seeing in the Mona Passage in the Caribbean, which is a very tectonically active canyon, and we would often find that there was nothing living on the walls, despite it being good current, great conditions.
Sounds more.
Sure. Yep.
See if there's anything that spots up.
Yeah, this is excellent. I think on the way back up, we might want to zoom into the limestone to see if there's any interesting shapes and morphologies, maybe old coral that might be in the exposed surface.
There's another question. Is the pH in the water higher in the area of the carbonate cap?
That's a really good question. That's not something we're equipped to answer on this expedition. However, in our 2017 cruise, one of the analyses we were doing was to look at pH and carbonate saturation in the vicinity, or rather in the water column more broadly. And we did, but we did not specifically look at pH levels in the vicinity of the carbonate caps versus non-carbonate areas. And I would suspect it might be a different situation.
There's not that much sediment on there either.
Yeah, it's really not. I mean,
Just a little bit.
that's why I'm thinking it could be years or maybe decades fresh, which kind of sounds bizarre, but sedimentation being how slow it is, it could just be, it's not necessarily just sedimentation from the water column, but sedimentation from just landslides and things.
It's a big break. That's all.
Yeah. Something big fell. Yeah.
We'll go to the easy spot when we get here.
Yeah. Great. It looks murky too, or is that the camera?
That might be the camera we got the flood on.
I
And
got
I
it.
just tried
Yeah,
to
a little foggy.
see if there was any sediment. See how much sediment was on it.
Yep. Yeah, so I think I'd like to zoom somewhere up here near where the edge is, where the fresh break is, just to see. Yeah, right here. Perfect.
Okay, let me I'll sit down.
Just to see if we can see any things like maybe an old fossil coral head or something. This also gives us some perspective of how thick the crusts are here without having to actually collect a rock.
There you go.
Thanks. So working from the surface of the sediment, right? Look at this. That's all old coral right there. So, let me start, yeah. There's some more. So I'm gonna just, fossilized, exactly. Working our way down. Yep.
Collect, please.
Better or worse for you?
It's
It's a little bit reflective. So.
It's a little bit, yeah, turn it off maybe.
Okay.
Yeah.
Poke the carbonate. I want to see how easy it is to scratch.
Just a second, let me do some camera work here and then I'll read the messages. Just kind of going down little by little. I'm going to get some lasers on this so we can get an idea of how deep some of these things are too. That looks like coral there as well. But yeah, you can get a real good idea of kind of what the reef matrix looked like at one point in history, or many points in history. Layer upon layer upon layer. And it starts to get a little bit less clear as deeper you go. Yeah, and then this is kind of the bottom of it where the base of the crack is. Okay, let me put some lasers on it so we can see how big this kind of overhang is. Maybe 1.5 meters, something like that. Alright, let's see. Can we scratch it?
Yes, you can.
This might give us an idea about, if this is really soft stuff, it may be more or less difficult for corals to attach onto. We're not seeing a lot of any settlement here. So it may just be friable, too friable for corals to be able to attach to. But like we said, if it happened really recently, there just might not be enough settlement, larvae, propagules getting at the area to really affect any of this. Yeah, really soft and brittle. Yeah, really nice. I think that's perfect. Don't even need to go anymore.
Good.
Yeah, that's great. Yeah, so really soft and friable stuff here. Yeah, I mean, I've dove on a lot of these types of features, and I've never once seen something so fresh as this break in a bank. In canyons, yes, but never in this type of environment. So this is really neat. I'm just going to zoom one more time down here at the base, and then we can be off. Kind of follow it up a little bit more until it disappears. Yeah, but you can start to see the outline of those corals here, where it has these grooves. Yeah. Yeah. Yeah, it is really cool.
You think that might have been a sponge right there in that little?
Could be.
The two little holes there.
Yeah, could be. Could be some sort of branching coral too. A lot of the times the inside of the coral will hollow out. Since sometimes it's usually less dense than the stuff on the outside. Yeah. All right, this is really cool. Let's follow this up and see what we can see on the way up. So this, when we talk about carbonate cap, this is exactly what we're talking about, and nothing illustrates this better than what we just looked at.
That looks like something fell. This fell from somewhere.
Yeah. Yeah. All of this stuff fell from somewhere. I mean, a lot of the it's the corals grow on top of this carbonate cap, right? The coral growth is typically at the shallowest point, and they'll just keep growing and growing and growing, consistent with sea level state at the time. But the calcium carbonate they produce, if they keep building up and up, it may become unstable over time because it's not as cohesive as basalt, the volcanic stuff that makes up the antecedent topography, the topography, the structure that gives a lot of the seamount the rise from the abyssal plain. Still a fair amount of corals here, but you can see definitely nothing on those fresh faces, but plenty of corals on this this crustier material. Ctenophore.
Is this a cucumber or tourniquet?
This is a ctenophore. CT, that one. Yeah. Do you want me to focus, or you guys? I have it in auto. Whichever. I know these things are hard to focus on.
polychaete or something. Scared of one of these holes.
Huh. Big? Big one?
It was decent size. There it goes.
Oh.
Might be a scale one. Open out a little bit.
Oh yeah, in the back there?
Yeah.
Yeah. Yeah, wow. It's fast mover. It's one of those hamalid crabs without its little hat. Oh, is that a, hold on, what's that? Is that an ophiuroid swimming in the water column?
Yeah.
Interesting. I've seen this a few times before, but it's always really impressive that they're able to maintain. I mean, it's really effective.
It is.
I don't think it had that much surface area to be pushing.
Yeah. Oh, this one in the back we're going to look at. Yeah.
It's swimming so hard.
I think this might be a big plexaurid fan, but I just wanted to double check since it's one of the larger ones.
A little bit chilly to hold.
Oh, a sponge.
A little bit chilly to hold.
Sponge, yep. Sponge in the background. There's a glass sponge.
Look at the jack.
Oh, there's a snail on that.
Yeah, there was something on it. I wanted to take a look at it.
Crinoid snails and crab.
Oh, yeah. It's a anemone. It's all sucked in. They tend to grow around colonies like that. There's looks like a hamalid crab in the background, maybe. It's tough to tell from this side. A bunch of these snails, though. Small ophiuroids in this one. So this is a different plexaurid that we've seen, but it's one that I've sampled before. In fact, we have a lot of material from this, from the Phoenix Islands, and it's actually might represent a bit of a change in community composition, since the distribution for this species of plexaurid is quite shallow. Can we take a snip of this? Let me see if I can find something that is multi-purpose for all of us. Maybe something fresher than that. Let's see what's going on down here. Barnacles, hermit crab, ophiuroids. All right. Well, I think there's enough ophiuroids on this one that this piece right here might be great.
Okay.
Just
Stick it out.
yeah, that'll fit in a quiver, right?
Mm-hmm.
Yeah. Okay.
Do you need a shot for that?
Yeah. Sorry. Let me get an image.
Okay.
Yep. All right. Since the polyps are closed, this is a great time to do an image. All right. Go for In-situ. One more.
Yeah.
Awesome. Okay, we are ready for collection. And maybe one more, kind of just like this.
No?
Yes. Yeah.
Okay.
Yeah, awesome.
Is it 480?
480, yes.
Biology. What's your additional text?
So it's gonna be. I'll write it out for you and you just copy it. just Plexauridae here. And then just write this comment there, C. Plexaurids, species 0018 from FK 171005.
Zero
18, yeah.
FK 21. FK 17
17, yeah. 1005.
1005.
Yes. And that's quiver number 10.
I'll just rub this one in.
That's fine. Yeah, that's fine as is. Yep, looks great. So if this is similar to a collection we made in 2017, this suggests this is the genus Paramaricia, which is quite common in parts of the Atlantic, but less common in the Pacific in certain areas. So it's a very noteworthy collection. We sampled something very similar at a much deeper depth in 2017. However, with a lot of these plexaurids, very fine differences, very small differences can only be noticeable when they're sampled and looked at under the microscope. So this one will be a very interesting collection to re-image. Interestingly enough, for Tim, if you're watching or listening to this in replay, this last time we saw this in Pippa, a lot of these Paramaricia were covered with Asteroschema, but I haven't noticed any Asteroschema on this one. It's mostly been Ophiocanthids, but take a look at that if you have a chance. Great, awesome, thank you. Keep moving. Filled up, Tig?
Yep.
Okay. Great, right there.
Oh.
Sea cucumber, maybe? Let's see. Siphonophore could be.
Siphon.
I don't know. Get a better look at it if we can. No, maybe it's a worm? Siphonophore. I don't know. I'm gonna go with Siphonophore since it's just a very small one.
Is that a sponge? Oh yeah, glass sponge.
Yep. That's Regadrella. Almost certainly it's a Euplectellate.
Mm-hmm.
It's very similar to in the same genus to the one we collected last night. that large cone-shaped one that was being predated upon by 2 sea stars.
Mm-hmm.
But there's a little bit of a difference now. We've kind of moved out. Oh, never mind. We're still in it, just different overhangs.
Surprise.
Yeah. Well, we haven't moved too much in the vertical, so it makes sense. We see a lot of the same species, but the terrain has definitely gotten a lot steeper in certain places. So we've got Calyptrophora clarki, a couple species of Narella here, mushroom corals, heteropolypus, as well as a few plexaurids.
Is this a jellyfish?
Yep, that one right there. Yep, coming on the camera now.
So cute. Jellyfish.
You can put it in auto if you want to focus on that. It's tough to get at it when it's moving.
Focus on here really quick. All of them are
Good.
They're always in focus.
Always on focus. That's my camera split.
Yeah, this is spectacular. I mean, I really enjoyed the deep dive we did the other day, but just these overhangs are just spectacular with coral coverage.
Yeah, it's cool.
It's not even really that dense. We've seen some pretty, what we're calling the wall of awesomeness from the other day, that we were sampling those stony corals on. That's kind of how as good as it can get sometimes. However, what would be better is if we were able to find a stony coral reef that's flat and propagating.
It looks like crabs loves to stay in this area.
Yeah, they definitely like these overhangs. Yeah. Hold on, slow up a sec. Can we zoom in on that thing? White blob. Ready?
Yeah.
Sea cucumber? Wait,
I think it's a
looks like
nudibranch actually. Hold on.
coral.
Nudibranch. Yeah. It's a type of mollusk. They're usually predators down here. They like to eat things. But yeah, it looks like a nudibranch. But the first thing I noticed when I saw this was like that's got to be a soft coral, since it almost looks like it has polyps, right? But it does look like nudibranch structures. That's really cool.
What is
I've never
that?
seen one of this. It's a nudibranch. It's I don't think we have a button for it even. I think it's a. I'll just type it in here. NUDIBRANCH. BRANCH. Yeah. Nice bamboo coral.
Nice, the bamboo coral.
Nice bamboo coral. Oftentimes, haven't seen any yet, but these bamboo corals, when they do fall, grazers will come along, eat off all the polyps, and what's left is these large bamboo stalks. They start to dissolve away after a while. Wow, that's a big crab. Is that 2 crabs or 1 crab? Lower left. Lower right.
Lower right?
Yeah. Do you see these crabs down there? Looks like there were 2 crabs on top of each other. It's a big crab. It's like 30 centimeters. Tip to tip.
Oh, looks like it.
Yeah, I think that might be a mating pair of crabs. Pardon the interruption.
What are they doing?
Well, we'll leave them be. I've never actually seen one that's dark purple like that. That's actually really interesting. So for the, if you were with us yesterday, we sampled a curious looking bit of tissue. Yep, Anelopsamnia. We sampled a curious bit of tissue, or what looked like tissue, from a rocky surface. We were calling it rock skin for a long time. And some folks were interested in what it was when we got up to the surface. Oh, yep, a bunch of octocorals here. Really high density. This is nice. So the rock skin. Can I, yeah. So that looks more like madrepora. Can I zoom in on the underside of that rock?
Yeah, get it set up for you.
Sure.
So the current seems to be going from north to south.
Yeah. North to south. Okay, that seems right, yeah. Any stronger or about the same?
It's enough to give me a little nudge.
Yeah.
Not too crazy.
Yeah. Ready? Okay, going in.
Yep.
I'll finish the story about the rock skin in a second.
That's
Just
like a
some
coral.
imaging here. Yeah, this is stony coral here. So do you remember that when we were at that large wall site the other day where it was like covered just like top to bottom? This is one of the components of that, madrepora. Really interesting though that there's not much buildup here. It's growing kind of downwards, and a lot of the older reefy material is up top. But there's a lot of
So many.
chirostylid crabs in the matrix here. Are you the Grim Reaper now?
Do it.
All right. All right, we can go for a collection.
Okay, I'm gonna just pop up and pop those.
Yep. Yeah, that's fine.
Is it 481?
481. Yes.
What additional text you want to input?
It's going to be Madrepora, Madre Pora. Yeah, that's fine.
Oh, Metropora.
You gonna use Joe?
I'm sorry.
Really needs to be sound, struck out.
Might be able to, might be able to get in. Let's see.
I'm kind of surprised at this particular little outcrop and how little debris there is. I would expect to see, if this was in here for a very long time, there'd be like a rubble trail just extending downslope. So the rock skin, okay. It was in the middle of the story that the rock skin might pick it up after the collection here. Sorry to make this a bit more of a cliffhanger.
So the ridge comes out like right here, and then there's lateral below it. Go like that ridge, goes lateral. For going. Just like over. Nice and smooth. There's nothing on top of this. It's all on the side. We'll get the stony floor, lot line, just kind of legislative on the left side of it, it's a wall. Right side's good enough.
Yeah, she says she'll finish up.
Metropora chunking at higher rocks if you need to. So try not to get a huge piece, we don't need it to be rich.
Yeah. Yeah, we can biobox it too if you do it.
Yeah, no, I just,
grab a chunk. That might work. Yeah. Can I zoom in on it to see what we got down there? Yeah, I mean, I think that's plenty of polyps. That should be plenty of polyps. If it's not, let me know. Alexis, and we can go from there. But I think that should be enough. We can put that in a quiver. We don't need a lot, but if we did get a lot, we wouldn't want to waste it. We'd put it in the firebox. Yeah, if we can get, something like half or even 2/3 of that would be ideal. Nine's open. It's okay if it breaks going into the quiver, you can just jam it in there.
Do you want us to grab the other little piece of
No, that's perfect amount. That's great. Thank you. All right, so it's going in quivers 9. Yes, you can submit it. As soon as it goes in, just make sure it has the right number and hit submit. How many rock collections? So far we've got 2.
Got it.
Only 2. We have we've filled up our expected allotment of rocks for the expedition, but we're going to try and just take a few more samples over the next few dives, maybe opportunistically. But for the most part, we've accomplished our rock collecting objectives. And then on the way back, we'll see what kind of opportunities we have for rock collection. But so far, it's been a great success. Yeah, we're all set. So one of the things that I've been noting in our explorations of the Pacific has been the occurrences at which a lot of these coral colonies occur, mostly in Elapsamia, but also Madrepora, and it does seem that the 600 to 1,000 meter depth range is ideal for these kind of reefy growing species, and that seems to hold true for here. We do find Elapsamia and sometimes even Madrepora deeper, but they definitely don't form these large structures. They're actually just singular colonies. So it's really interesting to note that that pattern seems to be holding true here. Fantastic. Hermit crab. We're starting to get more of these blocks. Yeah. Did either of you see that fresh carbonate, how it broke off?
I saw that
Before you
on
came
the
on
camera,
watch?
yeah.
That's pretty wild stuff. It's like it could have happened yesterday or years ago, who knows. It's incredible. There we go. There's some nice Madrepora too. This is the same stuff we just collected. Very large colony. We were noticing some of these at Howland Island, I think, was the first time we saw them, and we were pretty impressed with that. Oh, there's some fossil reef in the rock too up here. It's a fossil coral head. Yeah, so this is kind of mirroring what we saw at Howland and Baker Island, which is expected. We're kind of diving on a similar feature, even though this one doesn't break the surface. It still can have a lot of the same deep sea biology. Can it tilt up a little bit? That's okay. Tilt up a little bit? I got it. I just wanted to ask if it's okay. Nice. Okay. Yeah. I'm really just trying to see what's ahead, so we can see if give enough notice if there's something to sample. Some old bamboo coral skeleton. Nothing live there, though. Mostly dead. It's like sword sea fans. They're definitely dominating this habitat. Yeah, they're everywhere. Although it should shift slightly in favor of the scleractinians, the shallower we go. To go back to the situation with the rock skin, if you were with us yesterday, we sampled some what looked like leathery, almost some sort of biofilm looking thing on a rock that was quite cohesive. We actually still don't know what it is. we took a few strips of it and preserved it, but the best guess seems to be some sort of benthic foraminiferan right now. But stay tuned for that. Could most likely will change, unlike something most of us have really ever seen. So it could be some sort of animal mucus or some sort of benthic foramen. Current's getting stronger.
Just try not to dust everything out.
Sure, yeah.
See what's going on.
It seems like there's a lot more particles, marine snow in this area as well. Sure, yep, go ahead.
Just peek over at this one every now and again. I'll move up and down when I see something.
Okay.
on top.
Yeah, it seems to be right along the ledge here. There's not much on top.
Is that a coral rubble?
Coral rubble. Yep, there are few small pieces right there on top of the rock. So I bet if you followed that up, for example, you might find some of that coral that's producing the rubble. Yeah, there's little bits of coral madrepora here along exactly like we saw it the other day on that sheer wall
Oh my
on the
gosh.
backside.
So many. But pretty much the dominant coral is the best, right?
Yeah, they tend to be. They're mostly shallow water, so most of the species within the group are shallower water, but they definitely have been very successful invaders into the deep sea.
What's that?
The fish, that one?
The other one.
The other thin fish?
Yeah.
Not sure.
The hind that stick up to the rock.
So far, not much evidence of predation other than a couple of instances. We saw one sea star predation instance as well as another jellyfish on a coral predation instance from jar 2. We were never able to really identify if the jellyfish was actually in jar 2, so we're interested in getting that back on shore, on deck, to take a look at that further. Lots of these Oreos, this is their depth range.
Viticulture. Is that a bamboo coral?
It's tough to tell. There's no nodes on it, so I would say probably not. It's probably a primnoid base or a precious coral base. But a group of Anthomastus there.
Yeah, mushroom.
Habit of saying mushroom corals, Anthomastus. They're now called Heteropolypus, most of the species. Yeah, so that base we saw is probably actually one of those, the hemicorallium. the pink coral. There's some Analopsemia, smaller one. It seems to be that the Analopsemia don't really always. It's really confusing because what we were seeing in 2017 in the Phoenix Islands was oftentimes Analopsemia were the reef producers, and they formed the largest colonies. But it seems north of the equator, Madrepora, from what I've seen, is the biggest producer of rubble and skeleton and debris. I wonder if that has anything to do with oceanographic differences across the equator, but I wouldn't expect it to be too much differences. Metallogorgia? Yeah, could we take a quick look at it in the associate specifically? We haven't seen one in a while, so it would be good to compare with some of the 500-meter collections that we made. We did end up collecting a Metallogorgia earlier on in the dive with ophiuroids and some sort of mysterious egg mass. Oh, Halasar, Aldrivandia, almost certainly. Oh, hey, another egg mass. So we've got, oh, this is almost exactly like the collection we made, a bit deeper. So we've got some sort of ophiuroid stereoscomatid with some shrimp, maybe Bathypalmonella, and then in the background there's also another egg case. This one doesn't look ruptured, so it could be recently settled, recently placed. But this looks like what we have been calling the shallow Metallogorgia morph. Do you think we could rotate around to the backside to get a shot at the egg? Or even just a little bit more behind it? If it's going to kick around like that, I guess it's not too big of a deal. Yep. We had collected one just like this, strangely a bit deeper, and I just wanted to check if that egg was also not hatched yet. It'd be kind of a neat observation to get to. So with the Metallogorgia, there's almost certainly 2, maybe 3 species in this area. It's almost definitely a new species, a different species than the deep water Metallogorgia, which is Metallogorgia melanotrichos. But we've been noticing. Oh, that's okay. Can you reset the focus when you get a chance? Kind of got the focus is all lost. Yeah, sure. Yeah. Still a tough call. It looks whole, looks intact. which is pretty interesting to see 2 of these on a dive. We thought we would see more on this expedition so far, but it's been pretty sparse. So we've been calling this a shallow water species of Metallogorgia with good reason. There are some small differences in both the branching pattern and in polyp shape and size. So that's what we're going to call it for now. We're working on it, seeing if it may be a new species and hoping to describe it. Whoa, big fish. All right, that's okay. We've seen that fish before. We can move on. Push the fish button if you see a fish, though, please. Corallivory, yep. That's very similar to when we collected a few dives ago when we were diving shallow last. This kind of very narrow bottle brush. We collected a bunch of associates with it as well. I think 2 or 3 crabs were in the last one. Calyptrophora, another one. Surprised how patchy the madrepora is here, but there's definitely more of a mixture of madrepora and Anelopsemia all over this place, and they're becoming more and more common as we're going shallower. Precious coral, Hemicorallium, pale morph. So just as the color is variable with a lot of these plexaurids, one of the things we've noticed in the past has also been color variability with the precious corals like Hemicorallium. There are some differences in color associated with different genera like Corallium versus Pleurocorallium versus Hemicorallium. But for the most part, even Hemicorallium, there are many different color morphs from dark red to pinkish white to straight pale. Yeah, that dinner plate anemone we've been calling it. But
is the Hemicorallium quite different with the Swiftia? How can you tell the difference?
The differences in
Between Hemicorallium coral and the
Hemicorallium. So the Hemicorallium are usually these pink with white.
those
Yeah,
ones?
those ones. Yeah. Swiftia, haven't seen it in a while. It's actually disappeared. This sparsely branching Swiftia. Haven't seen it.
Star.
Yep. Big ophiuroid vascus star. I've seen a few. We've seen a couple of different branching ones. Nope, that's another primnoid.
Is there a common name for primnoid?
There actually isn't, not that I know of. Common names are often given by people who work and encounter them in their regular lives, and it's it's really difficult for a lot of these exclusive deep water species for people to happen upon them. So a few genera have been given common names in the Primnoidae. For example, Primnoa pacifica off of Alaska in the Pacific Northwest sometimes is called red tree coral because it grows off of the sides of glaciers and things. That one right there. Yeah, let's shoot over there and just do a quick zoom in, just for the associate confirmation. So red tree coral is popular for Primnoa pacifica. But yeah, it's only very few species. A lot of the primnoids we're looking at, they don't have common names, and there's not really a common name for the family overall. Yeah. That's okay. That's okay. That should be enough. I'll let for a few seconds. But it does look like the same Metallogorgia as before, just maybe a little bit less developed.
A sponge. Looks like isoculture colony. Fossilized. Fish.
Sorry, can we look back at that purple coral that's kind of at the top of that white one there? Just a quick zoom in. Yeah, we can take a look at that on the way out. But this one looks just a little bit different than all the other purple ones we were seeing. So I just want to do a quick... Yeah. Yeah. I was just going to do a quick zoom in and out. Going in? Oh. Very deep purple. Usually they have the axis, the main stalk of the coral is usually whiter. But Tim sampled one the other day that was really quite purple, and it was like juicing all over the countertop. Purple goo and yeah, definitely very densely pigmented. Oh, yep, it's a bamboo. Yeah, just for the associate, I'm going to zoom on the crab as we swim, as we go by. Alright. It's on the wrong side, so I still have to suffice. Okay. 971 meters.
Fish.
Our next waypoint is somewhere around 882. Yeah, really dense. Relatively, for the deep sea, I guess, relatively dense Oreos. They're typically associated with a lot of these overhang communities. The depth of our last Niskin. 78. Go another 100 meters or so and see what it looks like. I can't quite make it out any closer. The left or the right, or it looks the same to me. Either you can pick either one to approach. Can't quite make it out though. Let me know if I can push in at all. Might be able to get you a yes or no with a better zoom. Wow, that's confusing. Yeah, can we look at the colony to the left, maybe? Also? White plexaurid now. Or white or pale blue. If we take a closer zoom on this one, kind of like a tight zoom, we may want to sample. Yeah. Yeah. Just giving you a heads up ahead of time. I don't have an ID for you on this one, unfortunately. Yeah, not at all. We'll take a scan around for associates as well. Maybe if there's a collection we can do, it'll benefit all of us if we get some maybe brittle stars with these as well. No shortage of brittle stars on this. I can already see dozens of them all over. Yep, coming in. Well, it's a plexaurid. I have no idea. But we'll sample. What? Yeah, it's a plexaurid. I mean, it just with these plexaurids, it's very difficult to differentiate a lot of these species by imagery. Oftentimes we'll have to look at the sclerite morphology in combination with some genetic tools. We are going to move for a sample though. I have a suspicion about this and I'm going to have to wait until it gets up on deck, but we'll move for a sample. What's easiest for you to grab, upper or lower? Lower. Okay, so just take a cluster of branches there and go for a quiver-sized bit. Yeah, go for it. I was actually going to go after that, after the collection, try and image the crab, but if we can get it now, that's great.
that on its back?
A different plexaurid, strangely.
You see the Tim's message?
No, I didn't. Tim, let me know if that zoom was enough, or we can go back after this collection here. Did you snap a in-situ image? Yeah. Sorry, I'm gonna re-image it here. Oh, is that okay? All right. Press 1. And this little bit is more contrast. All right, press 2.
Yep.
Okay, we can go for collection now, and this is going to be sample number
82472.
482.
482. Any additional text?
White plexauridae.
Why?
AU. Yeah.
That's it.
That's it. Can you write with many small brittle stars, so we know that they're associates with it? 482.
You see that that's about how we collected it.
Yeah, I mean, we're trying to collect as much as we can. You know, in that allows us to accomplish our science goals, our other science goals as well. So we'll keep an eye out for those black corals. Now, all of these have a very proteinaceous skeleton, so they're very flexible, but they're also very fibrous, so it does make for hard to cut sometimes.
I think Adam totally understands that right now. Looks like that last piece, Adam, is in the curves.
We can go back and grab more of that. That's helpful. It's cooked down there.
enough? You want to have a piece?
Can you get one tiny branch, maybe half that size, a little bit more? Thanks.
What quiver is
Just because
this?
there's a lot of associates in there. This is going to be quiver 7.
Red box.
That one could have gone either way.
Same spot or different spot?
Same spot, whatever you guys can get.
You might get a better angle on it. It's not thin.
Yeah, that's fine too. Yep, absolutely. Right. White blue plexaurid, white/blue. It has a blue tinge to it, and I want to make sure we note that color.
Yeah, it does blue, like
Yeah.
white blue. It's a mixture.
You have sample number 482, right? Yeah, great. Okay. Quiver.
I'm just waiting for them to put it in and
It's already in there. One piece is already in there, so you
Okay.
can take it. Fantastic. Thanks a lot.
Except from this spot.
That's it. We're doing pretty good time too. Our goal is to get somewhere around 300 meters or 279, somewhere around between later on today. Originally, we were supposed to go for a little bit shorter, but we had the notification that we had been extended a little bit. So let me find the off-bottom time here.
Anything else in here?
That's it. deck 10:15, 10:30. So assuming we make it up to about 300 meters, that means off bottom.
Well, we'll make it. We have
About-
Speedy Stevie here.
I don't think we're moving that fast. I thought I was moving really slow today.
You actually, you're not your usual.
Well, I mean, the other dives we were doing were much longer. This is not nearly as long of a trek, but the topography is challenging.
Yeah, this isn't the kind of terrain that you zoom around.
No.
2.5 kilometers, 2.25 kilometers.
Okay. It's a good length then. But
Averages.
yeah, the thing is, it's so it gets very hard to determine how long it's going to take you to cover the more steep stuff that we're heading into. And so you could cover, 200 meters in about half an hour vertical, right? But it's taken us, we've been on bottom for about 500 meters. We've covered a kilometer and a half, something like that.
You expect to see a lot more stuff over the top too.
I don't know. Sometimes you do, sometimes you don't. It's pretty patchy. If you get the right overhang, like we saw, or that coral garden we drop back into in the following dive. Sometimes you get lucky. Sometimes you don't. So one of the dives we did at Jarvis Island a couple of years ago has a similar morphology to this, but we basically just started at 700 meters, went up to about 3, pulled back off, dropped down again, doing every transect we saw something different almost.
Where's Jarvis?
It's about 1,000 miles to the east of us, right on the equator.
Are we gonna hit it on the way home?
No. It's like due east, so yeah, it would be it's straight due east on the equator. In fact, we're a little bit far south of it now. It's about 0.3 south, and south, in line with the Line Islands.
In relation to Iron Man?
I don't think so, but
Have you been to Palmyra at all?
I have.
Do you have any crazy experiences there?
I did not get off the ship. Yeah.
There's one of those big stonefish.
You mean on the island or around it?
Around it.
Around it. Is it? Really? I didn't know that.
Is that the eating stuff?
Yeah, let's look over on the right-hand side one first.
Palmyra is what? It's atoll that's got all this crazy folklore about shipwrecks and murder and ghosts and aliens. And
Really? I didn't hear any of that.
Really?
I didn't hear any of that.
Oh, look at Google Palmyra atoll.
I will.
There's a book written about it.
Oh, there's a lot of UFOs in the news these days. I don't know, maybe there's some stuff going on there. Maybe we should pop in for a visit. So we're setting up to look at an instance of coral predation here of this sea star on this colony of Metallogorgia. It's not usual we see sea stars predating on Metallogorgia. In fact, I think this is the first observation of this particular association that I've seen. Thank you. I'm going to do a quick zoom in. So, this is actually really interesting. We see here, thanks. So what we're looking at is this sea star seems to be working its way up from the bottom here. You can see where it's torn the tissue, has its stomach extended through the body of, through its body, and then it's kind of working up, eating the, actually probably just the Seamountkima tissue. No polyps really on the base of this, which is really interesting. You'd think it would go for the, you know, the nice juicy polyps, but it really seems to be happy with just the tissue that's along the skeleton right here. It's very neat. I think since we didn't get the last one, I think we might do a full collection here of everything. Let me just check in. So if we do a collection here, it would probably be from the very bottom into the bio box.
Like the whole thing.
Yeah.
Do you need us on In-situ?
Yeah, can you hit the corallivory button also?
Or
I'm gonna scan the colony one more time and see if there's any other predation here on the tissue.
this is a chrysocculture?
We've been calling it metallogorgia.
Metalliculture. Okay.
Yeah. Or you could do
But, you know, it does share some characteristics with chrysogorgia, but the polyps, when you look at them, are more metallogorgia-like, which is really interesting.
try to get it up.
Peter star and
There's a shrimp in there, too. Another egg case.
shrimp.
This one looks broken, though. This egg case looks like it had a former resident. Could be live. It's tough to tell. It looks like the coral, the whole colony is responding, though, to this predation event. It seems. It seems that perhaps the tissue loss at the base is resulting in all these polyps to close up. It could be something else as well. It's very tough to tell since we're really just getting a snapshot of what these corals experience in time.
Oh, and the star. That's true.
Alright. So I think we're going to set up for a whole collection and do everything, kind of from the bottom, try and put it in the bio box. C is the biggest box, right? Do you think this will fit everything?
Yeah.
Okay, let's do it. I think if you're careful, you should be able to get it all,
Base and everything.
base and all. Yeah.
So maybe I'll try not to clip it, just grab it with the pictures of
Yeah, yeah, I think it's fine. Yeah. Yeah, I mean, worst case scenario, I guess you could clip the top and then clip the bottom. It's just that you can't really get the predation event without getting the whole thing, and that's unfortunate.
Just try to grab it with just the edge of the red.
What is the other thing hanging on The green, kind of like a green thing.
Where do you see?
Like
Oh, there was an egg case down there, I think.
egg
What,
case.
yeah. I think it's some kind of egg case. I'm not sure what it is.
We actually need to stop for these two.
He'll stay on there. He's not going to come off. He's got his whole stomach wrapped around that.
Nice the mouth.
I think what I'll do, Steve, is I'll cut it and grab him and I'll come back for the base.
That sounds fine too.
Just because the base is kind of in the wrong.
Yep.
If you got rid of the red, you might be able to.
So we can try. I just suspect because it's holding the weight of it.
Yeah, whatever you need to do. Either is good for us.
Okay. Wait, do we took the picture of that thing?
Yeah, we have images of that. You can do one more if you want to. No, set up for the sample. I'll do the in-situ imagery shots.
Okay.
And you can just enter this.
Make it easier for grabbing that stand.
483. Right?
Bio box.
Yep. C.
Okay. Any additional tips?
Metallogorgia.
I think you already started here. META, Geo. Okay, Metallogorgia.
You just write this.
Swimming
Oh yeah? Above the camera there? Saw the shadow of it. The description should say something like this. If you can add a little bit more information, like sea star predator, ophiuroid, shrimp, and egg case, just so we can document all the different types of associates with that organism.
Sea star predator. OVH. Shrimp.
No, I just don't want to. It just takes a long time to get to the point where it starts moving.
Alright. Very nice. You don't need the. Are you going for the base of it?
Yeah, I thought you said you needed it.
Oh yeah, no, we don't need that part.
Oh.
Oh, sorry. I meant the whole colony, not all the little.
That's when he said that.
bits and
No,
bobs.
yeah.
Sorry, sorry.
Yeah,
I thought it was implied. Sorry.
That's why when he was trying to get that.
Yeah.
I thought-
That's why I was trying to get the base there.
Oh, I thought you were trying to clip it so you could take it all at once with the star on it.
Yeah, he was, but also he was trying to get the whole base.
Yeah.
Oh no, sorry about that.
Cool.
Yeah, saved you some effort there.
Good to move on. Good to move on. Steve?
Yes, all set to move on. Thanks. At least we got one predation event in the bucket.
Sea star.
I think that's what's important. Yeah. Another solastrid sunstar.
Is that a standing for floors, like on top of
No, there's no xenophyphores here.
Okay.
There's a hydroid and then a sea star.
No, like the brown thing on top.
No, that was a part of the rock.
Okay.
Hold that for a couple seconds. And good. I've never seen any these solastrids eating corals for some reason. Not even on sponges. I think they have a different feeding mode.
Maybe they didn't starfish.
It's possible. Yeah, there's a number of those.
Maybe that's what keeps the numbers down.
Yeah, in the Caribbean, where I've done a bit of work, there's been at this depth and even a bit shallower, sea urchins, the Sideroid, the pencil urchins. They're voracious. They'll like eat the stalked crinoids that they're super dense. They'll start nibbling on the bottom of them, and then they fall over, and then they just like pin them down. They use these spatulate kind of spines. They have modified spine to pin down other crinoid feather stars before they can take off and swim around, or they pin down these stalked crinoids. And they're just voracious. Yeah.
Alexis mentioned this morning that some of these corals have neurotoxins in them.
Corals?
Yeah.
No. Not familiar with that. Yeah. Supporting the entire weight of the rock above, maybe. I don't know. I always thought it would be really nice to do some scuba diving in this type of terrain. It's like overhangs and little crevices and stuff.
Cave diving.
Cave diving, spelunking. Yeah, I mean, if you do dive, the deep water habitats, even at 100 feet, 200 feet, something like that. If you do have the ability to dive that deep, sometimes you see features like this as well, depending on where you are. This is essentially the same kind of stuff, it's just deeper. And in some cases, it's broken off of the main carbonate cap. And some of these features might be just massive blocks that have fallen off, like we saw earlier.
Oh, sea cucumber.
That one? There's a fish in there.
A fish? Okay.
Yeah.
It looks like a sea cucumber. Metrodora and coral.
Yep. Hemicorallium and Plexaurids. Big overhang. There's a lot of ophiuroids. This rock is covered with these planktiverse ophiuroids that kind of have their arms up in the water column here, fishing. More of this Copetrophora. Crusting sponges. It looks very similar to what we were saying about it. Maybe 100 meters, 150 meters deeper. Just the terrain, the topography is almost identical. So it's not that we've moved out of the depth range, rather, but look at that little chute. It's a nice little hole. Lots of colonization. So it's not that the depth range is any different, it's just that these particular corals that we were seeing down deeper, they prefer these larger overhangs. Perhaps the flow conditions are right.
What's the worm life thing at home?
You can see that in this particular hole, all of the corals around the outside are probably taking advantage of water moving up and down through that hole. But it doesn't seem to be very successful. They're rather small, and some of them are pretty ragged looking. More of that clavularia we sampled earlier. Great collection. We just broke a big chunk of that off. Yeah, it worked really well. Yeah, don't go under that. Don't thrust too hard. We've also been seeing a bunch of nudibranchs around here as well. They kind of look like corals. I think this is one of them, that white blob there. Really neat looking. Oh no, that one's a sponge actually. A lot of these large, long-armed crinoids. And I haven't seen any of them like this before. Crinoids are notoriously difficult to sample. Yep. Another big one, dinner plate. Another swiftia in the background there. Yeah, so it's like we've I don't know. I thought we moved out of that zone, but apparently everything is still here. It just has very specific habitat preferences. It really likes these overhangs, and it seems like the overhangs need to be of a certain in a certain angle, maybe to the slope, where you get better conditions. It's not quite clear. It's a black coral right there. Let's see, maybe bathypathes. Yep. It's the first one we've seen in a while. Hemicorallium. We've been seeing all sorts of color morphs in this, both from very pale white all the way up to dark reddish or even pink. Surprisingly, though, we're not seeing. So we saw some madrepora earlier. These stony corals. This is something that we're really on the lookout for, particularly the rubble fields that are associated with them. The depth range is ideally for those is within 600 to 1,000 meters. We started seeing them about 10-20 meters, but we expect them to be more common. I think 600 to 700 meters seems to be the sweet spot for them. It's really before the slope starts to get too severe. So we'll see what we find. Should be pretty interesting. 6. I think they call a group of Oreos a pack, but I'm not sure.
A pack of Oreo?
Yeah. Nothing too unusual here. Most of these kind of ragged-looking corals are a lot of these hemicorallium or corallium. There's another one of those Paramaricia that we saw earlier. Some a little bit of different terrain here. Yeah, that's interesting that it's we're seeing all this different. Oh, there's our first big Slidenia. Yeah, we did actually sample a bit of the dark rock, like this. We saw one of these like random rock falls. Looks like it was pretty heavily encrusted with carbonate. Heavily encrusted with iron manganese crust, a bit shallower. And so what happened was we grabbed a piece for our second rock collection. We'll be able to see if that actually is carbonate or if it's something that's been maybe displaced significantly. A lot more of this madrepora. The rubble fields, though, are not really extensive, kind of just really patchy immediately below here, so not sure what to think about that. Maybe these colonies have not been here for too long. This is a good example of the mixed madrepora and Elopsamia. So we're seeing two different species of stony corals here. This is what we typically see in some of these reefy areas. Is that it's a mixture of the two. It seems that madrepora appears to be the better producer, kind of producing more fine branches and more structure. But the Elopsamia is larger overall, has more massive skeleton.
Steve, how old would you say those corals are?
With the madrepora, I'm not sure. For this part of the world, we kind of have to take a little bit of local knowledge since they have differential. Oh, the hydroid, the gelatinous stalk thing. They're solitary hydroids. But for the Elopsamia, there's actually been some growth studies for smaller Elopsamia colonies in the area of somewhere 20, maybe 30 centimeters tall, and much like some of the stuff we're seeing now, and a number of these are actually on the order of several hundred years old.
At this height?
At this height, yeah. Using radiocarbon dating techniques. But it's important to note that growth rates are very difficult to expand blanketly, blanket statement about growth rates, because a lot of things can affect growth rates, like the amount of marine snow and food inputs to the benthos that the corals can take advantage of. And any sort of disturbance is going to, just bioerosion is, for example, is pretty common on stony coral reefs. So that can change how much size is a reliable indicator. And without a collection, we really don't know for sure.
Just trying to get an idea of when that rock might have fallen.
Yeah, that's a good indicator. I mean, what's more interesting to me is maybe the amount of rubble and debris. We usually on these more intense slope areas where you maybe have stronger currents, they're growing a little bit faster, so they're producing more and more rubble that's falling down. But I'm a little confused by how dark some of this rock is at this depth. You know, what we're seeing is almost exclusively carbonate, but there seems to be some pretty hefty iron manganese crusted stuff falling downhill. But I wonder if it's because the terrain has modulated a little bit and the slopes are a little bit, you know, where you have an overhang, the slopes are greater than 90 degrees in places, but now that we have these more gentle slopes, I think it's filtering out a lot of those overhang creatures. But we're still seeing plenty of Metallogorgia, Anelopsamia, some Plexaurids, riddle stars. But in addition, there seems to be a maybe slightly higher sediment accumulation in this area, possibly due to the topography, but also possibly due to us being on the edge of you know, one of these large carbonate blocks, or at the base of one of these slopes where sediment seems to accumulate. James, could you, when you have a chance, could you throw up the bathy map, the overlay? Yeah, for everyone. Yeah, I'll give another overview of the site. So we're going to throw up kind of the dive track where we are and where we're going to now, so everyone can kind of see where we are and how far we have left to go in the dive. We came up a pretty severe slope, and actually it's going to get more severe as we get in the next few hundred meters, maybe the next waypoint or two, where we're going to start to see more of this verticality. We've had some pretty tremendous verticality right now, but you know, there's a lot of overhangs and still overhangs and karst-like features hopefully yet to come. But this is where it gets interesting. Yeah. So here's the dive track. I don't know if you can see it on the feed overlay, but it's pretty steep here, and it's going to get steeper. And this is where we expect to see a lot of this stony coral reef and build up. Oh wow, Iridogorgia. That's quite shallow for Iridogorgia. Can we take a look at the associates with it? Just to see if they're different from the ones we see down deeper. At this depth range, I'd almost expect to see Rhodin Iridogorgia, which is a sister genus. Yeah. Okay. But this is almost certainly Magnaspurus. Yeah, the crab, and then there's one towards the base of the colony as well. If we can just kind of go down the length of the colony, that would be perfect. Hamalid crab with a yellow plexaurid. Yeah, working your way up. Yeah.
There's a shrimp.
Long-legged shrimp on the back. Yep. Oh yeah. Chirostylid crab on the back side of this. 2 shrimp at the base. Not usually associate shrimp, these nomadic arsinids. It's a small yellow plexaurid fan, also at the base. Alright, I'm gonna zoom out. Or, yeah, that looks good. That's alright. Yeah. Very nice colony. I have to check my depth range for Meredith. I didn't think they got this shallow. Oh yeah, it's right up there, 877. Wow, it's 877 on the dot. Holy moly. That's impressive.
What was 877?
So I analyzed all the video from 2017 in Phoenix Islands protected area, and 877 was the shallowest record. It's like on the dot. Wow.
Let's beat it. Let's beat it, yeah.
The deepest is 2200. That's a really impressive depth range. It's almost like ground truthing on the opposite side of the equator. These plexaurids, or the metallogorgia, what we're calling metallogorgia. Oh yeah, yeah. Let's image that. Yeah, that's great. You want to go kind of from the base to the tips of the polyps on this one as well?
With the zoom?
Yeah, with the zoom. Yeah.
You want to be on the side, be able to see if he's eating or
I think we might need to do a quick zoom on both sides, since to ID the star we might need the top side. This one looks pretty ragged. Yeah, so we'll take a look at this side for the sea star ID, and then we'll do a bottom to top on the other side to see what it's actually feeding on, because I don't see a single live polyp on here. But I'm wondering if I don't know if the star is actually responsible for eating all of these. It seems like it would be a pretty precarious way to get up and around all those polyps. Maybe there's something else going on here. Alright. No live tissue. No live tissue. Can we rotate around to the backside?
Yes, sir.
All set. Sure.
Are we doing some collections on
No collection here. We've filled up all of our available containers for something like, and then there's no live tissue, so this is kind of just a document.
All right. Go ahead. And you're in main.
Great. Got it. So it looks like just taking a look at the stalk from the base up here. looks like hydroids at the base, so this is clearly an area that's probably not recently grazed. But if we start to work our way up, the amount of attached hydroids and little bits of things like small gastropods on there as well.
That's such a cool color.
The metallic golden color?
Yeah.
Yeah. It's like a bronzy, iridescent bronze. Yeah.
Is that a chrysopulture?
We've been calling it Metallogorgia. Yeah. But you can see right where the star is. It actually has a pretty bare skeleton. So, you know, it was clearly feeding on something here, but it may not have been the coral, because the entire coral seems to be dead, or the sea star is the culprit for the predation. But you know, it is interesting to note because there's a fair amount of fouling of the upper part of this colony from the sea star to the left here.
Yeah, it's like really dirty over there. You just cleaned it.
Yeah, yeah, it's like it's has a preference for the mid colony or something, or it's an extremely slow process that we don't really appreciate how slow it is.
Going up trying to find food and there wasn't any.
Yeah, yeah. So yeah, it's interesting. Yeah, I've never seen a sea star on top of a Metallogorgia like this. I don't know how easy it would be to manipulate to kind of travel through this bramble, since these are fairly fine points. But I've seen sea stars do some pretty crazy things before. But this homalid crab is definitely faithful to the last. It has its little coral here, coral hat.
Keeping it all.
Yep. Trying to make something out of it. Oh, yeah, there's a little individual Metallogorgia polyp.
Is that really?
There's a single polyp, dead center.
Wow.
Yeah, with a little bit of tissue. That's impressive. Probably not long, since there's a bunch of hydroids, and I typically will they'll typically go for killing off the live tissue. There's another tiny little polyp in the lower left. There, kind of hidden by one of the branches. Got live tissue there. The two polyps. So I don't know. Cost-benefit analysis for the sea star. He was probably like, maybe not. Happy to feed on something else. All right. I think that's about it. Can we get lasers on though, and before we take off, just to get some size perspective on all this. All right. Base there. And on the sea star. I think that's gonna have to be good enough. Just put it in the same plane here. All right, good to go. they really are starting to like these more than 90-degree slopes on these faces. So this is this yellow one here looks like yellow metallic, yellow Anopsemia, which is a different color morph than we've been seeing, but it's all the same species as far as we can tell. They just have a slightly different pigmentation to parts of the tissue. But we've been seeing both of them concurrently, so you know, we did sample quite a bit of it as well in 2017, and there was no morphological difference between any of the yellow or pale or translucent Anopsemia. No, we've seen a few of those already.
Sea star or urchin?
It's a urchin, yeah. All right. That's a crazy anemone.
Oh, that's a big one.
Oh, there we go. Yeah, this is what I was. This is not quite. Fooled us again. So this is a separate genus in the same family. This is called Rodaniridogorgia, and this one species it seems to be only in the Pacific. Let's take a quick image of this guy, because I mean I don't even know what type of anemone this is. Anemones just don't get enough love, I think. Yeah. The thing is, they love to soak up a ton of ethanol that we don't have to preserve them. They're so full of water and gelatinous goo that you really have to dry them out a lot of ethanol, or preserve them in formalin, which is also an option. But
So you have to like submerge the whole thing in just pure ethanol.
and probably do multiple change outs, yeah. Yeah, because it dilutes very.
So ethanol
This is
drops
a
water.
beautiful.
Yeah. Usually they're formalin preserved, and you can take a small piece for genetic analysis, but when you put something in formalin, it doesn't preserve the DNA very well. It's more for morphological analysis, since it hardens the tissues a little
So
bit.
what would you do? So you put it in ethanol like overnight kind of thing, and then swap out ethanol like 3 times to do that?
Yeah, I mean, it depends on what you're doing. You know, a lot of the times we don't, ethanol is more expensive than something like formalin, so what we'll do is, just take a small snip in ethanol and
Sorry.
put a small snip in ethanol to preserve the DNA portion, then we'll put the other bits in formalin, harden the tissues. You don't need to worry about the DNA as much, because, it'll preserve it, fix it. So yeah, a Rodaniridogorgia, very nice one. More of a wavy axis than a helical, but this is actually the depth range where we start to see this as well. So we kind of transitioned from one of the helical chrysogorgids into the other one.
So what was the consensus that was part of the same family or not?
It's part of the same family, yeah.
So J-Rod got the record there or what?
That's where it doesn't transition.
It starts to transition. So the shallowest record I have is like 595, but they go down to 1300 in certain places. It depends on the terrain. Lately, in Helen and Baker, this unit of the US EEZ, We've seen a lot of them in this depth range, not so much deeper. So that's kind of interesting that they are a little bit shallower in this vicinity compared to further south. This was a roughie, I think. Starting to get up into the roughie zone.
Saw that before, this black stuff right here, that thin line of it.
Yeah.
We saw that in the Emperor Seamount, and it was like where there was a transition, everything else eroded, but that it erodes slower than everything else.
Can I take a look?
Yeah, I'll try to get you a side profile.
You know, I think I've heard something like that.
It was like a crack where something built up right there, a heavier kind of thing.
Yeah, I think I have an idea. Well, while we have Stable here, I'm gonna look at this shrimp. It's black and purple, which is kind of weird. Before it swims away. Look at the mobile fauna first. It's gone. Yeah, so
Same thing, Cody.
All right, bro.
I think what this might be is bits of crust that have formed in the past, and the crust is probably less soluble than the carbonate rock, so it just results in a crack exposed. Welcome back.
So easy. You know what? I'm gonna get you good. You try. You try, but it's plenty. Brewing.
Alright. We're all set to go here. Yeah.
Get my bearings and see where we're at, what we doing.
So we just passed 6 a while back, and we've been moving pretty quickly towards 7. Between 7 and 8, it's going to pick up significantly, but I'll leave that for the next watch to help you through. But
okay.
It's been kind of like this, kind of unremarkable,
Weird
smooth little cliffs. Yeah, not too many overhangs here, like you were saying before.
And the plan is to try to get to like 110, obviously.
Yeah, we're just gonna cruise along for the next few minutes, and then next shift will come on.
Well, let's start cruising. Cruising forward, cruising.
There's another partial tissue loss in the telegorgia. Redfish, yeah, sure. I haven't seen this fish at all. Some type of, I don't know. Can you hit the fish button, please?
Yeah.
When we get a focus on it. Huh.
Can I hit it?
Yep, you can hit the fish button. No idea on that fish.
Swimming fish.
Flatfish, redfish.
Jesse, batfish?
Batfish?
Yeah.
Yep.
Ongoing.
too. Look what's in there.
I see it.
Jelly.
Yeah,
The jelly made it.
it is jellying along.
Stuff.
What's in quiver or is that jar one right there?
Jar 1 has something. I don't know what's in it.
It's okay. Don't worry about it.
There's a possible crab.
Okay. I don't know if I see a crab.
Green.
Okay.
Dude.
All right. Let's see. I think our squall officer friend may have run off.
Sun's coming up. He has somewhere to be, let's just
I guess so.
get people to see. Alright. Here we go.
Then we'll just carry on.
Okay.
Seeing a lot of these Natalagorgias that look like they are not doing so well. I've seen a couple that look like that have had sea stars on them. One that just had a sea star at the base, but the top of the coral looked healthy, and another one with a sea star at the base and the whole coral looked dead. So, somewhat curious. We have a couple anthomastus mushroom corals. This looks like the ledges that or overhangs that Steve was seeing through some of the dive earlier today.
That's a big basket star.
Yeah, big basket star. Lots of Natalagorgias. Looks like a couple colonies of Anelopsamia, this yellow stony coral, all facing directly downwards. I'm just gonna come in a little bit on that one, Cody.
The ones to the right?
Yeah. Thanks. So we have two different kinds of coral. The yellow one is an Elpsamia, and then the white one on the bottom right, or at the bottom right of the screen, is a Madrepora. And like earlier, Steve was saying that when we see one of these types of scleractinian corals, the hard corals, we sometimes find them growing together, and that seems to be what's happening. This Madrepora has settled on a dead portion of the Anelopsamia and started to grow its own colony. And then at the top portion of the coral, you can see a lot of dead skeleton in the back. So it seems that this has started to grow in essentially in layers, and we've seen that in the past where they kind of grow in plates, with the back portion eventually dying off from either, it looks like this might be from sedimentation or from lack of flow back to those polyps. That's good, Cody, thanks. So we had a science watch change, if you can tell the different voices. I'm Alexis. I am with the U.S. Geological Survey.
And I'm Brian Kennedy with Boston University.
That's interesting. Is that a roughie?
Looks like it.
Got a homalid crab back there with an anemone. I've seen a lot of homalids today with their little backpacks on. Going to school. I think that's good for the fish. So we're currently at 820 meters, and we are working our way up to what should be a mesophotic zone at the top of this Seamount feature, around 300 meters. So I would say, I guess we won't really see mesophotic transition. That's kind of the limit
Depends on where we stop. I mean, the very peak is 200.
Oh, is
so
it? Okay.
We might get there.
We're seeing more of the yellow Anelopsamia as we've kind of come up in the last 100 meters or so than we were seeing down below. These plexarids are pretty ubiquitous across this dive. Same with these. There's a lot of Metallogorgia.
Yeah, this has been the Metallogorgia dive certainly.
There's something you want to look at, just yell at me.
Okay.
Just keep cruising otherwise.
I'll just scream in your ear.
Sounds wonderful.
And we're up here on what is more what we're calling the carbonate cap of this feature. So when we started the dive this morning, Brian and I were in here for the very first 30 minutes of the dive, and there was more of that really heavy crusted, possibly igneous rock. And now at some point during the day we moved up onto this carbonate where it's likely the relics of an old reef that would have been closer to the surface, with shallow water corals growing, and then over time those die off and it turns into this fossilized rock formation. It was really cool. I think maybe you were flying, Cody, earlier, and you guys saw that really white break of the stone. It was pretty. I've never seen that. I think that's I don't know, cucumber? Yeah, we got a lot of sediment up here. Okay.
No big outcome.
Let's do it.
We'll get a little blue for
Okay.
20 meters.
Yeah.
30 meters.
We're headed to what looks like another rock face based on the sonar. Also takes us back towards our track, which we draw these tracks and they're kind of tentative outlines for what we need to follow. Often we stick pretty close to the lines, but if your option is to traverse over sandy bottom or rock, we generally choose to hunt for rocks and stick on hard bottom because that's where we find more coral and sponge diversity. It's an interesting fish.
Let's look at him.
Okay, looks like he's got a needle nose. Just completely vertical.
I've never seen one quite like that
He's
one.
gonna boop his nose, yeah.
Looks a little bit like a snipe eel, but it's different than the ones I'm
Okay.
used to.
Wow, look at all the different shells down there too. Oh. Ah.
I don't know if he could have eaten him. I
Could have
don't
been.
know if that fish could
Could
eat
have
that
been
shrimp.
Nat Geo right there.
Shrimp after fish.
I know, that's what.
What a weird beak.
It does have a very. Oh, is it? Trying to see if it was. Yeah.
It looked like he was eating.
Okay, I think that's good. We got some good imagery of the head. What's that? Is that a shrimp to the right? Yeah, it looks big. Maybe it's just a big shrimp. It's a pretty. Guess they're just pretty big shrimp.
Where it starts getting steep out.
Okay.
So,
Yeah, this is probably going to turn vertical real soon.
Yeah, vertical.
found us some rocks, so.
Good job, Cody.
You can see as we get shallower, the density of fish is really picking up.
Ooh, hole. We are kind of cruising because we are trying to cover a pretty significant distance, but it's also with all of these, with how porous these rocks are. If we sat down and really, oh, it looks like another dead Metallogorgia. Just surprised by the, yeah, or hermit crabs on there. Just the different health state of all of the Metallogorgias that we've seen. Maybe. But if we were to sit down and peer into all of these little pores and tiny caves, we would find lots of organisms living within these rocks. Different types of crabs and probably even small sponges that we're not seeing all of. There's a nice little cup coral skeleton. It's broken off of a very dark rock. See that little crab hiding out in that hole? Yeah. Oh, big homalid. It's okay. We don't have to zoom on all of them. We've seen a lot today. But we do need to look at this big sponge. It's so white against the carbonate. I think this is quite different from some of the sponges we've been seeing on this trip.
Yeah, it definitely got those, I don't know exactly what the technical term is for them, but in a mussel they call bissel threads, so the things that it mounts to the seafloor with.
Yeah. And kind of all around the ridge of the sponge too. There's all those projections. Lots of ophroids. Looks like an anemone living in there, but not really on top of the sponge. There's a crab over here. Looks like a little bit of a different...
Yeah, that's definitely a slightly different looking crab.
You got me.
Oh, Kido, thank you. Looking a little scruffy.
Yeah. Like he's been around the block.
It's got some hydroids, looks like, growing on them.
Got red eyes, some black.
Yeah.
So mean.
Looks like maybe a different type of homalid. Does it look like it has modified hind legs? Or am I...
I think
No?
so, actually.
Yeah, you're right. No.
I think it looks more like something like a spider crab group or lithodid.
When you said modified back legs,
So the homolids, when we see those crabs that have like coral or anemone, their hind legs are modified to where they're sticking up and they hold that
Whatever the decoration is.
that little backpack on
Yeah.
there with those legs.
Kinda like this one would.
So this one's legs, yeah, are like normally a crab has all of its legs
Mhm.
down, right? Sorry. But we'll look at a homolid and you'll see them pretty clearly. But is this something, do you think we could, I'd like to sample it. I don't know if it will, I think it will get stuck in the slurp tube. It's not huge, but the legs are, it's gonna extend its legs probably.
He's pretty small. He might contract his legs.
Okay.
A little bit.
Well,
Rusty,
I would, yeah.
Small.
I would suggest if we try and
if
suction and if
it
we,
does
yeah.
spur out right before he gets into the actual tube itself.
Yes.
Oh.
Oh, good eye, Jared.
Wow.
Let's get in there.
Keep rubbing the sheep.
I'm trying. Oh, I think it's it's got claws.
It might be a mantis shrimp.
Oh no, come back from. See the claw
Yeah,
down below?
yeah. I think it might be some kind of stomatopod.
Oh. It's so blue. Come over to this left section, buddy, so we can see you. I'm frustrated. Wow, it's definitely some type of crustacean.
Is that his eyes that are blue too?
I think its eyes are like red, black, those little dots.
Yeah, okay, I see that.
But its whole like head looks to be see-through. Do you have any more? Here we go. Wow.
Do we have a new species?
I don't know.
You're the scientist, you should tell me these things.
I'm not an encyclopedia, Jimbo.
People are saying mantis shrimp.
It could be.
Got very bright blue
Yeah.
belly.
We have not seen that before. I don't think we could successfully sample it, so I don't really want to maul the sponge to try. I don't mean that against you guys, I just mean it's gonna, I think the crustacean would swim or, just go deeper in, so, but that's probably,
Maybe he'll pop out after we've done like a half.
yeah. So yeah, let's go ahead and try. You know what you're doing. We'll try suctioning.
Did you get your images already?
Are we, want me to go in?
Yeah, go on one more
Okay.
time.
It's so spiny.
Okay.
Okay. We'll go for that.
Alright, here we go. Sit down a little bit for you. Watch out. Okay. Before coming up. Let me know when you're happy. Or I'll let you know when. Is that good? Perfect. sir. We're good to go. We're going in 3. Let me know when you're ready.
Stretching the crab
Yeah. I think I can slide to the left some.
It's an interesting view of the sponge. I assumed it was like round all the way.
There, sure.
Oh, nice.
Amazing.
Okay. Wow. Good job, Jared. And Cody. That was like a fly sample.
What's the number?
That was literally last second.
484.
Let's see if this little move around there.
Is that jar 4?
That is jar. Is that jar 4, guys?
Yes, that
Okay.
was jar 3 that went into.
It went into 3.
Yes.
Okay.
Where I indexed to jar 4.
Gotcha.
Before the
See it anywhere? Yeah, that's okay.
Alright, I think we're done
Yeah,
here.
we're good to go. I think he's still stowing the.
I was on the EP3s. But he was probably
Yep.
EP30.
I'm gonna swim to the other side of this thing and see if
That's
it's
probably the
Okay.
day what squadron he was
It looks. I thought it would be like round all the way, but it looks like the other side just has those threads. Yeah, see, it's like. Yeah, so Alvin dives are normally like a day long. So you go in at 8 in the morning and Alvin's back on deck by 5 p.m. So it just depends on the depth that you're going to, how much time you have on the seafloor. There are ways to go to the restroom in Alvin, so nobody wears diapers. You just plan well with your water intake for the day that you know that you're diving, and you do your best. I don't see that. He must be just
He's really, I think he's
embedded
in the
within
matrix.
the, yeah.
I think he's literally living in between.
He's got a mansion.
He's got
Yeah.
but almost like a maze, right? He lives in like a mansion that's a maze at the same time. That's good. Thanks, Cody.
It's amazing, and when you look at the associates of a shallow water sponge and like collect a chunk of it and really dissect it or dissolve it, they are just chock full of all kinds of other inverts living inside of them. It's amazing how much life there is inside the matrix of the sponge in a shallow water reef setting.
Yeah.
Alright. Time to make the journey. Up. Up.
Up and away.
Away. Here we go. Hang on to your seats. Going fast. We're stagnant. Not on currently.
Yeah. And at the moment with like current technology, we can't you can't stream like human occupied vehicle or submersible dives because they are not tethered. The tether that's on the ROV is how we're able to get all the video data back pretty much instantaneously, and we're able to then stream it live with a very minor delay. But when you're in an HOV, so a human occupied vehicle, there's no tether, so with current technology we're not able to send that video back up to the surface, so you have to wait for that dive to be over, and then the people that were in the sub can kind of give a debrief. You can get all the video off of the cameras that are on that sub, and then people are able to watch what happened during that dive. So it's quite a different experience from a scientist point of view. Look at these big boulders.
Yeah.
I think that's surprising to me, considering that I would
Yes.
think that this would be mostly carbonate up here.
I would agree.
But yeah, so
My guess is there was some kind of much larger failure in the carbonate that got deep enough to get to the basalt. I don't know.
huh. Oh, we'll take a look at that coral real quick, that fan. There's also a dead stalk there with crab on it.
This is a hot mode, right?
Yes. Let's see.
Take a look at it.
Yeah.
Show me what.
Okay.
Oh, I see it.
So see how it has a coral on its back?
Mm-hmm.
So there's two appendages that are actually holding on to that. They're shorter little legs. There's one
Oh,
here.
yep. I see it. It's like a beak.
Yeah, it's like these little, it has these little short appendages that it just uses to hold on to that.
Okay.
Carry its hat.
Yeah, I call it a backpack.
Backpack.
Yeah. This looks like a, maybe a different, yeah, just a, we don't need to spend too much time, but I just want to get a look at the polyps. I think it's a type of Narella, but it's pretty, has pretty dense branching. it cool if I come in a little bit?
Of course.
Okay. Has downward facing polyps. Thank you. And a couple different looking ophiocanthids, those spiny armed brittle stars. Hermit crab. Not seeing any signs of damage. Okay, that's good. Thanks, Cody.
Do we have enough from nodes for Randy's experiments?
No branches for, well, that's I think from the jelly. I don't know how much was collected. We don't really have much. So we can do that.
Yeah, next one we see.
Yeah.
Probably.
Okay.
Grab some.
We did get some madrepora today with the idea of doing another madrepora experiment too.
Okay.
But I don't know how the last ones that we got at, was that Baker where we had all that, were really small, so they work really well in those chambers. I'm not sure if this one is that small, because the polyps might be a bit more robust, so I don't know if it would work in the respiration chambers.
Okay, I'm deviating away from that formation because that's taking me somewhere.
Gotcha.
totally opposite. I was going back down
Yeah.
and around. Some sediment for a little bit.
Cruise over some sediment as we head back over onto our track. Our next couple waypoints have pretty drastic depth changes, so
About to have, we're almost at waypoint 7 now.
yeah. See if there's anything of interest. There's some encrusting sponge, it looks like. Or an anemone, I think. A homolid that's running away from us.
So it's like lawn.
Yeah, it does. That's like what, yeah, Brian was saying earlier. We have these large chunks of.
For those of us who aren't really geologists, these kind of terrains, when you've got the manganese encrusting, make it really hard because everything kind of turns black once it's been exposed to the seawater long enough. So some of the shapes of the rock are pretty telling if they're limestone or if they're still basalt. But other times like this, it gets really tricky.
Well, yeah, and it's like it takes a long time for that crust to
millions of years.
precipitate out. So, and it's confusing to me when you have certain rocks that look like they have a really the shrimp looks humongous, or am I just like?
They're big.
Okay.
No, they're definitely big.
When you have a piece of rock that looks like it has a pretty even coating of crust, and then next to it you have a rock that looks like it's almost doesn't. It has a very thin layer of crust.
What do we want to call that?
I don't know. Just either you could check both paracordia and yeah.
Did you wanna go back and look at it?
No, it's okay. I'm looking good.
What is that on the left?
So do we want to maybe set here? We will maybe collect some of the white corals after we take a look at them, decide if they're good candidates for Randy and Anna's experiments.
So generally we consider the deep sea to be about 200 meters, where light really gets to be
There you go.
Thank
super,
you.
hard to find for or sunlight. But the last couple of days when we've transitioned from 200 to 300 meters, we started picking up the first signs of photosynthetic life about 250 meters. And this water is super clear out here. You really honestly can't get water much clearer than you find in this part of the world. So somewhere between 200 and 300 meters is really where all surface light disappears.
We definitely have a lot of damage on this colony. The branches are not only there at the top, but it also looks like they've been snapped off. We have a homalid crab hanging out down there. Or no, that's a Emunida. I can look around for a while until we have a nice Ellipsamia that looks super healthy. All the polyps are facing the other direction into the current, it looks like. Very light current that's up here.
That's the way the current's going. Towards us a little bit.
Yeah. Our artist on board, Constance, put up a post on Instagram yesterday or the day before, and she said coral or macaroni and cheese for this coral. And I thought it was hilarious because now that it's all I think about when I look at this.
Well, thanks for sharing. Now it's all we're I want macaroni and cheese.
Always, I always want macaroni and cheese. You can see the little dots in the tissue of the polyps, so the fleshy part. Those little specks are the nematocysts, so the stinging cells inside the coral tissue. So all cnidarians, which are the phylum that corals belong to, have some type of stinging cell. So jellyfish also belong in that group, as well as siphonophores and anything else that has the ability to sting with the nematocyst. We have another homalid. This one has a sponge on its back. Lots of homalid crabs today. We often see them on corals and sponges, but they have a different type of coral or sponge than the one that they are on top of. So we are going to make a couple collections here.
Okay.
So we're gonna do a collection from this coral and also from this coral over here. So if this one's easier, we can start with that.
Okay.
We just need a couple branches, so
Okay.
separate quivers.
Quivers.
Yeah. So if you cut like this section, that would work. I mean, we don't need the crab, he'll probably run away, so it's easier to come over here.
Yeah.
Go ahead and take a section from there.
Cool. Got some good pictures. Green light to go.
Green light to go? Or you want me to? Okay, I think we're good.
Jason's asking about those black spots we saw on the dead coral we collected a few dives ago, and we really still don't know.
Is that what we're kind of looking at?
Yeah, that looks good, Jason. I mean, not Jason, sorry. Jared. Yes, that looks good.
Oh, Jason?
That's cool.
Little bouquet.
Yeah.
And unfortunately, now slow, it's gonna push us away. Okay. Looks like you have one open now. All stop there. Sid's coming up slowly.
Too comfy in this chair.
Bring out the spires for you.
Get back in here. So we're doing this collection because there's a pair of scientists on board, Randy and Anna, who are running experiments looking at the respiration of corals, and they've been using primnoids as their study, I guess, family, because they have been doing really well when we bring them up to the surface. They live long enough, at least for 12 to 24 hours, to be able to run multiple experiments. And this has to do with some of the work into looking at innate immunity and how these corals respond to different types of bacteria, and just even basal respiration rates, because there's not much data on that for deep sea corals. So all of the collections that we're doing are often used for multiple reasons. So we'll also be able to identify the coral specifically and have a collection in hand for knowing what species it is for when we go back and do the analysis of the distributions of all the different types of corals. $485.
Yep.
Seven looks like it does something.
Yes, the rock skin from yesterday was very strange when it came to the surface. It's a very strange, like, consistency to it. So we preserved it. There's, I think, the closest thing we were able to say that it could possibly be is a benthic foraminiferan, but that's still kind of just a guess on our part, an educated guess, if you will.
What do you want from this one?
So we would like these bits that have the broken tops to them.
Okay.
So it's probably going to be coming down into here and clipping like as many of these branches as you can get. The parts that have, we want the live tissue on there, but
Live and.
yeah.
So when we use the term respiration, we're not thinking about respiration in terms of like breaths per minute like you would in mammals. This is cellular respiration, so how fast the metabolism rate is at the cellular rate, the breakdown of ATP and things like that. When we're talking about coral respiration, it's not breathing, it's how fast their metabolism works within their cells.
So, yeah.
Yeah, that's good.
We'll just look right that last little piece. Sits coming out.
And there is, yeah, like a pretty general understanding that deep sea corals have slow metabolisms and they grow very slowly, but we actually don't have a lot of, like I said, there's just not a lot of research about that metabolic rate. So some of this is just very foundational work that we have for a lot of shallow water species or terrestrial animals and plants that we just don't have for the deep sea. Is that funny, Cody?
That was hilarious. Love it. I'll probably just stay there. Let's go up to that big one. Out here. Those two. Get a little more zoom for you.
Yeah, that's great. Do you think that's enough, Brian? Do you want another piece?
Yeah, let's just get another
Okay,
piece.
let's go for one more.
Okay.
Yeah, that's great.
That's your point of one. Zoom in. Stop there.
There we go. Lovely. Did you make a note in the comments about the bare branch versus? Okay.
Ready, Freddie?
Yes,
Yep.
we are ready.
Let's go back to rock climbing.
On that rock.
8 and 6. Let's get to that nice vertical relief. Or what's Steve's word? Verticality? Never heard of that word before.
Still this intermix of brown and
Yeah, or black and
rocks that I don't have a really good explanation for.
maybe we can ask Steve to ping Kira or someone. Oh, she's on Okeanos, isn't she?
No, she's not.
Oh, that's later in
That's
the
later.
season. Yeah, sorry.
She's the next one. She's
You're
on
right.
This crew's next. Us both next.
Just help us get a better understanding of, why or how that happens.
Yeah.
Because
Tilt up on your side.
yes, sorry. You want me to?
No, I got it.
Okay.
Just so it can help me see what's coming
Well,
up as
yeah,
well.
and us.
About to climb.
About to be climbing. There was a couple comments and questions about the milk crates on the front of the ROV. I think it's just a matter of that it works well. They fit on the basket without having to make anything new. The ROV guys are more than welcome to give their 2 cents about it if they wish to. But my understanding is don't fix it if it ain't broken.
They're light.
They're light and they're
Yeah,
flexible.
They're bendy. That's super helpful.
Whole lot of oats. On a
Whole
small
lot of
pool.
oafs on that coral. We'll just take a quick look. Looks like Narella again, but there are a lot of these ophiocanthids, the spiny-armed brittle stars. A couple different kinds that have different oral discs. Is that us or is that the current?
That's us.
Okay.
Yeah. Have a little bit of roast action going on. Hold still.
That's cool. We can carry on. Lots of oafs up here on everything. Oh, Hamalid with the plexaurid. What's that?
Squid.
Yeah, that's a squid.
What's that nice place?
That's an octopus. Wow.
But any questions.
Yeah, we gotta follow that a little bit. It's gonna disappear into a cloud.
Yellow.
Nuts.
Is that an octopus, you guys?
We're trying to
Some
figure
kind
that
of
out.
cephalopod.
Where'd he go?
He's right there. He's
He's right
coming
there.
center screen.
Dude. Never seen that before.
Wow.
What the hell, people?
It's
Its
completely
tentacles are winged.
translucent. Wow, it's completely translucent.
That is the money
Oh.
shot of the whole trip for
octopuses.
viral.
Oh!
And he inked. Where did his ink stage? We
It's
didn't
in
see.
that little sack in the middle. It's gotta be
Maybe.
where that ink is.
Mike Vecchione, I hope
Oh,
you're
here
watching.
we go. We have its chromatophores are changing, so it's not as.
That will be it right there. That's gonna be.
Oh my goodness.
I think
It's
that
gonna be
might be
viral.
a fish he's digesting.
It sure looks like it, Brian.
What Brian said?
I think that silver thing in the mantle is actually a fish it's digesting.
That's what I
potentially.
was. Yeah.
That's insane.
But I'm not 100% sure of that yet. If that's the case, the only natural non-translucent tissues where it's the muscle bands around its tentacles and its eyes. Oh yeah.
I don't know if that's a fish.
I don't know either.
I don't think so. I don't know. Wow.
I'm gonna come out.
Oh my god. That'd be very unfortunate if he just got chopped up in the rotors.
Yeah, we try not to say stuff like that.
I'm sorry. That is insane.
Great, Cody.
Look at, you know, gosh, it's beautiful.
It is. I've never seen
This
an
is
octopus that is
this
this.
is different to me.
Steve says he's never seen anything like it either.
Cody's doing an amazing job of
Yeah.
keeping
Yes,
this in
you
view.
are.
I still think that's a fish.
You do?
I do. I see an eye.
I don't know.
I could still
That's
be
a
wrong.
cool
but
posture.
Keep zooming in.
Okay.
You're doing a good job. I'll just hold it in focus.
Yeah, perfect.
So you go wide a little bit.
Yep.
Keep moving down. Keep going. I'll just backwards.
Want me to come out a little bit?
Yeah, just come out just a little bit.
He's off to the right.
Oh, there we go. Cool. Yeah.
So we can get a really good video.
That's, does it have the same
Okay, go for zoom.
structure?
Okay.
Yeah, it does, so that would mean it's not a fish.
Sorry.
That's amazing.
No,
So-
the more I look at that, the less I do think it's a fish because it's
Can I-
symmetrically, whatever. What do
I'm
you?
trying to think about.
You're quick on the trigger.
I don't have. Do I have iris? Okay.
So kudos to our captain, who has very quickly figured out that it's, well, if it's not it, it's going to be very closely related to or very similar to Vitrella danella richardi, which was seen by Bruce Robeson from Mbari. In 2012, it was a very rare deepwater pelagic octopus that's completely translucent. He saw it in Phoenix Islands.
Oh, that's great.
And that internal structure is not a fish, if that's the case. Look,
What is it?
it's just part of the octopus. I don't know what it is, but it's exactly in the same position. It looks
Bruce
identical.
Robeson saw it. It's cool.
That's really cool.
Zoom in just a
Thank
little
you.
Yeah.
That's it.
Yeah, I should have snapped the names.
He's hanging out.
There you go.
Great job there, Alexis. Great
Thanks.
job.
And Cody. I
Great
mean, it's
job,
all
Cody. Really, really
I have to
professional.
imagine that this is, if it's been seen even a couple times, that this is going to be some of the best imagery of it.
Oh, definitely.
You can zoom in a little more. and try to
Okay.
get that his head right there. Perfect.
Oh my gosh. This is outstanding. Look at the eyes and how reflective they are around them.
That could be one of the coolest things
Go
I've
wide.
ever seen.
Go wide. Oops. In, out. There we go.
Perfect.
It has the connective tissue between its tentacles
Oh, it does.
too.
Look
Yeah.
at that. I didn't even see that.
We're out. Wow, Cody, that was amazing.
Excellent
Woo!
flying.
Amazing.
Wow. One little looks like one little like gelatinous. We're gonna be like, oh, that's nothing, and then this amazing octopus. That was awesome.
How we lost him?
Yeah, I think he's
Yeah,
gone on
I'll
his
let him
way.
go.
Yeah.
Yeah.
I'm just gonna dead stick it. So
Okay.
I see him come behind me or something.
Okay.
That was fantastic. What,
That was cool.
that was really great. You know, observations like this are just so hard to get, and I agree with you, Alexis. It's probably definitely some of the best footage out there.
Yeah. And I have a feeling that we'll make it back to Mike Vecchione, who is a cephalopod expert at the Smithsonian.
For sure.
And hopefully we can get, a little bit more information about it at some point. It was, I mean, we didn't have lasers on it, but I can't imagine it was.
It was tiny.
He was tiny.
Yeah.
Yeah, we had lasers
Oh,
on it first. So
okay.
We do have
Okay.
the video
Oh, good.
pictures of it. Yeah.
I'm sure he's not blocking.
That was fancy flying.
That was.
Glass
That was.
octopus.
I mean, we're so like used to Dumbos, but when we find a different type of octopus, we.
I don't think he's on me.
Yeah, I think you're.
Oh, no, he's hanging out with me.
Oh.
Right down there, yeah.
He's
You're
hanging,
manip
he's
cam.
hanging out with us.
Oh my gosh.
So for those who are watching at home, we're looking at one of our situational cams, and the
See that?
octopus is cruising with us just outside of the view of our main camera. You might see him here floating through in a second.
Oh,
So it
there it
looks
is.
They go from the
Goodbye.
mantle goes from 11 to 45 centimeters in adults.
11 to 45. Okay.
45, they get big.
Yeah.
He wants to be video.
Yeah.
What's the organ that you can see?
He's very curious about us.
We're actually not
It's found
sure.
worldwide in tropical and subtropical areas.
I would need to refresh on octopus anatomy in order to make a guess about what that organ on the inside is. It's very shiny, but I'm not sure.
Go for some.
Yeah.
Okay, good.
It seems to
He's chill.
become comfortable, yeah.
He's very chill.
Cool. All right. That's amazing.
Well, and you would imagine we came across it swimming close to the
Yeah.
benthos, but at the same time, with this translucent body plan, that it would spend most of its time in the pelagic zone, so up above the seafloor.
Well, the name glass octopus is certainly fitting.
Oh, is that the common name?
Yeah, that's
okay.
the common name. So there, believe it or not, there's a Wikipedia page on this.
Oh, wow.
A very small one, but it exists, so we can maybe add some of these photos to it. Yeah, the only picture on it is from what year was it?
So inside those lobes, I think are the gills, right? Inside those ones that look like feathers.
Maybe I'm wrong in misremembering. When it was first saw it, it almost seemed like the suckers and the tentacles were sort of a purplish, like we've seen some color change.
That's what it looked like at the beginning.
Yeah.
I noticed the suckers were purple.
Yeah, I did too.
And
So
now they're
cool.
orange, and look how docile it is now.
Yeah.
Not trying to run.
Well, and it did have like when I think it first felt our presence, there was a change in the chromatophore. So I didn't see any chromatophore coloration when we first came up. And you're right, the coloration around the suckers was different. And then now, even though it's hard to see, there are little spots of orange throughout the body plan. Wow, it's beautiful.
All day.
I know. We'll go a little bit longer, so we have hopefully a couple different shots of that are in long enough span. Wow.
Yeah, that makes sense. Just thinking about the behavior, this thing went from inking at us, right, so genuinely being scared, to now putting on a show. Do you have a size estimate?
It was very small. Like in the manip cam, it was like this big.
Oh, that's little.
Zoom out just a little bit.
Yep.
This is the rating C. Let's see if we can see some sort of. Zoom out some more.
Yep.
So most of the records of
I think I'm full wide.
are extremely rare, and only a few exist to date, mostly obtained from the stomachs of predators. I'm reading from the
Yeah.
paper. In spite of
He's
the
like,
more
oh,
than
that's why he's all
4,000
alone.
tectonic faults.
Wow.
Wow.
Golly.
But map of distribution is.
There's one dot. There's a light, so there's nothing.
Guys, this is the first record of this species in this area.
Oh, there's a little ink.
At least unless it's been
Yeah.
updated from a paper that was published in 2016. That's pretty cool. Going into Opus. And they identified this from a larva.
Whoa, there we go.
What's the digestive systems? So that silver thing that we're seeing in the middle
Yeah.
is actually the digestive system, so that is the gut. But it's not that is the characteristic
To have that, like-
color and shape to have that sheen. So it's not like a single fish in the gut, it's just the whole digestive system. You can see the gills on either side.
Yeah, that's what that was.
Which is pretty cool. Brain is actually a very small node, centrally located, sort of. Let's see if I can point it out here. This really nice diagram, but it's not well oriented. Let's see for me to point that out for you.
Ooh.
Funnel.
There's
Cool.
iridescent.
So the brain is basically just behind the mouth.
the
If that
lasers.
kind of gives you some orientation. It might be hard to see.
I don't see like
Moving
the
so
other thing
fast.
is that's interesting is I don't see like a clear beak. That's like the one part you know is solid in an octopus, and it doesn't seem to have like a large beak that we've been able to.
How big did you estimate he is?
Probably
With your fingers?
like about this big, so very small in reality, but being able to fly the ROV right next to it and then be able to zoom in is how we have such outstanding video of the behavior. You can see the siphon in the back of the mantle.
Let me
Which
read
is-
you a couple of choice lines from this paper. This glass octopus is considered to be one of the least studied cephalopods with circumglobal distribution. There you go.
All right.
Different life stages seem to inhabit different depths in the water column. Adults are usually found in the meso to bathypelagic zone, depths below 1,000 meters. But recently hatched paralarvae were caught during the day in the North Atlantic between 310 and 400 meters. So maybe the fact that this is small and it's 600 meters has something to do with its life stage as it migrates deeper.
Because you said the adults can get to the mantle is 44 inches. Oh, centimeters. I'm sorry.
Still, that's pretty big.
That's still, yeah,
Yeah.
much bigger than
And then-
this.
There's your beak.
Oh yeah.
Yeah, there
Yeah,
it
I'll
is.
show you that orangey bit. Yeah.
Ooh, it just like glittered.
And kind of other.
So basically, they spawn in the deep sea. Their larvae go up to about 300 meters, and then as they grow, they slowly sink. They kind of go to different zones as they get deeper and deeper.
Does it say what the object is inside that you can see?
So that,
That's their digestive.
yeah, that's the
The
digestive
digestive
system. And then when you, so there's some ink.
Oof.
I don't want you on me, dude.
So the long, like Randy said, that long silvery part in the middle is the digestive system, and then coming off of that into the mantle, there's two gills on either side, and then it uses that siphon to filter water up through the mantle and also to propel itself, and that's where the ink comes out of when we're.
He's more curious than we are. He has
Little
a whole of our
information
camera
about the
now.
beak.
Yeah, he's got our camera.
Whoa, that's cool.
The marks right here.
That is cool.
That's such a good mark.
What?
He smeared the lens.
He grabbed this.
Grabbed
Wow. A little information about the beak, because you were asking about it, Alexis.
Yeah.
So when they're young, and I'm just going to assume that this one is young because it's so small, they have a faint and less developed beak, which could be a sign of a diet on soft-bodied prey. And as they age and get bigger, their beak hardens. And the beaks, their teeth will disappear later, allowing crushing of hard parts of crustacean prey during a dietary shift as they age.
White spot?
The white spot between the eyes is the brain.
Yep, exactly.
Thanks,
Oh,
Dougal.
good. Dougal's here.
Yeah, that's what I was curious.
Never seen that ink before.
Never seen one in before.
People talk about that little white octopus as being the Casper
Casper,
octopus,
yeah.
but this is almost
Yeah.
more ghost-like.
It's true. But
Oh yeah, it's grabbing on.
yeah, it was only the first time we ever recorded the ghost octopus was on a cruise I was on in 2016, and they were deep though.
Yeah,
I don't know how
yeah.
deep it is, but it's the deepest inserate octopus we've ever seen.
Whoa, look at that.
That is
Whoa.
beautiful.
Wow. Oh yeah, look at the little beak.
Nope, 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 nope. Nope, nope, touch the field, buddy.
It's like,
Come with me.
it's just dancing for us.
Woo. It's totally putting on a show.
Or
Slash getting thrown around by the
slash
Slash a bit.
enjoying the change in water currents.
No.
Randy, it's your call when we
No.
move on.
Yeah, I'm just gonna keep flying until.
I think let's just take another minute. 'cause
Okay.
This is pretty stunning.
It
And,
is.
Wanna come into him a little?
I think this behavior, right, is really interesting.
Yeah, it is. Well, and just to see the different orientations, you get you know, inside the bell as well as on the side from the top of the mantle.
And I do think there's, well, either it's that our lighting is changing or they're seeing changes in, changing in chromophores and chromatophores. I mean, it sounds like this is a rare enough observation that we should take the time we're given and really enjoy it.
He is
What?
really, really curious, and he keeps trying to jitter into us like this right here, then grab on us. Right now.
Wow. It's really
Whoa, that was
That's
cool.
amazing.
Keep
What's
pushing him
amazing
away.
is that he's going for the camera, right? And now a different part of the sub.
Not that.
Push him away. It might be on us.
And he's just gonna
There you go.
go for a ride, yeah.
He was on us. Away, buddy.
Tim's camera footage will be really good from this too.
Oh, that would be interesting to see, yeah.
Alright.
All right.
He's gonna meet us halfway.
Okay.
He's coming right back towards us.
I'm gonna put a.
I feel like this little octopus is gonna chase us the rest of us.
He might.
I think we're. I'm gonna. Can you click for a minute?
Yeah. Does it say anything in that article, Randy, about their reproduction? Like, do they?
It did. Hang on.
I'm just curious if they lay eggs also, or if it's a different. Wow, look at that. Oh, cool. It's like in a little.
Go wide again?
I think I am one. Oh no, I wasn't. Sorry.
That was me that zoomed in on the bit.
Oh, okay.
Something.
That is. I can't wait to see J-Rod's like.
The beautified shot.
Yeah. It's got its tentacles all scratched up now.
Actually, it doesn't really. It just sort of talks about the different life stages. Probably because a lot of the records of the species
Right.
come from plankton.
Oh,
So
okay.
there are very few. This is by far. I mean, just this paper was published in 2016. So unless there's something more recent that we haven't looked at, there really isn't much adult footage. There's a couple of pictures that were seen from the Cocos Islands.
Uh-huh.
But this is
Go ahead, zoom
really
in.
a rare observation, at least from what we can quickly glean from the internet. But that said, we know that they're pantropical, so they're showing
Okay.
up in plankton samples all over the world, and there's sightings from all over. But the note that was published in 2016 was sort of the first observation from the Arabian Sea, and so and they found just a very small one in the plankton, and so that's how noteworthy it was. So I imagine that this footage will be extremely useful to anybody interested in glass squid behavior and really good. I don't know if this is a full-grown adult. Again, I doubt it, but I think, but just looking at something beyond the larval stage, right? We are at 650.5 meters
Yeah.
down. Just to say this out loud, the temperature is 6.6 degrees C, and we are hanging out on an unnamed, unknown seamount in the Winslow Reef complex, just at the very southern tip of the U.S. EEZ border.
Oh, that's cool.
Pretty
You can
shallow
really see
VI.
into the.
Yep. That, I think this is a really rare observation. So,
Cool.
yeah. All right. I
We
think
can let-
he's drifting off. I
Yes.
think we should let him go. That was really incredible. So I'm so glad we were able to get that, and that's just incredible.
Oops, sorry, Cody. Full
So-
wide.
We will let this octopus do its octopus thing, see we're off into the abyss. shoots off into the abyss, and we're back to the benthic.
Yes, that was amazing.
That was completely amazing.
Does that give you a rush air?
That was the coolest thing I've ever seen.
It was
There
really
you
cool.
go. So cool.
I remember that. Remember that little translucent. Was it an octopus or a squid that we saw in Pippa?
That was a squid.
That
It
was
was
a
a
squid.
squid.
That was
glass
amazing.
squid. Yeah. That was cool.
Oh, glass squid and glass octopus. Glass sponges. My goodness.
Yeah.
Fish.
Oh, stop, Cody.
That will for
Cody's
sure be
making
a
fun of
fun video to pull out as a highlight. There's plenty of footage there to
Yeah,
be
for
able
sure.
to stitch a nice little highlight together.
That was fancy flying, boys.
That
So
was
good.
really well done. Golly.
Do your like shoulders get tense when you have to do stuff like that, or are you just chill as a cucumber?
Trying to chill, and then when he kept coming to me, I was a little nervous.
Yeah, you were great though.
That was so good.
Cool whether they would send you video.
I want to give a huge shout out to our ship's captain who pulled the paper out in 13 seconds. That was pretty amazing. So huge shout out to Captain Al. I don't even have my computer with me in front of me, but he was able to find and identify that immediately. So great.
Gotcha.
And now, and look at where this octopus has taken us. Look at this stunning view of the landscape that we get.
Hawaii, something over there, and then Australia and New Zealand.
Okay, now
And
we
then
gotta
of
make
those
some
microplankton
some ground. We got to play, and now it's time to work.
You know, one thing I'll just add, just to say, is that we have so many plankton
Do
samples
we?
from this area.
Okay.
We have a whole lot of the cephalopods
Oh, way to go!
pulled out. I bet you we have I wouldn't be surprised if we already have one in our plankton samples. There were a ton of things that looked like this. Cool.
So it's gonna slowly come
A
up.
ton, by a handful. Neat.
I have you on station.
We'll have to tag Kelsey.
I was trying to work towards
Phenomenal.
the left side.
Yeah, just to keep the ship there.
Yeah, because he's over the top
Great,
of the
thank you.
Almost there. Because we still got
Original description, 1930.
till we're there,
Oh wow!
roughly.
Yep.
From plankton.
I can't tell, it just says original description.
I just can't imagine how they would have had.
I'm sure it was,
Right.
or it could have been dragged up in something.
1918. Obus has got the original description in 1918.
Oh.
Well.
Back
Did
to
you take a
looking
bunch of beauty
for
shots, Brian?
corals and sponges.
Totally forgot.
Wow.
That was sarcasm.
Oh. Really?
Yeah.
I really can't tell.
Look at all the
Brittle
Brittle
stars?
stars?
Yeah. Just all in these rocks.
picked up. Looks like we picked up a new species of sponge, encrusting sponge, on this wall too.
Oh.
Discovery, wow, you can get the original.
Is that the name you've been looking up?
No, we picked up a different one.
Cambridge University Press. So maybe the
Can we
first
take a
paper
look
on
at
it was 1930.
like one of these
Maybe.
white shells
I may have been
that's
looking at
on
a
here?
different member of the.
Thank you.
A different species name thing.
Can you scroll down?
Dougal put in is different than the
Or
one.
not populating.
Oh, no, that's a regular.
Oh, sorry. Oh.
No.
That's the Opus record. Described it.
Duh, I didn't really. Let's see what we have here. Well, there's sponge growing over. Are these brachiopods?
I was just thinking that, is it? Look at about the spikes on the shell? Is that
Well, so I think that's sponge overgrow. Oh, that's that's sponge overgrowing
okay?
an old shell, right?
I think so. I don't know. It looks very weird to me.
But we have other.
But there's another one down there. See?
Yeah. There's
That's
also one
sickles
here.
here.
Little scale worm right next to it.
Brenda, since you're here, you want to go ahead and, if you don't mind logging in on you first? Or?
Yeah, go ahead.
There are little spikes on there.
I don't know what. Hang on. Let me look it up. But that's great.
Scan over here.
Oh my goodness. Using someone else's computer, their keyboards are always just a little different, you know?
Yeah, that happens to me with his computer too.
Hang on. Let me just look up brachiopod shells on Google Images here.
There's lots of old.
I mean, brachiopod reefs used to be. Yeah, I guess some of them do have. There's a lot of different types. I can't quite figure out how to scroll on his computer, but it looks like some of them have that ridginess.
Yeah.
I don't.
I've never been great at determining brachiopod versus like clam, so.
It's a straight up clam. But.
There's a little tube. There's lots of old tubes from tube worms in there too.
They do still exist, but they were much more common as
Yeah.
fossil reefs way back when, and people thought they were extinct for a long time. and
Right.
Just recently,
I think we can keep
relatively
going.
recently, discovered that they were still alive and well in parts of the deep sea. But it's really a very different thing. Oh, my timer for the octopus just went off.
Wow, that was a long 3 minutes.
No, it went off before. It was just
Oh,
going off
okay.
and going off and going off, and I didn't feel it vibrating. That was really cool. So let's see.
Yeah, we're starting up this more steep part of the feature now. Working up to, I think our shallowest point is around 200 meters.
Yep, and actually our shallowest point, if we want, we should try to go to the top of it. It might be off a little bit off the map, because the map wasn't made at the top, shallowest part.
Okay.
But what station, where are we? Waypoint 8?
Yeah.
1, 2, 3, 4, 5, 6, 7, 8, 9, 10.
Yeah, so we're 1, 2, 3, 4, 5, 6, 7. We did 7. We're not quite up to 8.
We're very close.
Yeah.
So I think we'll go to the final and then a little bit just until we're sure we're on the top.
Okay.
There's another good example of
Yeah.
the clam or
Can we
brachiopod.
look at that on the left, Adam?
Yeah,
That
the,
white circle, yeah. Just take a look at one and see if we can get a better image of it.
I love the structure here.
Thanks.
Yeah, it's the same. I don't think there was a sponge overgrowing it before. I think
Oh,
that's
really?
just the. Yeah, I
Okay.
think it's the maybe it might be the mantle, right? A lot of.
Do you want me to look down? Or?
Yeah, you go.
Okay.
So you look at that, I think it's the same.
Yeah, you're right. Oh, yeah.
Cool. Well, hi, Alexis.
Hi.
It's like the first time I've been at the controls with you in a while.
Yeah.
It's fun.
There we go.
Take a look at that. That looks almost like a giant clam edge.
Oh yeah, a clam fossil.
Yeah. maybe.
I don't know, this is weird to me. I mean, I know mantles can like come out, but
Yeah.
that seems really thick. Okay, let's keep going. Yes, sir. Yeah, we've really hit a different terrain from what we were on right before we found that octopus.
It led us to new places.
It did.
Look at all this sort of crusty, thin.
Well, it's like we were seeing a lot of like this really dark looking crusted rock, but also very exposed carbonate. So Brian and I were saying we really do need a geologist perspective as to like why you have both of those at the same time.
Kira just sent me an email.
Oh, really?
Not about that, but about other things. So I intend to write back and ask her exactly that.
Yeah. Or like yeah. It's just a strange, to me it just seems strange to have all that dark coated rock at the same time as the exposed carbonate.
Well, if only we knew someone who worked for the U.S. Geological Survey who
Well,
might have some contacts.
I technically, yeah. I just don't have context.
Yet. You will. Look at that.
That's
how
cool,
flat that
yeah.
is. It's like
A slab.
Can we get a
Can
look
we-
at that?
It is, and look at next, like this. It looks very, like
We don't have a geology button. Yes, we do.
flowish. Yeah, we do.
Okay.
That looks like a countertop.
Wanna take a piece of it?
I'd like to redo my kitchen, and I would like deep sea slab, please.
It looks like we have kind of this strange
Yeah, it is really weird.
rock morphology kind of as we go up too, so we can kind of scan around. Yeah, it's very like, it looks fresher to me, but again, I have very little geology context.
Well, it's kind of like, you know, when you see hoodoos.
When you
get
see,
the, you know what a hoodoo is?
no,
Hoodoo is like, kind of like a sandstone below and then like a slab of like hard rock on top. And
Okay.
So the rock protects it and all
Oh.
erodes, so you get these like pinnacles of
that's
rock.
what that looks like. You're right.
Hoodoo? It's called a hoodoo. It's like, kind of looks like a mushroom of rock.
So texturized.
I mean, I was just thinking
Wow,
about that.
look
I
at
was thinking
this.
about, well, when it's in the shallows, it makes
Wow.
total sense. And then as it falls down, you just
This
you have to
is
think
so
about
angular.
the trauma that these, right?
Yeah.
But this is a pretty place to hang out.
Let's just get a
I-
good view of what it looks like.
Yeah.
It's really great. You get a,
It's a massive wall.
Oh, look at all those stars on that overhang. I'm not seeing much coral or sponge colonization, but there's
You can both directions.
definitely other invertebrates living
What you think?
among all these crevices.
so when we talk about carbonate caps on the top of underwater volcanoes, this is really what this is. And the carbonate cap is built by calcifying organisms that, you know, when this used to be, well, it's already still pretty shallow, but when it used to be even shallower, however many years ago. So these seamounts are all 80 to 90 million years old. Sea levels changed about, I don't know, 18,000 years ago, I think. And so with all of that, and then there's subsidence of the seamounts that keep going down. And so things, what used to be shallow keeps going deeper. But what you're looking at is remnants of an ancient reef. And it is these volcanoes, these underwater volcanoes sort of form these seamounts. And then when they're shallow enough, you have photosynthetic calcifying life, which creates these limestone calcium carbonate structures. And then as it subsides and sinks deeper, these structures go deeper down. And yes, there's still, you know, precipitate coming out of the seawater, which is what gives you these crusts. But I wouldn't expect it to be uniform, given that the calcium carbonate isn't uniform, right? The geology here is really different. They were made by a variety of different organisms at different ages and stages. And it may be kind of intertwined with the volcanic rock because the animals might have set up really differently on different parts of the volcanic base, the basalt's base, at different stages in this seamount's history. So I'm not surprised, but I don't understand the details of how that happens. But these carbonate caps are really typical in a lot of these seamounts that are shallow enough to have been shallow once, if that makes any sense.
This is very like
demo sponge.
Yeah, lots of like flat layers.
Yeah, look at this one.
Yeah.
This is ophiroid heaven, though, it looks like.
Okay, I cannot figure out how to use this computer. Oof, terrible at it. I got it. Scrolling is even hard. What I don't understand is that I guess it's iron oxide.
Yeah.
It's like a reddish tint, but maybe that's just the camera.
No, I don't think it is. I think that's real.
You think it's real?
Yeah. All these ophiroids with their arms out, catching particles in the water. Wow, just keeps going straight up, huh?
It's like a massive wall. I'm gonna back
It's a
up
wall
and
of
see
Yeah, go
stars.
ahead,
a picture
for
so you
sure.
can see something you want to have a look at. Maybe a big sponge coming up going to the left. Above there. Oh, there's something.
Oh, there's a coral.
There it is.
Hello.
Yeah.
Looks like they continue to.
A couple of them. See, there's another one behind it.
Mm-hmm.
They always seem to set up right before you get a turn.
Yeah, I think it's like the way that the water moves around
Mm-hmm.
the edge of a feature. Similar to finding them on an overhang. The water is going to move more quickly around that.
Makes a ton of sense.
Some type of primnoid.
Do you see? I see associates, but I can't see what kind. Do you have any? Do you mind if I turn the ISO down a little?
Way. I think,
Are you looking this way, Ryder?
I just wanted to turn that down a little bit. Astroschema
they are some type of.
and a little.
This
crab.
is actually different. This might be the Christmas oaf, which we would be interested in collecting.
Want me to line up there?
Yeah, would you?
Yeah, just point out where you want to collect there so we can set up.
Right here. This guy. Is Tim sleeping?
I hope so.
Okay. Me too. I just was gonna, if he wasn't, I was gonna.
I'm gonna. Yeah. Let's just.
Yeah.
Oh.
And I don't think we've been seeing this primnoid. I can't put my finger on what the genus is, but it's definitely not quite the same as we've been seeing.
It's different. So for those of you just joining us, this is the first of a 3 or 4 dive series on this same seamount. We are in for a 48-hour dive slam, basically, with 2-hour breaks in between. So you'll hear all of us on at very different sleep-deprived times, although everyone's going to try to get their rest. So this is a dive. We're going to go up multiple sides of the same seamount and check it out. And so it'll be super interesting to see if what this looks like on the other side.
Yeah, especially with the carbonate cap to see if there's different formations on various sides. It's up in here.
Thank you.
Yeah.
Like, how do I even look? Steve says he doesn't need any. It's Paracalyptrophora hawaiiensis. Thank you, Steve.
Oh, I'm sorry. I moved before you gave me Yeah, Dive Slam 2020, 2021. That's about right. It's going to be a long but also short 48 hours. Often that kind of round-the-clock work ends up going by fast, even though it's exhausting.
How do I? Come on. Got it. Paracalyptrophora hawaiiensis. Scroll. I am.
Oh, I know that part actually really annoys me. That when you're entering something in, you then have to scroll
Yeah,
down
yeah.
and able. Yeah.
I don't know what we're getting wows to. The coral, our
Are we
dive
good,
slam.
Adam?
I think it's our dive slam. That's a great observation.
Oh, there's a bunch of little trirostylodes in there too.
Yeah, there's a couple more over there, but I didn't see the ones over here until you just got in.
Yeah, this is something that from the Phoenix Islands Protected Area Cruise in 2017, we were calling the Christmas ophoroid, just because it looks like a peppermint. Yeah, peppermint stick. And I don't think we've collected this one or seen it on this expedition yet, so it is definitely going to be of interest for collection.
Can you zoom in again? Sorry, one
Yeah.
more time for a frame grab. Thank you. Helpful.
And then we're gonna just go around the colony and look at how many little squat
1, 2,
lobsters
3,
there are,
4,
because there's a ton.
5.
1, 2, 3, 4, 5.
6
or
7
7.
8.
Oh yeah, they're all over. It's kind of unusual. You normally see a couple of them, but not
Yeah.
over into double digits.
No, that's a lot. 9.
There's one in there.
That's 9. Yeah.
There's one down
I got that one
there.
already. Sorry, I'm just.
It's okay.
I've got this going. 10.
Yep. There's one here,
11,
here.
12, 13. Okay, that's a lot. So for those of you asking, yes, the sub will come up in between. We'll have 2 to 3 hour turnarounds and then back down in the water every time. So let me just mark down the fact that there are 13.
Well, since there is a little crab down here too, if we can cut right around here, so we get the ophoroid and the crab and the coral.
Okay.
Does that work for you?
Yeah.
Okay.
Okay.
Do you want to go eat?
Oh yeah, sorry.
Brian's behind you, so yeah, I just figured.
Dozing in. Okay. Do you want to eat, Alexis?
No, go ahead. I'll eat. Well, I mean, I will need to, but
I'll send somebody up.
yeah.
Yeah, I was just thinking. This way.
bye, little crab.
Wow.
That's okay.
Tim sheds a tiny tear.
That's okay. We get the Christmas elf, which is like a Christmas miracle. This will be 487. Yeah. Oh yeah, there's another crab. As long as you get the piece with the ophroid on there, then we're good. The other stuff would just be extra. Nice, that's perfect, Cody. That is Cody, correct. Do you see all the tiny little corallivories on there?
I saw a couple, but not.
There's like 20.
Wow.
We want more for the associates, so yeah.
I don't even want any at all.
Oh, can I go eat when they?
Yeah,
Okay.
you wanna go?
Yeah. If you don't mind, have you eaten?
Yeah.
Okay.
We did one more, right?
No, that's perfect. Thank you.
487 in
We got a credit
Quiverful.
now.
Yes, we did. You get bonus points for 2 associates.
I thought you all were still up here looking at the octopus.
I mean, would you blame us? It's you.
Going back for more, or is that it?
We're done.
Okay. Q4. These are flip flopped. 4, 5.
You have to move on?
Yes.
Okay. You're good. What's up? The you chicken.
Looks like this is where all that black manganese coated rock is
It's
from.
gone from falling off, yeah, I agree.
I wonder why it's stuck here. A little confused.
Yeah, I have to admit, Alexis and I were talking about that earlier, is the geology here is a little confusing to me.
There was a paper I read about crust precipitation rates being higher in or around areas of lower oxygen. And I don't know, what are we looking at for oxygen in this area? Do you guys have an action oxygen number?
It's,
Or
Oxygen
Gravana?
level is 88.8.
Pretty low for the water column around here. Maybe. I'm not sure. Could be some sort of just rock that was embedded in the carbonate at one point. Just,
Yeah.
It's just dissolved away. Or eroded away somehow.
You, Jimbo.
those pillars.
Yeah.
Not a lot of rubble though. I'm very, very surprised there's not much rubble at this site.
You mean coral rubble?
Yeah, rubble for the stony corals.
Yeah.
None of those long, large fields. Look at that. It's like a pedestal.
Yeah.
Can I zoom in on that?
Differential.
Can I zoom in on the pedestal with the rock?
Yeah, differential weathering with the limestone dissolving out from under the encrusted maybe basalt or whatever it is.
Yeah. It's like a bizarre. What are those brachiopods or bivalves? It's kind of a neat feature.
Yeah.
Okay. All set.
I'm really surprised at how undercut so many of these rocks are. But look, you can see that rock embedded still. That
Yeah.
black rock.
Yeah, I would love to know if this is like some sort of tectonic uplift, or I don't see a mechanism for it, or it's just stuff that's fallen down from.
Yeah, I don't have a good explanation for why you'd have all this volcanic rock embedded in the limestone. Actually, I do. Think about it. If you've got volcanic spires eroding, the erosion falls down into the reef, and then it gets lithified inside the limestone. So you probably had harder volcanic rock.
Yeah. So there
mass wasting down into the reef, the back reef, or the central lagoon, and then that gets lithified and incorporated into the coral matrix.
Yeah.
That's what that is.
Yeah.
So this was at one point a high island. We're seeing the part of geological time where this was a high island before it probably became an atoll, and then before it subsided.
Yeah.
Is what we're moving through here.
I'll buy that. I think our second rock sample, this is what we got. Some of this stuff that had fallen down is very loose. This is definitely something that I have never seen before.
This is very similar to the terrain we saw at Baker.
Really? Baker this expedition?
The Baker this expedition, yeah. There's another coral and an overhang.
Maybe I was sleeping.
Yeah, Baker Shallow was different, didn't have the same kind of inclusive volcanic rocks in here, but these overhang erosional limestone features.
This stuff, but
that's what I meant. Not the included volcanic
No, this is
rocks.
definitely Howland and Baker, yeah. I love these pedestals though. Very, very neat.
I was gonna turn the windows on. And then that way when I get close, I'll turn them on. Look at that.
Did you see the freshly cracked limestone earlier? So down about 1,000 meters, there was a block that had broken away very recently. Like it was stark white, no encrusting, anything. And you could see the reef through a cross section.
Oh wow, no, I didn't see that. I'll look at the video later.
Yeah. Yeah, it was definitely a big breakage sometime in
Geologically recent past.
No, probably less than 10 years.
Yeah.
Maybe even more recent, who knows, I don't know. Just didn't see any hydroids, no nothing like that to colonize that surface. It's clean.
Look at that one, just hanging out.
Yeah. That's where they're coming from. That's why a lot of times when geologists want us to find rocks that date a place, they want in-place stuff.
Totally.
Because this stuff is all falling down at one point or another.
No interest?
No, I think it's the same one we collected. Eucalyptriflora hawaiiensis. We have an abundance of material from 2017. It's one of the most collected corals.
That's an interesting growth form. That swoop.
Yeah. Sort of current effect. This is interesting because these are the first colonies that I've seen of this species, the whole cruise. And it was one of the most abundant crinoids from 2017. So
It's the green stuff.
yeah, can we look at the green stuff on the left side? 9 o'clock?
Sponge.
Yeah, I agree, but that's definitely a new sponge for this dive.
You wanna stop and have a real good look or just a quick look?
Probably just a quick look's fine.
Crusting sponge.
It's probably good.
All right. Saw one of these brittle stars swimming earlier.
Nice. That's always fun.
They make some good traction in the midwater.
I'm surprised how fast they can be both in the water column and on the seafloor.
Wow. I'm surprised a lot of these corals can settle on this stuff. You know, depending on how fast they grow, I would imagine this stuff is probably exposed for a decent amount of time, but not as stable as some of the larger carbonate blocks in the area. But maybe
Yeah, absolutely.
maybe it's just an area with a lot of dissolution or
Yeah,
erosion.
I feel like this, from an ecology standpoint, I feel like what you see here in terms of those kind of turnover would be similar to the Atlantic canyons, where you've got those serious mass wasting along the canyon features of the U.S. East Coast.
Yeah. I was drawing a similarity before with the canyon walls of the Mona Canyon in the Caribbean.
Yeah.
Where it's ideal for coral habitat, but there's just no corals because the substrate is so unstable. It's falling down in sheets.
Slack for slow down.
I didn't see what she's talking about. So.
I didn't see any corallivory. Couple of hermit crabs. Korea, probably, I don't know. There's a couple of different species in this area. Ophiuroids. Oviroids for sure have changed.
We got a good look. Is this an Apsamia?
Yeah, probably. Yeah. Yeah, the Anelopsamia will stick with us for up in the 300-400 meter range, and then it kind of disappears. That's a sea star down there, but it's not on the coral.
Yeah.
Updated with conversion.
Looks like the little peak of this rock here in the middle has got a decent
that?
number. Oh, just J-Rock, you want to fly up the center rock?
The center rock, roger that.
Hey guys.
Hello.
It's not paid.
Yeah, so, yeah, so, okay.
Are these all the same Paracalyptrophora hawaiiensis?
All the same. Yep. This is the depth range. You typically find them, they just explode. I was noting we didn't really see. I didn't catch every minute of the dive at Helena Baker, but I didn't see any of these. We saw them in 2017, but just not on our transects this year.
We saw one this year because you pointed it out somewhere.
We saw Paracalyptrophora, but not hawaiiensis.
I thought you did point it out. Maybe not.
It's not hawaiiensis.
Okay,
It's,
fair enough.
It's definitely in this shallower depth range. We saw Paracalyptrophora a bit deeper, but it was a different species.
So could you guys clarify something for me? Because I bet if anyone in this cruise really knows the details, it's you. Which is the brachiopod story about sort of when people thought when they were first rediscovered, because people thought they were extinct for a while, right? And then people figured out they weren't, and then everything that we're seeing that's been alive and white, a couple little nubbies, and those living brachiopods. What do we have here? Do you know?
I don't know the backstory behind the brachiopods, but I mean, this is the depth we typically find them, up here to a couple hundred meters.
Do you know?
I don't think they're well sampled.
I don't know anything about that one.
Well, everyone wants to sample, but I think they were thought to be extinct for a really long time. People, when they started doing deep sea exploration, started finding them alive. But I don't know the details of.
I mean, you can find them in some scuba diving depths, in the North Atlantic, for example.
Maybe it was just out here that I was reading that then. Scuba diving depths, like 120 feet?
Deeper end, so like technical diving. Not super shallow. We've seen them in the Gulf of Maine in the 100 to 150 meter range, sometimes a bit shallower.
Got it.
That's a large crab.
That is.
Ah, Tim, here's your mushroom crab. If he's listening. This is one crab Tim and I have been like, nobody knows what it is. And it's stuck in a crevice, of course. Yeah, so this is the one crab we saw on the Tokelau Ridge at one site in 2017, as well as on the EX. And just like, nobody knows what this is. It's a lithodid for sure, but it's definitely a bizarre, kind of looks like a big mushroom cap. lots of spines.
Looks remarkably similar to coconut crab.
Yeah.
You find on land around here.
Are coconut crabs related to hermit crabs?
They are.
Yeah. Yeah. So then this group of lithodid, this family, are within a group that the hermit crabs, or not hermit crabs, but crabs of that body plan
Yeah.
are. Yeah. Enumerates, I think they're called.
Right, so not a deck bug.
Yeah.
Oreos.
Yeah, this is interesting. You know, it's the corals are mostly on the hard basaltic with air quotes.
The black rock.
Black rock, yeah. Who knows?
Well, the black rock is clearly harder because you can
Yeah.
tell from the differential weathering that the rock is more resistant to, or the black rock is more resistant to dissolution or whatever.
It makes me think that this carbonate, you know, might not have been like actual, you know, solid reef, but rather, you know, maybe sediment that was
Consolidated.
loose
Sure.
density.
By that.
Looks like a
reef.
climbing wall.
This looks like a good way to die climbing with this stuff is so loose.
But you're right, I think it sort of is sedimented rather than accreted. Little corals, they're so small. Do you have lasers on? I don't see them, but maybe I'm just
Take the lasers on, please.
What's that?
Take the lasers on, please.
Black
Lasers
fairy.
coming on.
No, the sheet I gave you was all the way.
Yeah,
All the sheets are here.
I guess the Enolapsemia, they do form larger colonies, and we did see them. Rona, for example, had massive Enolapsemia, but here it's mostly Madrepora dominated, reefy type, communities that are shedding a lot of this debris and rubble. That's kind of a striking difference. This is actually more similar to what we saw at Jarvis, smaller Enolapsemia but larger Madrepora, but sometimes you end up with these mixed assemblages, which is what we saw a little bit deeper.
Oh, really? They were growing together?
Yeah, I mean, I wouldn't say, it was like starting to form a matrix, but they were definitely growing in the same vicinity, same proximity.
Do they ever hybridize?
I don't think so. I think they're separate families.
Okay.
So yeah, they're pretty distant.
Looks like at least 2, maybe 3 species of sponges here.
It's a popular rock.
Are those little white stylastrids or are those
Stylastrids, yeah.
Stylaster sponge rock.
I'm gonna zoom on the stylastrids if we have a minute.
Cool.
You want to have a stable?
Yeah, they're smaller.
You get the camera center back.
Sure.
Inside?
Yeah, the right side seems to have larger ones, although I think any side would work.
Found anything good?
Rocks with a bunch of stuff. I just wanted to get a zoom on the stylastrids here.
Cool.
And then it's all yours. generally alive, kind of nondescript. They don't have polyps like scleractinians, but
Here in
oh,
manual
that's
focus.
full zoom. Those are neat. Yeah, I don't think we have anything like that.
Wanna collect it?
Can I slurp it?
Yeah.
Okay. Can we slurp just a few of those?
Sure.
I'm just keeping the seat warm.
Special there, huh? That was fun.
So this is Stylaster species FK21488.
Four is open, correct?
4 is open on my sheet, yeah. Hey, that jelly ended up in jar 2. I don't know if you saw.
It did. Yeah.
You got it. It was just stuck on the lid apparently.
Ready?
I forgot to do an in-situ shot. I guess you could just do it here. That's fine.
This is waiting for the zoom in.
Yeah. And just one more, I think, is fine. Look at all those ophiuroids.
Oh wow.
Yeah. Yeah, it's plenty. What's more stubborn, crabs or stylastrids? Getting out of the tube.
Well, there we go.
It's the wrist flick maneuver.
488, right?
488. Yeah.
Jar 4?
Jar 4.
Do you want a few more, or are you happy with that?
I think that's fine. Yeah.
Okay.
That's great. All right. I think I've hijacked the chair long enough.
Thanks for letting me eat dinner.
Sir?
Already up to 500 meters.
Let's find some CCA.
Moving on up.
She said no point on it. Let's get to
Let's
business.
get to business. No, I'm just impressed at how far we already have.
Something else in Austria.
What?
Or we would have said something different in Austria.
Really, either. I think I consider it.
That's funny.
They have
Are these the same little white things Randy and I were looking at?
That might be brachiopods.
Yeah, that we don't know.
Do that in a couple hours.
Is that what you would like to look at a little bit more, or
Yes, I'll just once I.
the one underneath that ledge right there?
Sure, yeah.
Or the one off to the left?
Under the ledge is good if we can do that. Sorry, wasn't very clear. Well, it wasn't very clear about what I wanted. See, they have this really like this one looks like a shell, right?
Yep.
This one though, a couple of them have looked like this, where they're like very. It looks like to me like they're covered in sponge, but maybe it's just their tissue coming out, like their mantle.
I got nothing on that one.
Yeah, me neither. Okay. Let's just quickly get a real close in on these.
That's you and me?
Cool.
Sponge just grow.
Well, that's what I don't know if it's sponge or if it's like the tissue of the actual animal.
There's a little window right there.
Window.
Oh,
Oh, yeah. I don't know. I don't think so. No.
Probably, probably get some monkeys, close to those coral and algae up there.
Ready to go?
Let's keep going.
Let's turn them sirens on.
There goes that stick.
Shark.
Oh, shark.
Shark.
Oh, goodbye, shark. It went fast, though. I think it.
You do have a science species thing.
I've heard.
Well, you don't see it.
Isn't it over there somewhere?
No.
Oh, no.
It's in the office.
Is it for.
It's got two ends.
But I might be spreading some rumors to him about the period.
Getting into the
Oh, yes!
polychaete realm again.
Oh my gosh. Now I kind of want to collect one of those again.
I wouldn't be opposed.
Okay. Let me know. Let's see if next one we see. I mean,
Try and get the whole
yeah, we
worm
will.
out.
Yeah, right. We'll collect the whole tube again like we did in a quiver. Unless those are the only two in the whole vicinity.
They were so common on Baker.
Right. Lots of like rocks sticking out off of this. Do we have any interest in more water other than just like an off bottom?
Not that I have.
I think Steve is trying to conserve.
His filters.
Yeah.
How many have we shot by?
Two. Look at those brittle stars just.
Yeah.
Did you see the one swimming earlier?
I didn't. Steve was telling me about it, but no, I missed it.
It was cool. It was like very high up in the water column. Oh, fish.
Starting to pick up the diversity of fishes.
Oh, can we take a look? I think it has a parasite on it. Oh, maybe not. Maybe it's just got different colors. Oh no, just different coloration.
Stabbed an eye. Oh, he's hurting now.
Fish urchin interaction.
He got stabbed by an urchin.
We can leave him alone to recover.
First up. So much going on in this one
Oh,
scene.
let's
wait,
look in
wait,
there.
it's
Yeah,
the
yeah.
Matapod. Let's look at him.
Oh, yeah.
It's a mantis shrimp.
Oh, wow. Can you zoom in on that?
We're,
I saw one of these in Pippa too, remember that? Alexis?
yes, I do.
In almost the same habitat.
Yeah, the one in Pippa was deep though. It was
I know,
deeper than this.
It was like 700 meters or 800 meters. This is much shallower. Question is, do you think he sees us?
Who knows? Can't they just see everything?
Oh.
Pippa wasn't reacting to light at all.
So there's a lot of conversation, and so this is a mantis shrimp, and there's a lot of research about their vision because they have, I forget how many different types of visual receptors, but way more than humans do.
Yeah.
And so biochemically they should be able to see a whole bunch of different colors, but when you put them through kind of light color response tests, they fail miserably. And so no one's quite sure what the reason is evolutionarily for why they have so many different color receptors.
No response.
That was eerie.
Those things look like sort of teeth coming under it. Those things. Looks
Yeah,
like a scary
and
movie.
you've got like breaks.
Was that?
You've got like breaks on his arms.
No, the thing's coming forward.
Yeah, I saw that. Cool. They like to live in those little small caves though. That's what we saw in the Phoenix Islands protected area. It was a similar kind of just sitting and waiting.
What was on the roof of this cave? Was that a little bit of sponge or something?
Let's see when the lights come back on.
It's funny watching the autofocus try and figure out what to do with the
Oh.
lasers.
Whoa.
Yeah, the pale thing.
Totally
Yeah,
outrageous.
sponge.
Looks like some encrusting sponge.
And there's little shrimp living in there too with them, so it must not be
That's a great
interested in.
shrimp.
But they're pretty much like ambush predators, right? So they'll just sit and wait until
I believe so.
something comes along and then they'll snatch it.
Look at the structure on the right side there. It almost looks like a tube, sediment tube has been built into against the rock.
Yeah.
It's kind of, they've got one of those powerful punches.
Oh, right here, you mean?
Right
Or
there.
there?
Mechanical
Both sides,
punching is
actually.
incredible.
Yeah.
I like how he's like perched.
He's totally not.
He's
fussed
like
about it.
sideways.
I don't know. Does not change his stance one little bit.
Nope.
He's on a mission.
Lying.
How do you know he's not asleep?
Good. It's a good question, Jimbo.
With the lights wake him up. You can see him.
How big is it? Can I zoom out a little bit?
I mean, he hasn't got eyelids, so presumably he can just turn his light, his eyes off, or his brain can.
He's got a little on off switch for his
So it's probably like 10 centimeters in length, maybe. But gosh, they look so much bigger. Like I just envision it being so much bigger than it actually is.
Do shrimp ever sleep?
I don't know.
It's cool, but I think we're ready to carry on. Goodbye, mantis shrimp.
Don't eat me.
Enjoy your.
One window.
That's just one window? You think he has a cut around? He has a front door and a back door?
skate pod.
Maybe he has a friend living in there.
Apparently shrimp do sleep.
I mean, it's got a nice little ledge,
Yeah.
little balcony.
Yeah, it is a nice.
He's set up good.
He's in the penthouse. The penthouse of rock caves.
Weird thought. What if the inside of this is totally close to that thought?
like an anthill, but of stomatopods.
Of course it's not, but it's just crazy to think about.
You never know what's inside. All burrows and holes.
Maybe it's carbonate, right? It's limestone. It's carbonate.
Got a urchin.
Just closed.
Oh, that.
Yeah, that one was open, this one's open over here.
Oh, yeah. There's also something coming out of that hole down there.
It looks like Pompeii.
It does. Oh, that's a good. So it doesn't have the mantle like wrapped around it. It just has it. That's okay.
So we just picked our points for tomorrow, and I'm pretty excited. We're literally going on the exact. It's almost like we crossed the top. We're just going to go down the other side and look for the east-west comparisons on the same feature. Should be pretty fun.
Cool.
See how this sets up on the opposite side.
Is that one of the tubes? Can we
Yeah. Yep, I
look
think
at
it is.
this?
The one on the right?
Yeah, on the right, on the left,
Either
middle.
one.
Oh, there's a bunch
There's
of them.
3
yeah.
or 4.
It's right around the right depth range too, where it was before.
No, we
Is
were
there-
about
how much
100
that-
meters shallower, but
I thought that forest went from 400 something to 300 something.
thought I went from 300 to 400.
Oh.
But
Yeah.
look at all the brittle stars, though.
I know.
Wow.
Ooh. Oh, we can get a brittle star with it, maybe. Some of those are living on there.
So this is actually the created sediment tube of a parchment worm. And we saw these the other day and collected one thinking it was some type of sponge. And then we got it up in the lab and realized there's actually a very beautiful polychaete living in here that creates this tube.
Pretty substantial in size,
Yeah,
too.
it's substantial in size and substantial, I guess, not fall apart. Like it actually took us kind of ripping at it to get into the sediment housing.
And was the worm in there?
Yep.
Yeah.
It was, but we damaged-
weren't expecting it,
Yeah,
so
we
we
were
did.
kind of confused with what we had.
Brian.
What? Oh yeah, absolutely.
It's like pretty big. So we'd like to collect this again and just put it into a quiver.
This time we know what
Now
we're looking at, and we'll try and get a full specimen of
I
the
think
worm.
last time you like scooped it. You remember?
Don't grab the bottom.
Hello, Freud's like, no, I'm out of here. pteropod
Forams?
shells.
Pteropods or forams?
I think they're pteropods.
Okay.
Whatever has been eaten.
Oh, snaggy snag.
That one was harder than that.
Yeah, this one's a little flimsy.
Yeah.
Well, we'll see what. It's like very smooth on the inside. The tube.
Does not look like it.
It is.
Now you have to.
There we go.
Do you want another one just in case?
No.
No, I think that's okay.
We'll be back at this depth tomorrow, so if we fail to extricate this one intact, we'll have another shot tomorrow. 489?
Yes. We're gonna have a full basket tonight.
Oh joy.
Okay. Yes, ready to go. Tim. Oh, sorry, y'all are talking.
Third side.
What are we, like halfway to 9?
Yeah, have
Tilt up a bit. So we should be getting into the land of green fish soon.
The land of
What?
green fish.
Pretty fish.
Yeah, there seems to be a band about this depth where a lot of the fish pick up a greenish tinge to their scales.
That's that little like fault line just all of a sudden got
Yeah.
covered in sediment right here. Here's a sediment line.
Wow. Very different looking sediment. Just like a sediment chute.
It's an avalanche path. Or shoot, yeah.
We're now above 500 meters, so we're at 475 meters. Do you remember what depth we were at when we came on, Brian?
I do
I
not.
don't either. But it was like 800, I feel like.
Look at you all.
Oh, there's a little fish. J-Rod's got eagle eyes. He says goodbye. Oh.
Hey, what you doing?
Look. It wasn't as awkward as the octopus that wouldn't leave Cody alone.
Just got my butt.
there's the
There's
first
your green
kind of green
fish.
fish.
That's the green fish you were talking about?
Well, no, it's several species. It's not a specific one. Just there seems to be something about the light conditions of this depth range that there must be something advantageous about that kind of green iridescent color. There's multiple species
Yeah, look, there's another one
that
right
seem
there.
to have it.
These little ridge lines are interesting. There's a redfish.
There's a scorpion fish.
Right about 10 centimeters, a little less. That's good. So many of those tubes. There's a very sad Amelsamia. It's a different fish.
It's the start of the little red and white sea star circle of sea stars.
So it's a conversation to have.
Shrimp. Those little ophiroids can definitely feel the vehicle.
This little forest of white encrusting sponges.
You want to take a look at one?
Yeah.
Please zoom on one of the white sponges.
Which one?
That one right in front of us looks good. There's a couple little sponges right here. You have the cresting one and then that little bulbous one.
Good thing from exit. That would be a magnet.
Sorry about the extra wind.
That's okay.
It is spelled wrong.
Is it?
Should be this.
Is that good, Brian?
Yep.
Okay, we're good to go. Oh yeah, there's one of those. Is that the sea star you're talking
That's
about?
the sea star I was talking about. That's not you looked up. That's the button name. That's not the field in the data.
What are you looking for?
You're trying to search for it.
I need to
For
follow the Ivories.
corallivory. It's the whole word with a capital C. Is what you search by.
Do you want me to look at that starfish?
No, it's
No.
okay. Now we're getting into more of this cavernous terrain.
Could you show me how corallivory is spelled on the button?
So,
Like anything coming on the new one?
No.
if I can log in, if you here, let me log into mine, and I can tell you what the event is because I'm editing ability. So, Tim, you want to sit here real quick?
Sure. Goodness. Lots of sponges.
Lots of sponges. Only started like 20 minutes ago. Too.
Yeah.
Is this fishing line?
Oh, yeah.
Yep.
Alright.
Some kind of fishing line.
Let's just see. It looks like it's on the ground.
Here comes our first spike fish of the dive.
Is that fishing line?
Where? Yeah, I think so.
Sorry, Tim, you don't have to stay there.
Here. No, it's okay. Take your seat.
You're more than welcome.
Sorry.
You're bumping into
Yeah. Sorry, I broke it. Yeah, I was trying to scan it. Alright.
Oh.
We got some scans, but
Yeah, I thought I saw something. Yeah.
I was trying to scan this whole. Yeah, the book took article and
Did it like shatter, or is it just a couple pieces?
there's still one really big piece, but a couple pieces shattered.
Okay. There's a sea star. Yeah. Kind of looked like you only had 4 legs. Lots of these encrusting sponges.
Yeah, it was like a light switch. It just turned them off and across the sun. I forget why I didn't see what depth
Yeah.
exactly, but we just suddenly saw came to that one rock with all of them, and they've been pretty continuous on any overhang or vertical since then.
We're not really seeing any corals, or if we are, they're really small.
We're gonna do a white balance, Jimbo.
Okay.
You was at home. Sorry. Too slow. Really?
Oh, some of those tubes were like in the sediment. Did you see that? Like, oh, never mind. They're just sitting on top. Other spike fish. Sponge land.
you turned the corals off.
I know.
It's okay, we don't fish instead.
It's like Steve left and they all said,
Jimbo really needs you to plant narwhal.
I mean, you know, I can't make miracles happen.
Well, I know.
Is that? You think that sea star might be eating the sponges down here? There's a bunch of them. What's he saying?
Don't worry, we'll just.
Wow, this is cool. Lots of those same sea stars.
suddenly a lot of those sea stars. You're right.
That's it, yeah.
Just keeps going.
So what is this
What's
telling
all
me about
this?
the
Wait,
some
can I
rubble.
just zoom
This
on
is-
the well, I want to look at the rubble first.
She wants to build the anticipation as much as possible.
Yeah, but this does not look like, this looks like shallow water
Yeah, it
coral.
looks like a crop. Mixed in with the polychaete worm tubes.
Yeah. Definitely does not look like deep water coral. So let's keep going, see what we find.
So what is this telling me about not having corals which past 20, 30 meters?
I don't know. It tells me it is not a optimal place for them to live, but I don't know why. There's a big urchin.
But there's been a pickup of sponges.
There has been a pickup in sponges, and a very specific type of that encrusting sponge, and those sea stars. Looks like a pencil urchin. That's good. Thanks, J-Rod.
You are most welcome.
But that, I wonder how old that shallow water coral is. That could be very, very old.
Yeah.
Remnants. Yeah, there's a lot of it up there.
Well, because if this feature is only 200 meters deep now.
Oh, here's a crab. It's one of these.
And a coral.
And a coral.
Crab looks like it's meditating.
It does look like it's meditating. He's like laying backwards.
Who's the crab?
Who's that crab? Like,
Maybe he's resting. And then we have a promnoid coral up here. Looks a little scraggly though, so we'll see if we get any more coral types of coral or abundance of coral as we move up.
It's like one of those little cutouts.
What little cutouts?
When I was growing up, like the houses I used to live in, so it was that little cutout.
Oh,
like that.
yeah,
What
yes.
was that?
Oh yeah, look at that.
What's that?
No, I'm looking at that on your
Look at all the
screen.
coral rubble.
Wow.
Pretty exciting on top of this.
It's kind of like what we were seeing with the,
There's a little cup coral.
yeah, little coral. With the madrepora. Wow, look at all that.
Must be something up top.
Well, there was.
Just a reef.
I mean, there was something up top.
Yeah. A long time ago.
Right. Oh, what's that? Oh, it's a hermit crab, yeah. Pagurid with an anemone.
There's some rare.
Hey Jimbo.
Yep.
Are you cycling on and off the CTD winch?
No.
It's come on and it's come up in the LC90 panels.
LCI90i, it just means it's moved fractionally.
Okay.
Yeah.
Wow, there's a lot of rubble.
Big.
Well, so this we think is old shallow water coral, so it doesn't look like deep.
Can you pick up a piece of it?
Yeah, we can.
Or zoom in? It almost looks like a prep row.
That's
I
what
was-
Brian
Yeah,
said.
that was my first thought as well.
Can we just sit down and?
Yeah.
Cool.
I got a little bit of a.
Oh, I see. You have a.
mean, it's uncannily like that, but it really is, you should be able to see. Whatever your screen's better.
So this is kind of a different rubble situation than what we had at the when we've been tracking rubble before. This looks like rubble from very old shallow water coral, which means I would think we would not find a source at the top. This would have been.
But I mean, on the order of geologic timescale, from this would have gone from a normal photic shallow water reef to this. This should have dissolved.
You know what? That's not a proper. Can you pick up that piece and just put it in for later?
Yeah.
That's like a tubicora or a.
Yeah.
It's like it has those long polyps, right?
Yeah, it's definitely.
Or like
but it's thick.
It's a big polyp.
Yeah.
Not a small one. Looks almost like more like a large Lophelia.
So like this right here, Jared, would be great. So you can have those polyps.
Stick it in like the rock box or something.
Yeah, the rock box is fine. We got a Christmas elf.
Hi, Courtney. It's Randy. It's so fun to see you on here. No, I think it's ancient. I think it's old and fossilized.
It was on Paracrypta for
It still doesn't make sense to me though.
Why not?
Because when it dissolved, it's not fossilized.
Oh, it's true.
And on the order of subsidence timescales, it's not going to have.
But a
The
bunch of
seamount
little
would not
baby
have subsided
parasites.
from 50 meters to
All
200
over
meters in the time it would take that to dissolve. That would have dissolved by now.
Well, I don't think it's grappa, except that, see that
Well,
piece over there that looks bumpy,
yeah,
but
I'm not
whatever
saying
it
it's
is.
a
Yeah,
grappa,
yeah.
whatever it
Close
is,
to
but
tip, I guess.
like, even if this is thousands of years old,
Yeah, I know.
I still think we're going to find some source of this.
I don't know. Well, the only way to find out is to go to the top. Luckily, that's where we're going. Courtney, can you try to imagine making a photo mosaic of this? Courtney's the one who wrote that photo mosaic paper. Awesome.
This one, the left page was on.
Oh, gotcha. Thought you were going to on the right.
Yeah, I had the right page on. Right page. That one.
That's not a bad suggestion. I would imagine this could have survived from a 10,000 years potentially from an ice age from a different sea level stand. It's totally possible.
Oh, it's bigger than where. There we go.
Yep.
Great. Thank you.
490, right?
490. Yes. Now we can keep moving up. Do you have like a ledge above you?
So Courtney, if you have any ideas, share it with us. Courtney's a shallow water biologist who is based in Hawaii, who works out in remote U.S. waters, including the Northwest Hawaiian Islands, and so it's great to have her perspective too. There we go. Crazy fish.
Oh, there's a squat lobster hanging on. Looks more of that sponge
Yeah,
we
more of
saw
that strange
before.
sponge.
Could we take a look at what's under that? Right under here, J-Rod?
Sure.
Courtney,
Or
that's
we
not
can,
a
yeah,
bad
we don't have to
call.
go all the way.
There was a ton of Clarigyra in the Mesophotic and Hippo.
That's that same weird tubey sponge that we
Yep.
saw on those big ledges on Baker. Looks like some form of, it's just such a strange growth form.
It is weird.
Yeah.
That's good, J-Rod, thanks.
So when we said it looked like worm tubes.
Yeah.
Yeah.
Was it?
Gotta,
So I don't know if you remember this, Alexis, Tam, but when we were on, I don't even remember which island it was, but we were using the little falcon, and we got to the top, and it was all that bubble swirl, it was all Clarigyra. It was Clarigyra. I don't, this might be a little too deep, but actually at the top, this might be the same depth. If this is all, if Courtney's right, this is all Clarigyra, then at the top could be a giant living Clarigyra.
Yep. Yep.
Pretty cool. We'll see. Something to think about.
wow. I mean, it looks like tubes, but I still think it's sponge. It's a little Anelopsamia sitting at the top of that rock.
More cherry in the top.
Oh, it looks like we got a bigger sponge and a coral. Oh, that's a big coral.
That is a big coral. It's almost like dogs.
I will come back.
There we go.
Wow.
That's what I'm talking about.
What's that? I'm a fly.
J-Rod, do you let me know? Do you want to control the camera? Okay. Well, there's fish
Look
living
at
in
the
there.
white fish in there.
What on earth?
Wow.
So I think
4,
this
5,
is
6.
this is zoanthids. This is not. This is a whole
10 of them at least.
colony overgrown by
I don't
zoanthids.
see anything else big in there. Yeah.
You've got any fish associates yet? Is this a
No,
new?
it's a rarity. No.
This is a new associate finding. What?
There may be nothing else in here.
But
There's all these fish in there.
right. Well,
But it's very specific type of fish.
Rather minor. I don't know.
It's
There's
zoanthids,
two types of fish. I think it's all the.
but I don't know. Oh, look at that! There's your dinosaur oaf.
No way.
Yeah, look.
It is. thrombus, right?
That stalk is so thick. Whatever this was before the zoanthids overtook it, it was humongous. Yeah.
Are these the same kind of white little pearly fish that we saw in the Pinoid forest?
I don't remember seeing.
No, they're different, but there's some similarities to them. There's little baby ones. There's a whole colony in here.
Unless you wanna look at that crab.
That.
Or
The oaf. Can we look at that? Yeah.
Crazy, Don.
Look at the density of the polyps on the main stalk.
Yeah.
They're all pulled in.
Yeah. On the main, on the.
See anything that's not pulled in.
Oh, there is a crab down there too. There it is.
There it is. Astrothrombus or something. It's so rare. I haven't. Can't remember the name. Yes.
It looks so prehistoric.
It is. It's a monster.
Yeah, you've got a couple crabs at the down here at the base, but not
And a
up around
sponge
where
right on the
the
base.
yeah. Look how thick that base
That's
is, though.
huge. Amazing. Wow.
Is that good, Tim?
It's good.
Okay.
You gotta come up.
Yeah, that's.
And then I'll come back.
Wow.
And so most of those polyps have now closed up since we've arrived.
Do we scare all the fish off? Are you still in there, tucked in the bottom?
Looks like we definitely scared a few off.
There's still some down at the bottom.
Yeah. Is there anything else here that you guys want to look at on this coral?
No. I just want to see if there's. It looked like there might have been another one. I kind of want to see if it also has fishes or if this is.
There was some more around the other corner when we go.
No, there's one straight down, I think. Or if you,
One of these?
yeah.
Something similar directly beneath it.
I'm not sure it's the same one, but
Neither
it is
am
a
I,
pretty
but it
big
was something
coral.
down
I think
there.
it's a bamboo.
Yeah, it's right. It's different.
It is different. Wow, it's extremely thin.
Yeah. That looks like.
It's starting to get overgrown though. That's some of that same zoanthid near the base.
It's definitely a bamboo coral. Yeah, there's some of that zoanthid going along those branches.
Different oaf or dinosaur oaf.
Is it different oaf in there?
It's a different one. It's cool. It's really.
What is it?
No, I don't know.
All right.
Did you wanna look at the other ones that I thought I saw?
Yeah, why don't, yeah, if you think you saw them.
It's cool. I've seen fish like that. Yeah, we always talk about fish and coral
I know.
partnerships and how
Yeah.
it matters to the fish, but
That's one of the first instances I've
Yeah.
seen of fish actually
yeah.
mingling inside. Oh yeah, there you go. J-Rod knows what he's doing.
Where are we now? What happened?
We're gonna look into at another coral to see if they're fishing it too.
There's,
I see one.
yeah, two.
Two?
Yeah.
Three. Yep, you're right. There's a little baby one too in the top.
Look at the size of this.
Is it just sitting,
Five,
is it
six.
sitting on the branches?
Oh look,
Oh, there's
Tim,
a
you've
wait.
got
There's a yeah.
you've got those oafs in that in
Might be a
Alsamia.
dinosaur oaf on that.
Wow.
I think there's two of them.
Are they lying on the coral? We really gotta.
The fish. Yeah, they're like
they're
perched.
definitely they're nestled in there.
They're just
That's
sitting
home.
Okay.
Those are all zoanthids too, right? Or not?
Yes, they are.
Can we get some focus on the fish?
Just give me a second. He's working.
Yeah, let him.
Okay. Get a good shot of it.
Yeah, I don't recognize the fish either, but we're shallower.
They do. Look at him. He's like nestled in. There's another big oaf down there. These coral. Yeah.
Massive.
So is this the
I was gonna say, is this the rubber we've seen?
Kilamoana thing?
Yeah, is this
This is.
gold coral? Like
To me, it is because look, this is.
Right there.
This is
That
what it's
massive.
doing to
Is
the
that-
skeleton.
Is that what the zoanthid is?
No, I'm asking if this is the debris. This is
Oh.
what we just sampled.
There's a piece of debris lying right there.
So I don't think that this is the debris, Tim.
Okay, that's what I was wondering.
What Brian and I are asking is this
The
the
zoantid.
so the actual gold coral? What? How do you say
Oh,
it?
I see.
Kula mana mana.
Yeah.
Wow, it's massive. We're at 412 meters, 10 degrees Celsius.
Probably be a range expansion if it was, no?
Yeah. Might go big.
That is a massive.
That's what I mean, like
That is
the
ridiculous.
size of whatever it was before it was
Have you thrown laser
covered
on it?
in the zoanthids. What's that down there? Is it just a hole?
I was gonna ask, is that a hole or something on the floor?
There's the lasers.
Oh, it's a rock. I think it's a hole.
Lasers are on that little tip. Good God.
Oh, jeez, yeah.
That is a monster.
I mean, just the thickness of the base to me is
We're trying
amazing.
to get a better view of it from the other side.
Okay.
At least
It'll be lit up better.
meter and a half by 2 meters.
At least. That's conservative. Yeah, I think 3 a long ways.
Wow. Learn something new every day?
Cool. Yeah.
Every minute? Every day?
Yeah.
Do you want to pick up that dead piece?
That's really big.
It's really similar.
Yeah, it does. It's especially with the density of the polyps on the main branches,
Yep.
you know? I mean, I would think it's worth sampling so that we could, okay.
You think it's the same thing?
We think it's worth sampling so that we could have a,
Yeah.
we could try and confirm if it's
Mystery.
A clip on
This one or that one?
either one, whichever you prefer.
I'll start with the roof right now.
Is she out?
She's gonna work in the lab and I'm gonna do. Is it okay?
Yeah.
I'm gonna, get a couple of what happens to monsters.
Huh?
No, not right now.
Yeah.
Very
I'm not
good.
thinking that was coming down low enough to do
So we don't think that's covered in zoanthids then, if that's right?
No, that is zoanthid covering a base. Kulamana is a parasitic zoanthid.
It's just it's a
Oh, I see.
species of zoanthid that is known to put down this gold. It's called gold coral because it puts down a gold
Right.
skeleton itself
Yeah.
over.
Super prized to jewelry making.
It's protected.
Yeah.
Yeah.
No, it actually doesn't look like a range extension. They're documented
Oh.
this far out. There may have been
I
some
was gonna
here.
say, is that us?
Yeah.
I think it's still worth collecting
I agree.
these.
You may look around on that.
Okay. I want to look at this interface where you can see the skeleton. Barnacles. Oh, there's a little sea star down there.
Is it?
Yeah, look at it on the base.
And we're right in the peak of its depth range too.
Oh, are we?
Yep.
You want me to?
It's okay for right now. I think we're still. I don't see. I mean,
It's
we.
different. It's Circeaster, though. It's interesting. Sea star right there.
We had the.
Can you find the fish?
No, I think we. Well, there's some over here.
Get a good zoom on the fish so you can see
Okay.
everything. Look at the barnacles. I don't see much of the other associates.
No, other than the fish.
Just a small.
All right, are we ready to sample? You say no?
Yes.
Okay.
Yeah.
You want the dead stuff or you want live stuff?
Live stuff, please. We don't need a big piece, just a piece to go in the quiver. So wherever is easiest to reach. Okay. I'm more interested in what that coral originally was, honestly.
The species of Zoanthid was only described in 2013.
Oh, I didn't know that.
Yeah.
I didn't realize
It's
it
a
was that
relatively
recent.
new discovery.
This fish is like, I want to go home.
I think you'll have options with your arm.
We just need a small piece to go in the quiver. So wherever that is easiest for you.
After you grab it on the move, so you can still do you need a better zoom in on any of this stuff while it's right up front? You can take a grab and then you can do it.
Okay. I mean, I think we're pretty good, but might as well.
Might as well, it's all right. That look good, it's all right.
Like that?
Yeah, that looks great.
Cameras.
Okay, we'll just quickly come in real close on these live follows. Or, I mean extended, they're all live. More zoom. Wow.
That's maximum effort.
Max of max zoom. Okay, I think we're good.
That's fun. One of the those.
Right?
491. Is that what you said?
491. That's
Yeah.
not at all what I said. But it's what my brain meant.
Great job. Well done. They're definitely going to break 5,000 samples by the end of sampling tonight.
See, there you go with the same thing. Your brain and your mouth aren't connected like mine are.
Did I say?
I thought you said 5,000.
She did say 5,000.
What have y'all been doing while I'm gone? I've been doubling up sanding in the covers.
500. Yes.
Oh, we can use those?
The ones that are not ready.
I think there's 2 or 3.
Did you guys see
The oafs.
the order for the now salmon?
Yeah, I was gonna
Correct.
go
Okay,
back.
there's-
I was gonna ask to go back.
No, there's one on.
There's
There's
right
one
here.
Yeah, it's just
So
over
there's
the
a
ledge.
small
Yeah.
yellow coral right below, right where your foot was. If we can look at that coral, there's
In like the most
opaloids on there.
difficult place to access.
Yeah. You can almost, no, can't see it.
Oh, doggy, we're gonna be up late tonight.
Y'all gonna go party? You invite us.
You wanna come party in a wetland? Give me a real doozy.
I'll believe
Gonna be a long night.
We're all doing-
Thanks, man.
A long morning. Thanks. I'm gonna watch it a half hour, but,
There we go. See that down there?
Yeah. Are you gonna want a sample?
Tim.
Well, if you can get into that, right? Is it open below?
But yes, you would like the dino or no, if
Yes,
it's possible.
I'm sorry.
If it's at all possible, we'd like to
If it's
sample it,
possible,
but
yeah.
if it's not possible, that's okay.
Yeah, not at the expense of the big coral.
I'll do that.
Or at the expense of the ROV.
Let's see how sneaky I can get in this already.
RV can take a little more damage than the coral. I mean,
I don't know. Coral's a beast.
little Marlin's coming out to see what's going on.
He's like, oh, nope,
nope,
never mind.
Never mind.
Yeah, this one, I don't think you'll be able to reach down that far. It's almost across the zone.
It's too high, yeah.
Yeah.
That's very good.
We're staying.
I don't know that you're gonna get, I can try to rotate a little bit.
Let me just come.
You
Let's
want to see a little bit of side view?
let's see this first.
Let's just snag
I
and
won't
go.
be able to get the left side, I can get your right side.
That's fine. We don't. The right side would be the side that we would want to see.
I'm gonna slowly start coming around.
Okay.
Astrostegus.
Astrostegus. Astrosaurus.
Astrosaurus. That's a great name.
It is.
You wanna come around more?
If you can.
They're coming around all the time, that's just fun. Gotta really like 1 degree at a time.
Yeah, that's fine.
Maybe we should just grab it.
Alright, hold torches.
There you go. Okay. I think we can sample that.
It's a hard one, right?
It is. It is a hard coral, yeah.
So it's gonna shatter.
It'll break off, but
It's
we
gonna hold a lot of it.
if you come in and try if you grab the